Right, quickly, go ahead and start. Good morning, I'm Mark McClellan. I'd like to welcome you to today's event sponsored by the Duke Margolis Center for Health Policy. It's a public workshop on oncology clinical trials in the presence of non-proportional hazards. And we're very pleased that you all could be with us today despite the frigid and icy weather here in Washington. This is a very important topic. We're convening it under a cooperative agreement with the FDA, and we have together leading experts in government, industry, academia, and advocacy for a productive exchange on what sounds like a technical set of issues, but has very important implications for the design and interpretation of evidence related to new cancer therapies. As you all know, the pace of development and approval of new cancer treatments has been steadily increasing, leading to a much more significant range of treatment options available to patients and the potential for even further transformations in oncology care. However, there are ongoing and significant challenges with oncology trial designs and analysis of treatment effects for cancer therapies that may impact our understanding and thus the use of currently investigational drugs. So today, we're here to talk about some of these specific challenges and potential solutions. During today's discussion sessions, we're going to hear from a number of industry, FDA, academic experts who have been engaged in a cross-sector, pre-competitive working group to address some of these important issues related to the presence of non-proportional hazards. The goal over the past few years of this effort has been to better understand how non-proportional hazard ratios may impact traditional trial design, analysis plans, and thus, again, the interpretation and potential use of cancer therapies. This group set out to create and test new methodologic approaches to better characterize treatment effect when such non-proportional hazards are present. And today marks the first public presentation of their efforts. And our assembled panelists and participants are going to have time for feedback on the progress, on the remaining barriers to improving these types of methods and their interpretation in oncology drug development. So very important milestone in progress on addressing the significant evaluation issue. The open collaboration between FDA and industry to improve treatment development and impact lives through approaches like this is an important reminder how we can better address systemic issues in drug development together and with transparency. So we really appreciate the group's work to date and look forward to today's further discussions and potential next steps. For the agenda today, we're going to begin with perspectives of our colleagues at FDA, including some recent examples of cases where regulators have run into challenges with issues related to non-proportional hazards. Then we'll hear from the co-leaders of the Cross-Pharma Working Group, who will provide some background on their formation, their charge, the work that they've undertaken. Subsequent sessions are going to focus specifically on the work the group has done to date. Session 2, in particular, will review challenges with current statistical approaches used in the presence of non-proportional hazards, the development of alternative approaches, and simulations to show the value of these new approaches. Session 3 will use case studies to demonstrate how the proposed alternatives could have been applied in real clinical trials, as retrospectively, and how they may be more appropriate than current statistical approaches. And Session 4 looks forward. Session 4 will discuss how to design future clinical trials using these new approaches. We're going to close with a session to give us a chance to discuss the potential path forward. Our goal by the end of the day is to have a wide-ranging synthesis of the workshop's key takeaways from the range of perspectives represented here, as well as a chance for panelists and meeting participants to offer their thoughts on next steps for addressing the presence of non-proportional hazards as effectively as possible. And so we're very much looking forward to the planned discussion today, which will get into some technical details, but ones that have very important practical implications for study design, access, and use. Before we get started, just a couple of housekeeping notes. You can see from the agenda that we're going to begin with one or more formal presentations followed by a panel discussion. For each of these sessions, what we'd like to do is ask the lead speaker or speakers to come up, give their presentation from here at the podium, and then when it comes time for the panelist discussions, we'd like to ask the panelists to join us here on stage as well. Some of our panelists will also be presenting slides, so we'll go through that as well. In addition to their perspectives, we've set aside some time for broader discussion, feedback with audience participants. For those in the audience, we have microphones in the aisles that you can use throughout the day, so either end here. So we want to make sure we hear from you all as well. This is a public meeting. The event is being broadcast online. I'd like to thank everyone who's joining us online today as well. So keep in mind that everything you say will be part of the record for this event and will hopefully be available to help make further progress on addressing these important study design issues. For those of you who are joining by webcast, we encourage you to participate as well. There are two ways to do that. If you have a potential question for the panel, you can send them to margolis.oncology@duke.edu, margolis.oncology@duke.edu. Our staff will get them up to me or whoever else is moderating at that point to get those comments or questions in the discussion. For those of you who are in the room today, feel free to, again, participate via the microphone. I guess you can send us questions online as well, margolis.oncology@duke.edu. For those of you who are here, there are coffee and beverages located just outside the room. Please help yourself. Lunch will be on your own today at any of the restaurants close by. There are plenty. And then finally, just a reminder that although this meeting is being convened under a cooperative agreement with the FDA, this is not a federal advisory committee. We will not vote. We're not going to get to formal conclusions or recommendations. The meeting will be a success if there is an exchange of ideas and open discussion about the potential paths forward. So that's an overview of what we're aiming to accomplish today and how. We're now going to hear a few opening remarks from Gidon Blumenthal, the Deputy Office Director of the Office of Hematology Oncology Products at CDER and the Associate Director for Precision Oncology in the FDA Oncology Center for Excellence. He's going to provide some further context for today's discussion and an overview of the FDA's goals for the meeting. Thanks, Gidon. Good to see you.
Thank you, Mark, and happy Monday. I hope everyone had a good Super Bowl viewing for those who like to view the Super Bowl. Congratulations to all you Eagles fans out there, and sorry to the Patriots fans. It's great to be here with all the statistical colleagues. Not a lot of clinical folks. We see a couple in the back of the room, but hopefully we'll have a robust discussion. So I also wanted to thank Duke Margolis for hosting the workshop. It's been a remarkable time in oncology. In 2017 alone, we approved 17 new agents and over 30 efficacy supplements. We've seen a remarkable inflection point in the approval of novel agents, both immunotherapy agents, anti-PD-1, anti-PD-L1 agents, CAR T therapies, targeted therapies, a lot of new companion diagnostics, next-gen sequencing tests, and other biomarkers in the pipeline. So we've seen more of these immuno-oncology trials read out with non-proportional hazards. While this is not a new thing, and we've seen this in older oncology trials as well, we are potentially seeing more of this. This could be due to delayed effects from the immuno-oncology agents, maybe mixed and heterogeneous patient populations, with some patients responding quite well to the IO, but unfortunately, most of the patients, particularly in sort of these all-comer trials, don't respond and rapidly progress. Also, reassuringly, and we saw this with one of the first immune checkpoint inhibitors approved, ipilimumab, there's a potential long tail durable responders and non-responders. So we have to think about how to factor in a potential, dare we say, cure or at least durable survivors when we're thinking about trial design. So today we'll discuss the impacts of non-proportional hazard on trial design, how to size and power the trial analysis, the timing of analysis, including interim analysis, the best analysis method in the presence of NPH, and how best to convey the study results both to patients and to physicians. I think most practicing oncologists don't even know what NPH is. So needless to say, patients wouldn't know either. So how do we convey hazard ratios, which is challenging in and of itself, but also, medians are easy, milestone analyses, landmarks, et cetera. Is the hazard ratio median as a summary statistic, is it always the best way to describe results? Ideally, we'd like to come to some consensus on the methodology, where in a pre-specified statistical analysis plan, you could adapt from a conventional log-rank test to another method in the presence of NPH. One key, and Dr. Pazder actually stressed this at ASCO last year, we wouldn't want to see a huge penalty in terms of type 1 alpha spending if you had to convert from a traditional log-rank test. So these are some of the issues we wanted to discuss today, and I'm looking forward to the discussion. So with that, I guess I'll turn it back over to Mark to introduce session 1. Thanks.
All right, thank you, Eden, for that helpful context. So now we're going to start off this session on perspectives on potential limitations of statistical plans based on the assumption of proportional hazard over time with 2 presentations. We're going to start with a presentation from Rajeswari Sridhara, the director of the Division of Biometrics 5 in the Office of Biostatistics at CDER, who's going to provide an overview of the current statistical methods used in oncology clinical trials and, again, the challenges FDA has observed with the use of these methods in heterogeneous populations and patterns of effects that may not be proportional hazards. Then Renee Iacona, the therapeutic area head for oncology and immuno-oncology at AstraZeneca, is going to give a presentation on behalf of the Cross-Pharma Working Group describing the industry's experience with these current statistical methods and their objectives in forming the working group to address those challenges. Then we're going to hear from a reactant and have some time for discussion. But first, I'd like to turn to Rajeswari.
Good morning, everybody, and thank you for coming to this workshop. I would like to thank Mark and the Duke Margolis for putting this together. Particularly Morgan and Meredith have done a tremendous amount of work, and also our industry colleagues who have worked very hard on this for the last couple of months to make this happen today. And of course, the panelists and the participants who are here. So I'm just going to set up the focus of today's meeting. Time to event endpoints are the ones that we normally use in oncology clinical trials, as all of you know. So these are the endpoints when we mean by time to event endpoints: overall survival, progression-free survival, recurrence-free survival, disease-free survival, etc. In designing these clinical trials, the most common characteristics that we have are to fix the chance of false positive conclusion, which is the alpha that we talk about, fix the chance of winning or detecting a benefit if it exists, and that's the power of the test that we talk about, and define what treatment effect is meaningful or alternative hypothesis to the null of no effect. And this, of course, comes from the clinicians and the patients as to what would be a meaningful difference in the treatment effect. And in all of these, or in majority of these, we assume that the relative treatment effect, which is the hazard ratio, is constant over time. Now, assuming this constant over time, the comparison of survival curves use log-rank test, which is a non-parametric test, and an estimated median is also reported using the Kaplan-Meier estimates, where on an average we are reporting that 50% of the events were observed before the median time. We also test the hypothesis whether the two treatments, the relative treatment effect is null or there is an effect using a Cox proportional hazards model. And the hazard ratio provides an average relative effect over time. However, it is well recognized that the power of the testing of the hypothesis reduces as relative effect changes over time. I have a few examples here to show what we mean by this. So a constant relative treatment effect means over the time, whatever is the difference remains the same throughout the time. So you have on the y-axis the hazard ratio, on the x-axis the time. And so what we are looking at is the red dotted line, that's the null hypothesis of no treatment effect, or there is an effect, which is the green line. So the benefit is as the hazard ratio is smaller and it gets worse, that is, the treatment is worse than the control if it is in the opposite direction. However, what we see is sometimes this is not true, or when we observe the events, that there is no difference initially between the two treatments. However, you start seeing differences after some time. So you see this green line where the treatment effect you don't see in the beginning, but as time advances, there is an effect. So here we say the relative treatment effect is changing over time. Or we also see sometimes where it is worse at the beginning and better afterwards. Or you could have different patterns where it is worse, gets better, and maybe there's no effect or close to no effect. So here are some examples to demonstrate what we mean by this. In this survival curves of cisplatin plus fluorouracil plus or minus taxotere in head and neck cancer patients, you see that there is a constant effect of taxotere over the control arm throughout the time period that's followed. Now let's see some examples where treatment effect is changing over time. You see the nivolumab example where in non-small cell lung cancer, in melanoma, where you have the curves are all bunched together at the beginning, and then you see that the curves are separating. Similarly, here you also see in pembrolizumab where you don't see a difference at the beginning, but you see a difference later on. So we have termed these as delayed effects, and there is a changing over time, no effect at the beginning, etc. The reason for this has been speculated that there's a pseudo-progression or a delayed response for these PD-1 products. Now here is another example where you see exactly the same pattern, but by design, the comparison, you have a Revlimid plus dexamethasone for 18 months in one arm, and in the other arm, the blue line, it is continued, Revlimid is continued beyond 18 months. So you don't expect any difference between the RD18 and RD continuous for 18 months because it is exactly the same treatment both treatment arms are giving. So the reasons for this separation of delayed separation could be different in different scenarios. Here is another example where it's not quite visible, but there is a crossing at the beginning. At the beginning, the treatment arm, which was the tivozanib, and the control arm, sorafenib, tivozanib was doing slightly better, but then later on, the sorafenib arm was better. Here, the reason, at least from the sponsor side, as they explained in the advisory committee meeting, by design in the sorafenib arm, after disease progression, patients could get tivozanib, whereas in the tivozanib arm, they were not getting sorafenib, or these were the countries where sorafenib was not approved for second line. Now here is another example, nivolumab, where you see that the nivolumab was not doing quite well, somewhere in between. There was no difference at the beginning, a difference where the treatment was worse than a control, at least in the graph, but later on you see that there is a prolonged survival advantage, PFS advantage. And this is a classic thing in many of the surgical procedures. You see this, the allogenic transplant, where you don't see an advantage at the beginning because of the GVHD, graft versus host disease. But in the long term, it is better. So we have to think about what is it that's causing these crossing of curves. Here is another example where there is a difference, a bigger difference in the middle, but not so much at the beginning or at the end, but all along the treatment is above the control arm. This is in mesothelioma where pemetrexed was given with cisplatin. And we do see some others also where even though it is better, the problem with the median reporting is conveyed very well in this one, where the curve pinches somewhere, and it's quite possible it pinches right at the 50% or the median. And you see that the median difference is just 2 weeks or 0.5 months, whereas the overall hazard ratio is significant in this case. And this is not an issue that it's not that it is non-proportional, but you do see this different patterns. And here is another example in gastric cancer where, again, the medians come closer, but you do see that there is a separation of curves, especially at the end of the curve. So the challenges are when the assumption of constant hazard ratio is not true, the Cox proportional hazard model is inappropriate because it's telling us that right from the beginning you have the same relative effect, whereas that's not what we are observing. And the KM estimate of median survival may not be an optimal measure to summarize the results. So our problem is, what is the optimal analysis method to test treatment benefit, and how can we summarize these benefits when we see these different survival patterns? Many methods have been proposed in literature. Each have advantages and limitations, and multiple approaches may be necessary to summarize these results. So we initiated we recognized that there is a need for collaboration, and so initiated a dialogue with the industry statistician, cross-pharma industry statistician, and we met in 2016, September, I believe, and subsequently in 2017 at ASCO, etc., and concluded that a methodical evaluation of available methods is needed. And the goal was to identify appropriate analysis method for different patterns of non-proportionality that we observe, and all industries to work together as a team, and this is non-product specific, and FDA to participate in this effort. So why are we here today? So the current practice of using log-rank test and Cox proportional hazards model may not be the best when we have these non-proportional hazard patterns. Reasons for observed changes in treatment effect over time may be different in different trials, as I showed in my examples, and we have to keep that in the background. What is the best way to evaluate treatment effect? What is the best way to summarize an observed treatment effect? And working group will be presenting the work that has been done so far in this area. So the workshop goals are to discuss merits and drawbacks of these different analyses that are going to be presented today. Are the alternative measures of treatment effect clinically meaningful? Currently, how do clinicians explain change in relative treatment effect over time to patients? Or how do they understand? How do payers interpret the results when the relative treatment effect changes over time? And would the new measures be more or less useful? And how should we design future clinical trials? So this is what is our goal. And as Mark mentioned, this is not an advisory committee. We will take all the expert input, and probably in the future we may have an advisory committee meeting where we may discuss some of these again. So in the first session is on the limitations of what we have today. The second session is about alternative analysis methods with simulation results. The third session uses all of the methods in case examples and present the pluses and minuses of these methods. Session 4 is about the design of studies, design of future studies, and the last session is the most interesting one to understand the stakeholder expectations and implications if we use some of these alternative methods. Thank you very much.
Okay, next, as I mentioned, Renee Iacona will present on behalf of the Cross-Pharma Working Group on experiences with these current statistical methods.
Good morning. I am here to speak on behalf of the Cross-Pharma Working Group that has focused on non-proportional hazards. I am one of the co-leads for this working group, but my other co-lead, Tai-Seng Chen, is here with me as well, and he'll be sitting on a panel later this afternoon. Let's see if I can figure out how to, there we go. So just as a reminder, one of these disclaimer slides, I'm a employee of AstraZeneca, but these slides really represent the working group versus my company alone. And so I'm going to be a little bit repetitive, but hopefully say it in a little different way that our non-statistical colleagues will get the background of what we're talking about today before we get into the technical details in the next sessions. Raji has very much explained the issues that we're facing today, looking at the issue of non-proportional hazards when we see it in our trials, and has explained to you that the methods we commonly use, the log rank test and the Cox proportional hazards, really do assume that we're in the presence of proportional hazards, and in this case we're not. So then what's our best alternative? So this has gotten a lot of attention recently because of immuno-oncology. And these are some of the examples. Some of them also Raji presented earlier. But I want to share with you, this is actually not new to oncology itself. It's actually not even unique to oncology. There are examples of non-proportional hazards being seen in cardiovascular trials. There are also examples in vaccine trials, as well as the transplant example that Dr. Shidara shared earlier. But I want to share with you to make sure we set the scene for the types of non-proportional hazards, because we're not just talking about the lag effect that we commonly see in immuno-oncology. So the delayed treatment effect that Dr. Shidara explained is the one on the top left, where we do see this lag of the curves separating. This lag can vary in amount of time. We've seen it in 3 months, but in most recently examples, we've even seen it to be longer than that. But it's potentially important to know if you're going to have that type of lag, and I'll talk about why that's important later. We also have this example of diminishing effects, which is the example that Dr. Shidara shared, where the curves may separate sort of in a belly effect. We call it the belly curve, where you have this sort of split in the middle, but the beginning of the curves and the end of the curves come back together. Finally, you have crossing hazards, where the curves actually cross over each other. And gefitinib, a one close to my heart, the IPASS study was an example of crossing hazards. You'll be seeing that trial again later today in our case study example. But this is where AstraZeneca at the time didn't know that the EGFR mutation was driving the results of this trial, and the curves actually crossed over in this trial results. And then finally, the one that was referenced earlier by Dr. Blumenthal was the long-term survival, where you may see this sort of extension of curves towards the end of the curves, where one curve may extend out longer while the other curve may begin to go closer to one. And the subgroup driven may also show itself, but we're going to assume for the rest of the day that, like the IPASS study, if you're designing a new trial, you don't necessarily know what that subgroup might be that's driving your results, because if you did, you would probably design your trial differently. So why is this important? And this is where I hope the people in the room will help begin to understand, between Dr. Shidara's presentation and mine, that this can impact both your trial design. It can impact the size of your trial, the timing of your analyses. It can impact the power of your study. It can impact the timing of when you should do your analyses, whether that apply to your interim analysis or whether that applies to your final analyses. And as Dr. Shidara just shared, it can also impact which methods should you be using. Given the log rank and Cox proportional may not be the best methods, one of the focus of the working group is, if not that, then what? What method should we be using? And finally, as Dr. Blumenthal said, how should we be describing these trial results? Dr. Shadar has just shared that the medians may not be relevant because they don't exactly describe the result of the trial. In the same way that the hazard ratio has assumed proportional hazards, perhaps the hazard ratio is also not the best way to describe the trial. So these are all parts of the focus of the working group. So I'm going to share with you just a few examples so these land with you before we go into the technical details. So from a design perspective, these two plots show you a difference between a trial that has a slight delay and a little bit of a longer delay. And over time, you can see that the drop in power will occur if you analyze the trial and you analyze it too early, then you could be seeing less power than you thought you might have designed the trial with if you didn't know about that delay. It also may mean that the timing of your analyses, increasing the sample size of the trial, may not help the power of your study. It's really actually about the timing of your analysis and making sure you have enough follow-up on patients so that you are hitting that curve at the right appropriate time so you're seeing the treatment effect difference you're looking for. This also has an impact on futility analysis and whether or not we should be looking at all patients in a futility analysis or a set of patients in the futility analysis who have a certain number of events that may represent patients who have long enough follow-up at your futility analysis. Because if you assume everyone in the futility analysis, naturally some of those patients may not have seen an event because the curves have not separated yet. So, if not log rank, then what? And as Dr. Shadar mentioned, there's lots of methods in the literature. Some of these beyond log rank, you often see restricted mean survival time. You see weighted Kaplan-Meier test, you see weighted log rank. But which one to choose, given there are many methods you could be looking at? So one of the purposes of the working group and one of the main sessions you'll be hearing about today is what are those methods? And perhaps how do we compare those methods when in the presence of these different types of non-proportional hazard? Finally, we're going to try to begin to touch on how should we be interpreting these trials? What's the best way to describe the results? As we've talked about, hazard ratio and median may not be the appropriate measures. Then what, if not, then what? So some have considered hazard ratio over time, a piecewise hazard ratio, a restricted mean survival time, and some also have talked about a milestone, perhaps perspective. A milestone here means like overall survival at 12 months, overall survival at 24 months, as an example. So here's an example of a trial from non-small cell. It's an immuno-oncology example. But just like the examples that Dr. Shadar shared, the hazard ratios here are not constant over time, so clearly we're in the presence of non-proportional hazards. But also, if you look again at the medians, like Dr. Shadar's example, the medians may not represent the true results of this trial. So if not, then what should we be using to report these results? So, just as Dr. Shadar shared, these have been existing. Obviously, with the IO coming along, these have become quite predominant issues that we needed to address. So, in September of 2016, she asked a few of us that were attending the conference here in Washington, D.C. to come together and see if we could work collaboratively. Many of us had actually worked in a prior collaborative group looking at progression-free survival. So we knew this actually could work, this sort of concept of collaborating pre-competitively. And this is a very complex issue, so we knew it would take several of us to work together to try and resolve it. So here was the purpose and the objectives of the working group. We are a group focused on oncology and immuno-oncology. We recognize that this has occurred in other therapeutic areas. We did go look at examples from other therapeutic areas, but we focused here on oncology trials. We did focus primarily on phase 3 trials, but you're going to see some examples of phase 2 trials, because in oncology, sometimes those phase 2 trials can also be registration trials. We focused in on the regulatory space. We're not going into the payer space yet, but later today we're going to hear potentially what the impact of this work could be in that space, as well as considering what's the impact on interpretation for physicians and the patients. So the audience, we knew that eventually it can't just stay within statistics, and so today is our first great example of trying to share the results more broadly and get impact and understanding from the audience, including clinicians, regulatory body members, as well as patients. And so finally, as I mentioned earlier, our assumptions were that in your trial designs going forward, If you knew you had a subgroup effect, you wouldn't have potentially designed your trial. You would have known what was driving your non-proportional hazards issue. So for the rest of the day, assume that we didn't know this prospectively going forward. There were two substreams of the working group. One was looking at the design and analysis, and another looked at issues related to endpoints. These were just the original objectives of both of those work streams. Today, the work from the left side, the design and analysis, is the focus of today's entire working group presentations. The purpose and our goal is to have a working paper or set of papers, and we're working on drafting those now with the idea that following this conference, we'll take feedback we receive to go back and adapt and follow on next steps with Dr. Shidara's team. So basically, here's how the left side worked, the design and analysis team. We identified several studies that we felt were quintessential examples of non-proportional hazards, of the different types of non-proportional hazards like I shared with you earlier. From that, these example trials, we digitized the Kaplan-Meier curves and came with a set of data from those digitized curves, and we're able then to, our simulation working team was able to draft the data sets, provide them in a public space that we were all using the same data sets. We came up with code that was shared, so we were making sure that differences we might see when applied didn't come from different coding. We then were able to apply the different methods to those simulation sets. And you're going to see Dr. Hugh from Lilly present later today the simulation working group's results that led us to testing different methodologies. You're going to hear about the different methodologies from Satrajit. And you're going to be able to hopefully to take away what this working group focused on in the first few stages, basically taking simulation results, determining the best methods. Then we took the best methods and we applied those to actual example trials retrospectively. And so session 3 will be presentations from the working group where we've applied the best methods that we believe should be used in the presence of non-proportional hazards. And you're going to see some case studies presented back to the working group, back to you, the audience. So we're going to see examples of the different non-proportional hazards from the IPASS study, as I mentioned earlier. We're also going to see some examples from IO as well as a HEMS trial. So the methods evaluated were considered in 3 different buckets, and Satrajit will explain these later. First was the rank-based, obviously looking at comparing the results, the methods, back to the log rank test, since right now that is our method that we use consistently. We also looked at Kaplan-Meier-based tests, so some of these examples would be the weighted Kaplan-Meier restricted mean survival time. But we also looked at tests that combine tests, and that's probably going to be where we get into complicated methodology, but sometimes perhaps applying 2 tests might be better than applying 1 test or a combination of tests. So I finally want to close and say that doing something like this takes a lot of effort. And so there were many companies represented in this working group. So the companies are listed below, but it doesn't represent the number of colleagues from all of those companies that have done this in their daytime jobs, but actually at night in their spare time. And then I'd really like to recognize the leadership team who have spent weekly conference calls leading up to this session, but have been working over the last 2 years together quite collaboratively. And as I mentioned, you'll be seeing most of the leadership today presenting in the different various sessions. We're going to hear from Satrajit and Tianli on this methods and the simulation results first. Then we'll hear from the case studies with different members of the working team. And finally, Dr. Anderson from Merck will be presenting on, okay, so now if you know about these methods and you know you need to design a new trial, how would you go about designing your new trial, whether you know you have non-proportional hazards or not? To try to address what Dr. Pazder's advice was us at ASCO this past year, where he said he didn't want companies to be penalized if you design a trial and you don't know whether you have non-proportional hazards or you don't, then how do we design a trial and prepare for the potential that you could have non-proportional hazards? So Dr. Anderson will end the day talking about that. And then we really are looking forward to the feedback that we will receive today. We are sort of a working group in motion where we are going to take today's feedback, and that will definitely impact the guidance papers that we're working on and the next steps for both us and our partnership with the FDA. So hopefully I've explained and set you up for the rest of the day, and thank you very much for your attention.
Renee, thanks very much. Also, thanks to all the members of this collaborative working group for the hard work over the last 2 years. As Renee said, you're going to hear from many of them during the course of the day. Right now, I want to turn to Mark Thierat, the Acting Associate Director of Immuno-Oncology Therapeutics in the Oncology Center of Excellence at FDA, who's going to provide some perspectives from both a regulatory and clinical standpoint to help us tee up the discussions for the rest of the day. Or maybe I don't see. Mark's stuck on the ice. Right. Okay.
Maybe we should go into Q&A.
Yeah. So if you all wouldn't mind coming back up, our two panelists, Rajesh Shavi and Renee.
I'll join as well.
Great. And we have an extra wide panel for extra right set of seats for the larger panels that are going to follow. Gideon, maybe I could start with you. Any thoughts about what Mark was going to say?
I can never read Mark's mind,
Yeah.
but I think one of the things I think he was going to say was the log rank test wasn't handed to us on Mount Sinai. I think we're not bound by that. We can use other techniques. So I think we're a science-based organization. So I think this working group and I think the outputs that we're going to hear about today will help us think about other novel techniques. And like I said in my introductory remarks, and as Raji and Renee alluded to, we really want to be able to adapt if early on, say in an interim analysis, NPH is seen. What are the best methods to adapt so that you don't take a big penalty, if you will, and as long as it's pre-specified in the statistical analysis plan.
Great.
Those are
Great.
some of the
And we are going to obviously have a good bit of time during the course of the panels that follow to get into some of these specific trade-offs and issues. But I do want to make sure if any of you have questions or topics or perspectives that you want to make sure get addressed during the course of the day, now is a good time to bring those up. So feel free to head to one of the microphones, or if you're online, to send in a question or comment to help us frame the discussion that's going to follow. And maybe I could start with just a couple of questions. You all started with a wide range of deviations from proportional hazards. Wondering if in the course of the work group's analysis, one or more of those particular kinds of deviations got the most attention. I would guess that they have different implications for the kinds of methodologic responses that would be appropriate.
So one of the things that we did consider is that the long-term survival is really a delay. It's just a really long delay. But still, the point of needing to consider then how does that, is the right method, is that the same method as a delayed effect that's much shorter? We spent a lot of time looking at delayed effects, obviously because of IO, but we also have examples that we're going to share today where we had crossing hazards and the diminishing effects. So we tried to make sure we didn't stick with just the IO examples. Raji was very good at telling us this isn't just an IO project, so we kept remembering that. It was interesting that we presented the results back in April, or the concept back in April at another conference that Dr. Shadara had invited us to, and there we actually had some statisticians approach us from another company that work in cardiovascular. So I do think that whatever we come out with may actually have the potential to cross over into other therapeutic areas, which I think would be really interesting.
Just a related question to that is, how much has this been addressed in other therapeutic areas with respect to implications for regulatory decisions? Is this kind of out there?
It is out there, I think, and I've lived in oncology for so long, I can't speak for other therapeutic areas as well, but I do know, having talked about it within my company, that the concept of, for example, restricted mean survival time is something that has been looked at in other therapeutic areas. But I'm not sure that I've actually seen other therapeutic areas establish a method that then we might need to then take across into oncology?
I think, it's not just in the efficacy side. We have later on Kuntal Bai in the second session who is on the panel. He was actually with our safety division before joining us. And there also they have this, particularly the events they are looking there, of course, is the adverse events. And they do happen much later on, so there is always a delayed effect. And some of these non-proportionality is not uncommon to safety evaluation as well. But there it is more of the long-term risk versus the short-term risk. And I think it will come back to, even in efficacy, what is it that we are trying to estimate? Do we want them, the benefit to be seen as quickly as possible, or we are looking for an overall long-term benefit? And I think the key issue in any of the methods that is going to be presented here is the length of follow-up. So can we follow up long enough to use some of these methods would be a crucial thing that everybody needs to keep in mind.
Great, thank you. And for a first question, comment, please let us know who you are, and thanks for going first.
Elad Sharon, I'm at the National Cancer Institute. I guess I have a quick question, maybe it's sort of a two-part one, but we have been looking at some of our early trials and the prospect of durable responses, basically as a hint as to whether or not this would actually truly be a beneficial therapy, and as I guess a corollary, one would think that in earlier trials, whether or not you use durable response as an endpoint, you might actually be able to figure out whether or not non-proportional hazards might be more relevant when you're looking at OS or PFS down the line. So I guess I'm curious what you think about that as an endpoint, durable response or some other surrogate, basically to give you an idea, but also to help you plan those later trials, because you should have earlier trials that at least give you an idea.
Do you want me to go first? So I can say that our endpoints working team did look at what are the relevant endpoints, perhaps. But obviously, endpoints are not directly just related to non-proportional hazards. So trying to stick to objectives that were focused on non-proportional hazards was the challenge. But I'll let Raji speak, because her group's done a lot of work looking at correlation of endpoints in this setting. And I do think then that's where the durable responses come into play, because then that correlates to the progression-free survival and the overall survival endpoints. But that other side of our working group did try to look at this a little bit.
So, that's a good point. However, in the phase 2 space, you're looking at single-arm trials. And the selection of population is also one of the issues. It's not the same population as we see in phase 3. So to directly take the duration of response and conclude that there will be a delayed separation is hard to make. And it also depends on the control arm. So, yes, all of these separations actually in the IO we are seeing when we are comparing to chemotherapy, it remains to be seen if, it's a PDA1 versus another PD1, some combination you are seeing, whether you will still see this separation or not. So while it is an important concept to think about going forward to the next stage or not, how much we can use is questionable. And our, with the limited data, whatever we have right now of the PD1, PDL1 products, when we try to correlate between response and survival or response, PFS and survival, the correlation was very poor.
Just a follow-up related issue, how much in general have you found that prior evidence matters in the work that you're doing? And you gave an example earlier, I think, of Rajesh, of autologous versus allogeneic transplantation, where you got good reason in advance to think the curves might be different. Obviously, if you knew, as you said, if you knew that subgroup effects were going to, subgroups are going to respond differently, some patients would, some patients wouldn't, you just do a trial on the subgroup. Here, though, I mean, it seems like there might be some pattern evidence that could help shape the right approach to alternative to NPH.
Yeah, that's a very interesting trial, and also in any of the surgical procedures, perhaps, there is always some amount of risk immediately after surgery, but in the long term, there is a big effect. So perhaps when you are looking at that curve, in my mind, I don't know, I think Gideon or some other clinicians or yourself can tell that if it's a younger patient who is looking at it, they are interested in the long-term survival, so perhaps they will go to the one, even though it's riskier at the beginning. So it is a question of how do you say this, and certainly it is much more problematic when we are looking at oncology clinical trials, and I'm not sure there is a good way to explain that if you survive for 6 months, then the treatment will work better for you, but you may have a higher risk of dying before those 6 months. I think this crossing of curves is a bit of a challenge.
Any other comments about incorporating kind of priors into this framework?
I mean, I think we wish we all had crystal balls. I do imagine that most companies right now who are designing IO trials are assuming they'll have a lag. So back to the concept of you assume you have a lag, but as Dr. Sridhar just said, maybe in the presence of comparing yourself to another comparator, you might not have a lag. So one of the goals is now how do you design your trial up front, whether you have that lag or not, so you don't have the penalty for whether you see the lag. So I think now a lot of us are taking the past evidence of the IO trials and now designing our IO trials with that in mind, but the landscape is still continuing to change even in that setting.
So I have a related question.
And please be sure to tell us who you are.
Cyrus Mehta from Saitel and Harvard School of Public Health. Can we assume that typically IO trials may have a lag? And in that case, what are the implications for interim analysis? Because if you, the role of interim analysis aside from safety is either for early stopping for futility or early stopping for efficacy. It would be risky to have early stopping for futility because of the possibility of late separation, but it could be beneficial to have early stopping for efficacy. If you know that there's going to be late separation, it can benefit patients if you and get the drugs to market early. So there's that trade-off between getting more scientific evidence through long-term follow-up and at the same time denying patients the benefit of the drug through an early stopping group sequential type design. So I wonder, I didn't see much of interim analysis in your slides, but maybe you've thought about it.
You did mention those issues.
Yeah,
But,
I think probably in some of the later sections we may get a little bit more into it, but certainly that's a excellent point that in some of these, when do you do the interim analysis becomes a key issue. How much of follow-up you have, sufficient follow-up to make that kind of decision.
And I think that's the kind of issue that if others have key points like that they'd like to make sure that we come back to during the course of the discussion today. When do you do futility analysis? How often? What are the implications for interim analyses? Those are great topics.
So
for the
I see
groups to
Mark
discuss.
coming there right in time.
Please go ahead.
Yeah, so Boris Friedland, NCI. I just wanted to sound a little caution about the futility analysis. So we all assume that what's going to happen is the hazard might dip and then will go up and everybody will be very happy. But as we know with IO, the myeloma trials, that hazard dipped and never came up. So abandoning futility analysis altogether might harm a lot of patients. So one should balance this against everything else.
Yeah, these points are very important in the future design of studies. How do we do this? So hopefully it will come back again in session 4. Yeah.
And I'd like to welcome Mark to our panel. I know you have a bit of a commute to get to downtown DC, and especially on days when it's icy out and around, not easy. So we've been having a bit of discussion of different perspectives on issues that people think are important to discuss during the course of the day. And I don't want to put you on the spot, but any comments that you'd like to make to help set the framing for the discussion to come?
So I think, not having been here, unfortunately, for the initial, presentations, I'll probably just leave some, prepared remarks for the end of the session, session 5 of one of the panelists. So I'm happy to address any questions, that may arise, from the audience at this point, and, really looking forward to the discussion, both from the regulatory perspective, but also the clinical oncologist perspective here, as well.
Great. So glad to have you here. And maybe just as we get closer to the end of this session, Rajesh, Renee, what's your sense of how much of a difference this is going to make? I know that's not kind of probably a fair, easy question to ask at this point, but you all have been at this now for a couple of years, a big effort, certainly identified some important reasons why NPH methods could be significant, but we've talked about some of the complications already. I'm sure more of that discussion is coming. We talked earlier about not wanting to sacrifice too much in terms of alpha issues. And my experience in life and in statistics is that you don't get something for nothing. So is there a sense of how worthwhile or how impactful this effort can be?
So we hope it's impactful.
Yeah.
I think today is a great first test of that with the colleagues in the room and online that are going to share their questions and challenge what we've done. We're going to be transparent with assumptions we've made, the pros and cons. We're going to try to show you some case studies to show the method that we believe is perhaps a potentially better method than log rank. But we expect to be challenged today. That was the point.
Yeah.
I think the biggest challenge we have is, even if we land a method, how we help physicians and patients understand the trial results is still complex. And, the hazard ratio and the medians don't exactly explain the current results, but it's going to take a lot for us to shift away from the measures that most people think they understand, even though they don't understand. And I think that still is a challenge that we as the working group realize we have not tackled completely, to be honest. And we look forward to that conversation later today.
Yeah.
So, from a regulatory point of view, we put in the product labels when we see these type of patterns, we still go ahead and report the hazard ratio and the medians. And sort of the question is, are we misleading in a way that the treatment is the same for everybody and it's constant and it doesn't matter, throughout the time period, you will have this treatment effect. So if there is a better way, then we should do it. I think, to be perfectly honest, if at the end of the day we come to the conclusion with all these methods and everything, we still can use the same method and that's okay, and we are not misleading as much, that's also okay. But I think right now, knowing that the treatment effect is changing over time and sort of ignoring that and saying that it is constant and reporting as such, or reporting a median which doesn't really summarize in a good way the survival pattern, then we should do something different. So hopefully with all this effort we can do better. But if at the end of the day we come to the conclusion, none of these are better and we have to stick with what we have, then that's okay.
Well, Mark, I know you've thought about this a lot, and again, not to put you on the spot late in the panel, what I found most convincing from these initial presentations was just those non-parametric survival charts, which are clearly look like they're not proportional hazards. Now, there may be some issues of sample sizes and power of statistical analysis behind those, but maybe there is a distinction between, we get to the same basically correct regulatory decision, and also the importance of communicating to patients that life isn't as simple as non-proportional hazards and those key statistics. Any thoughts on this?
No, I mean that's a great point. I mean we've been thinking about this a lot, especially in the immuno-oncology space, and that these this product class has really brought this issue, I think, to the forefront of the conversation. Just looking at the approvals of all the immune checkpoint inhibitors, there's been 36 initial or supplemental approvals since the first approval of ipilimumab. And looking at the endpoints, 20 of those approvals were based on primarily objective response rates and durability of responses. But there was 16 for which time to event endpoints was a very critical piece of that demonstration of effectiveness. And just looking at the Kaplan-Meier plots that are in the labeling for those approvals, probably about 12 of those 16 were non-proportional hazards. If you look at them, mainly OS, but also represented with PFS and DFS, disease-free survival. So thinking differently, I think, for explaining the potential of the product to provide clinical benefit has been challenging with IOs because you can't think of medians depending on the shape of the curves, especially for something like progression-free survival for multiple products that have shown not necessarily a separation of medians, but you see this prolonged tail, and it's really that durability for that proportion of patients who response that has really described the benefit. So trying to capture that in a discussion with a patient, I think, is a little bit challenging if you're just talking about medians, but if you show and if you think about the whole curve, I think you can summarize that treatment effect for a patient, effectively. The other thing that's critical is really understanding the pharmacodynamics or the mechanism of action when we're thinking about, explaining the efficacy, especially with immuno-oncology products, at least with immune checkpoint inhibitors. We've seen this response that don't necessarily fit the conventional criteria. So thinking about how to explain that to patients has also presented challenges, but certainly work underway to help to better capture that type of clinical benefit.
Great. One more question.
Luis Rojas from Aquidia, formerly known as Quintiles and IMS. I think that talking about sequential designs and the challenges in adaptive designs is becoming a critical issue. And I am one of those statisticians working on oncology, also in multiple disciplinary areas. And the issues is across the board. And it's happening in GI, happening in cardiovascular, it's everywhere. Now, things that at least we're doing in our organization is what you do at the moment of the interim. What kind of report do we need to present at the moment of the interim? Because we may have to make decisions based on the futility boundaries. We may be in the presence of a possible futility issue from a milestone perspective, but things based on simulation may separate after the interim. Because upfront, we didn't know what was expected at the interim. And then we know based on preliminary data and a lot of data that is available that the separation is about to happen at a future time point. There are many issues around sustainability and durability, and as well as risk after follow-up period. So those are critical issues that they're going to have somehow need to be considered moving forward for those working in adaptive designs, for those working in study design and so far. So something that I wanted to share. So in oncology, we're not alone. It's happening for us a lot in GI, NASH, you name it, it's happening. So it's a common issue. We thought, and when I talked to clinicians, they said, well, we thought that the disease was constant. It is not. The relationship between patients and physicians is something that is affecting the placebo effect, is something that is also affecting the treatment effect. So that's why we see these weird curves at the beginning, and then the separation is going to happen, and then we see them sometimes crossing. So what to do, my humble advice is, we need to be mindful what we presented during the interim, and we need to support the result from the interim with data and simulation from known data to make the decision that we want to bound to the futility room or do we want to bound to the efficacy rule. So maybe it's not a true effect side that we're serving at that time point. Just wanted to share that with you guys. Thank you.
Thanks. And if there are further comments on this, necessarily, now a couple of comments about the timing of interim analysis and futility analysis, especially in this presence of potential late separation. So we'll try to make sure we cover that issue today. Any further comments at this point?
I think when we get to Dr. Anderson's presentation, when we talk about designing a trial up front, when we talk about designing a trial, then that's also around how do you design your interim analysis. I think that would be a great time for a panel discussion about the topic.
Okay, I want to thank our opening panelists for helping us get teed up for what will be a very interesting discussion today. And those of you who have raised issues, I hope you'll continue to do that during the course of the discussions today so that we make sure we hit on all of the important topics related to this work on non-proportional hazards. Right now we are running a little bit ahead of schedule. I'd like to take a 10-minute break, but plan on restarting around 10:20 rather than 10:30. Somehow I think there's going to be plenty of presentations and discussion to come. So see you back in 10 minutes, and for right now, thank you to our first panel. All right, we're going to get started a few minutes early with this session and hopefully end up in this session a few minutes early, but I know we've got a lot of topics to talk about in the context of Session 2 on analysis methods and simulations addressing non-proportional hazards. So I do want to get the discussion started. For this session, we're going to focus on alternative statistical methods for analyzing time-to-event data in the presence of non-proportional hazards. And we expect that this discussion is going to highlight not just the current statistical approaches, but how the working group decided on the specific alternative methods that are a big part of the work that they've done, and the simulations to demonstrate how these methods can be applied. We're going to begin with 2 opening presentations, followed by a panel discussion, and then an opportunity for questions and comments from the audience. And just like in the earlier session, I'd like our 2 presenters to do their presentations with both the panelists and the rest of us having a chance to see the slides and so forth along with it. And then we'll ask our discussants and panelists to come up for the discussion part of the session. So we're beginning with our 2 presenters, Satrajit Roy Chowdhury, who is the Senior Director at Pfizer, and Tianlei Hung, the Principal Research Scientist of Statistics at Eli Lilly. So they'll begin first, and after that I'll introduce our panelists. So Satrajit, if you could come on up.
Thank you, everybody, and thanks Raji and Renee for setting up the stage. So this session will try to concentrate on the methods that this group, the Cross-Pharma Working Group, has considered and looked into with simulation studies. We try to give an overview of different methods we considered, and then my other teammate, Dr. Tianlei Hu, is going to cover up the simulation part. So before I go forward, there are contributions for many, and here are just few names who has a very significant contribution, but there are other members who actually contributed this work stream too. And I just like to give one disclaimer. I'm employee of Pfizer, so whatever I present is my own perspective and the team, this working group perspective, rather than the perspective of Pfizer. So it's already been called, so I don't want to repeat that. It's already been presented several times now that our standard, or the gold standard at this moment of reporting randomized clinical trial with time to event is log-rank test, Kaplan-Meier estimates, and hazard ratio. But the problem is they start to drift apart when you have non-proportional hazard, because some of those measures and the test start to have some problem. Before I deep dive into more complex methodologies, or the next step, I just like to quickly mention a little bit about Cox regression and log-rank test, just to put things on a context. So we can penalize them very quickly, but maybe we first know what they are in quickly. And so log-rank test is a fully non-parametric test. So even when you have non-proportional hazard, it's not invalid. The problem is you start to lose a lot of power when you do such a testing. But the Cox regression is more problematic because you violate the fundamental assumption which says the treatment effect is constant over time. And you have seen the examples from Dr. Sridhara and Renee's talk that this is definitely an assumption violates in all those case studies. So today, most of the methodology that we have explored is confined to 3 types of non-proportional hazard. The first one, which you have seen examples in Renee's talk as well, that diminishing effect where the treatment effect emerges late, and crossing hazard when you basically started with treatment to be bad, and then treatment effects start to emerge out. And diminishing effect where you start to have an effect which later dies down. So with that, and The problem, what we have most of the time in clinical trial, and most of them mentioned it, if we knew that this is my curve look like, we know the methods to address it. But the question is we don't know it beforehand. We don't know when this delay will happen, when the separation will start. This is unknown to us at design stage. And that's what brings us to this forum and the working group that we are working for last 2 years, that what could be, so there are extensive, having said that, statistics has looked, the people in statistics and the researcher looked into this problem for a while. They looked into different methods of analysis. And the majority of them, if you know the method, know the curve well, you can find a method. But our challenge for us when we begin, how we pre-specify it, how we specify an analysis at the beginning when we exactly don't know what's going to happen in future. So here are 3 questions we are going to try to address throughout rest of the day. One is, what would be an appropriate analysis where there's a chance of having non-proportional hazard? The second question was how we design such a trial. And the third is how to efficiently communicate it. So in this session, I mostly try to focus on the first question, and my teammates in the next few sessions will try to address the others. Now, when we started this hunt of or looking for a good primary analysis in presence of non-proportional hazard, the first thing that came into us is how would we comply with ICH E9, which says you need to pre-specify appropriately this analysis into your analysis plan and in the protocol. The problem is you can have different tests in the literature which talks about different types of non-proportional hazard, how to handle that statistically. But when I don't know, I need a robust method which can take care of that uncertainty. That if I see something, suppose I assume that I would be proportional hazard, I see delayed effect. I thought I have delayed effect, but I see crossing hazard. So this type of, we need a set of robust method which can handle such a thing. Even if we look into delayed effect, suppose my primary belief was it will happen in 3 months, but what if it happens maybe later? So when these things are uncertain, we need a method that takes care of this uncertainty together. So we started to look into statistical literature, and if we follow the literature well, there are 3 types of non-parametric tests people had worked on or looked into. One is very popular rank-based test, which log rank test is one of them. And there are other popular tests like Wilcoxon and others, which is very well known. And you have a way to weigh earlier and later events. The second class, which would start to mix up component, where basically start to see different tests together, combining. And the last test, which gained quite a bit of interest in recent days, which is Kaplan-Meier-based method, which you look into the area under the Kaplan-Meiers and other things. And we'll go through each of these in this talk and try to go through their merits and demerits. So before we start with our rank-based test. So in the rank-based test, we consider different, but here we mainly focus on a wide range of tests known as Fleming-Harrington weighted log rank test. Why we consider that? Because this has a property of covering a wide range of non-proportional hazard with a choice of parameter. And log rank test is a part of that as well. So by choosing that, we can actually cover up different areas with different choice of parameters. As you can see, if you assume your rho to be bigger and the other parameter to be 0, it actually gives more weight to the earlier event. So if you have a diminishing effect, or if your separation is early, you may want to do that. And whereas the other way, you may need to think of a second parameter if you think about a late difference. But the major problem is how we specify this test early. So in order to specify it in the protocol, we need to specify these parameters. And each of the parameters goes into different types. So, but if we don't know what's going to happen in future, fixing that is problematic. Because the misspecification end up with a huge power loss and also it's a misspecification model problem. We are not estimating or we are not testing the right thing after that. So what we can do here? So what we tried to find out a way which is called combination test. It was been in statistical literature for quite a while and been introduced by Young and Prentice, later on by Harrison, also been discussed in the Fleming and Harrington 1991 book, and recently by Garris into some specific type. So what it does, it basically, instead of having one Fleming-Harrington, it tried to consider multiple, so that you take care of different types together. And what we looked into two types of Fleming-Harrington test. The first one, if you fix 0, 0, that's basically log-rank test. And we actually took a consideration of log-rank test as well as a test that takes care of late separation or delayed effect. We also consider another combination test, which is we thought to be more robust. And that's basically come up with different sets which takes care of proportional hazard, early separation, late separation, and also diminishing effect where it gives more weight to the middle as well there. The whole procedure, what it does, it's adaptively choose the best test statistics based on the data. But when you do a best selection model, you end up with a multiplicity. You may have a false choice. You come up with a false detection. So definitely when we incorporate such a method, it needs to take care of that multiplicity. One easier way to use a very conservative method, but that's give you a lot of loss in power, or you need much more subject. Whereas one can actually work out the exact asymptotic distribution and can do the adjustment based on that. And the beauty of it, now you can specify this completely in your protocol and your testing strategy beforehand, before starting the trial. Now, last and not the least, we also looked into the Kaplan-Meier best test, which is, as we say, it has a huge popularity based on its ease of interpretation. So what Kaplan-Meier test does, as you all know, Kaplan-Meier plots give you the profile of survival over time. You know how many proportion of patients is going to alive on a specific time zone. So what it looks into, so the restricted mean survival time, or RMST, which is very popularly discussed recently, is it looks into the area of that curve until a predefined point. So if you want to use RMST, you have to define a predefined point, and it has a nice interpretation with the life expectancy of a patient over the next 20 months. That was the beauty of this method, where it started to get so popular. And any of the Kaplan-Meier test, what it does, it compares this area under the curve between treatment and control. And that's how you conclude about it. So we looked into two of them, two of the very popular ones. The first one was Pepe and Fleming, we looked into, where you take the weighted difference of two Kaplan-Meier, and the weights are chosen about where the censoring of the patient. And the second one we looked into is the restricted mean survival time, where you look into the area of the curve, and the treatment effect is difference or the ratio of RMST. Though it's one thing I'd like to mention about RMST, it's not only the testing, it's only perspective. It has a nice interpretation of its clinical meaning, because we heard quite a bit in the first session how to interpret a treatment effect. Even though it's, in some situation, it may not be the best testing strategy, but its intuitive way of interpretation always adds value, and we're going to discuss that, of course, later on. But both of them suffer a point where you have to predefine a cutoff, but you're in the design phase or in the protocol phase. But when you go to the final stage, due to the censoring, that cutoff may be problematic, because you may have a lot of censoring before that or after that. So you have to be, that's a problem of pre-specifying such a test. Now, to complete the picture, we also looked into piecewise hazard model, which is, or piecewise log rank, where you separate the whole time space into different time axes, and you look into the global test that if the treatment is significant on each of them or not, or some of them. our treatment is actually significant in at least one of them, kind of a test. And the problem of that test is it's very intuitive because you get a piecewise hazard ratio, which is nicely interpretable. But the problem is you have to specify your intervals earlier. So if you know that your delay is 3 months, you can define an interval 0 to 3 and 3 and more. And then you can show that, okay, globally, you have at least one significant p-value. But the question was, if this changes, this cut point doesn't hold in reality, you lost the power of such a test. And that's what the main constraint to pre-specify it. And in next session, my colleague is going to present simulations of many of these methods to show the power of this combination method over many of this type of statistics. So far, I just discussed about the testing strategy, that when you look into a first doorway to go to the regulatory that needs a significant p-value and a reasonable way to test under these circumstances. But the more challenging question come in, and we heard it already from people, that what is the best way to report the treatment effect? Because a number, which is we call hazard ratio, and this in such a case where your treatment are changing or you use weighting scheme, you can look into average, so average over the time, but they may be less meaningful because we saw example from Renee that really when 50% people are not benefiting, what hazard ratio 0.62 really means on such a case. So what we proposed here, which is more aligned towards Royston and Permer 2010 Stats and Medicine paper, they proposed a stepwise approach. So the first step is you look into the first door of your significance with your sample. So you apply a combination test, and we will show in a why combination test is pretty robust under different scenarios. In the simulation talk, we'll see. And then once, irrespective of that, you first assess before going into treatment effect reporting, you first assess the proportionality hazard assumption into the data. One of the ways you can apply a GT test, but unfortunately testing is not sufficient in such a setting, so you still need to look into other diagnostics like log-log survival plot, cumulative survival plot, which gives you reasonable comfort about the proportionality hazard assumption. And you based your treatment effect, or what nowadays people also call treatment effect estimate, based on your assessment in step 2. But this is a shift in paradigm, because so far we were reporting log rank test and hazard ratio, which are dual to each other. They are technically dual. So this kind of allow a shift in overall paradigm, which we are here to really hear your opinion and your thoughts on. So here is the proposal comes in. So if my step 2 is my assessment of proportional hazard is reasonable, we actually we still report our standard Cox hazard ratio and confidence interval because the fundamental assumption of Cox regression still remains there. And we can also look into, because we're using a combination test overall, we can look into dual estimator, which is the average hazard ratio from combination test. In the second case, when you don't have non-proportional hazard, that means either of your diagnostics, so the plots we have seen in the trials, in such a case, we are saying a single measure may not be adequate in such a case. And you need to report a multiple, so which is clinically meaningful. The first one is, of course, we still report average hazard ratio in order to be consistent with the testing, but we also allow to report RMST at a specific time, which is clinically meaningful, and difference in milestone survival. Milestone survival has a very nice clinical interpretation that how much gain in survival probability you have, whereas also RMST has a nice interpretation of life expectancy. So we are proposing to include additional measure in order to have the treatment effect explained to non-statisticians. And in session 3 in case studies, we are going to see many of them. So I quickly conclude. So an NPH working group actually looked into many of these methodologies and tried to assess them with simulation and case studies. Max combo test, which is combination of different weighted log ranks, seems to be a potentially encouraging approach because it allows you to take care of different proportional hazard and give you a robustness over that. Also allows you to comply with ICH-E9 and specify in the protocol. And also regarding test estimation, we think beyond one measure is very much necessary. And all these testing methods are available into a publicly available software called nph-sim. So if people want to try in the public, it's available in GitHub and in the internet. So in the next talk, my colleague is going to talk about simulation result. Thank you.
Okay, thanks, Satrajit. Next is Tianlei Hu. Thank you.
Good morning, everyone. I'm very happy today to represent the simulation team to report the simulation methods for non-proportional hazard methods. My name is Tianlei Hu. I'm from Eli Lilly and Company. With Satyajit Roy Chaudhary from Pfizer, Kevin Anderson from Merck, and Julie Tong from Boehringer Ingelheim, we formed the leadership team for the design analysis work stream. The simulation is a sub-team which includes members from a lot of the companies across industry, including AbbVie, AstraZeneca, Bayer, BMS, Boehringer Ingelheim, Roche Genentech, Johnson & Johnson, Merck, Pfizer, Sanofi, and Takeda. A lot of them are in the audience today, and many of them are online as well. And here I also want to pay special thanks for Larry Leong from Roche Genentech. I think he's in the audience. He has made tremendous contribution for our simulation effort, and also to my colleague Honglu Liu from Eli Lilly and Company, who provide a lot of computational support for our simulation effort. So at a very high level, our simulation analyzed the operating characteristics of 3 categories of methods. So the category 1, as mentioned in the previous presentation, is basically rank-based methods, which include the log rank, the Fleming-Harrington 0, 1, 0, and 1, 1. The second category is the Kaplan-Meier-based methods, which include the weighted Kaplan-Meier as well as the restricting survival time. The third category, we have 2 combination tests in them. One is the Breslow combination, which combines the log rank with the test of acceleration, and also the max combo test. The max combo test combines basically all the tests in the category 1 tests, which include the log rank, the FH0, 1, 1, 0, and 1, 1. We considered 7 scenarios for our simulation. 2 of them are delayed scenarios. One is diminishing effect scenario, also known as the belly shape. And we have 2 scenarios which are the crossing hazards, and also we covered the proportional hazard scenario as well as the null scenario to evaluate the trade-off between the method as well as to analyze the type 1 error control. All the scenarios are actually informed by the real trials that are in the public domain, so we're not just making up hypothetical numbers for our simulation. One thing of note is for our max combination test, in the simulation, you will see the max combo actually uses the 4 tests in category 1. The motivation for that is because the simulation scenarios, we have a variety of scenarios. We have the delay scenario, crossing hazard scenario, diminishing effect scenario. But in reality, maybe you're only concerned about just the proportional hazard and delay scenario. In that case, when you do the max combo test, you don't have to use all 4 tests in the combination. Maybe you just consider the log rank with Fleming-Harrington 0, 1. To simulate the data for our simulation, we basically use the piecewise exponential models to simulate the data, assuming a piecewise constant hazard ratio. For instance, for this delayed effect 1 scenario, basically the hazard function takes 2 pieces of constant for both the treatment arm and control arm with a change point equal to 3. So that means before month 3, the hazard are 0.104 and 0.103 in the control arm and treatment arm respectively, resulting in a hazard ratio of 0.99. And after 3, the hazard ratio is 0.478. Similarly, for the diminishing effect, the treatment effect is 0.731 before the change point, and after the change point, the hazard ratio is almost 1. For the crossing hazard, you basically have a hazard ratio that's bigger than 1 before the change point and much smaller than 1 after change point. For the proportional hazard scenario, the hazard ratio is basically a constant. So it's 0.68 both before the change point and after change point. For the null scenario, the hazard ratio is just 1 across the 2 pieces. So that's how we come up with the scenario for the treatment effect. We also consider the impact of other design parameters such as sample size, enrollment duration, dropout. So for the sample size, we consider 300, 600, and 1,200. And for the enrollment duration, we consider 12 months, 18 months, and 24 months. So for every scenario of treatment effect we consider, there are basically 3 sample sizes across 3 enrollment durations, a total of 9 cases under each scenario. To enable a fair comparison, we also fixed the number of events to be 210 across all cases and all scenarios. So for instance, if your sample size is 300, that means your event rate is 70% in the simulation. If your sample size is 600, because we fixed the number of events at 210, that means your event rate is at 35%. So with all the housekeeping taken care of, I think we're ready to get into the simulation results. So first of all, we're going to look at the testing. So this first slide shows you the rejection probabilities under the null scenario, aka type 1 error rate. Every line in the plot is basically a method. So like I said, we consider 3 categories of methods. And the x-axis are those cases that I mentioned. There are a total of 9 cases that we consider for each scenario. And the y-axis is the rejection probability. So here we're looking at the type 1 error control, and we set the nominal type 1 error at 2.5%, one-sided. So you would expect that the method needs to basically, out of the 20,000 simulations we did, 2.5% of the time, it should reject the null hypothesis for the method to be a valid method to be considered. So as you can see, all of our methods control the type 1 error pretty well. There are some random spikes, but those random spikes over 2.5% are usually within the simulation standard error. For the null, we have 20,000 simulation iterations for the simulation, and the simulation standard error is 0.1%. So they're mostly within 2 standard deviations of the simulation standard error. There is this blue line, which is the Breslow combination, which looks like an anomaly right now. And so just to remind people, the Breslow combination combines the log rank test with the test of acceleration and assumes the asymptotic independence between the log rank and the test of acceleration. And that happens when you have infinite samples, but in finite samples, this independent assumption might not have kicked in quickly enough. So that's why you see this over-control of type 1 error, and at least that's our explanation right now. So we take care of the type 1 error control. Every method controls type 1 error pretty good. And the next, we're going to look at the power comparison of different methods. So once again, every line is a method. And then, because this is a power comparison, the higher the power, the higher the line, the better the method. So this first scenario we're looking at is the delayed scenario. As you can see, the best method for this first scenario, delayed scenario, is this Fleming-Harrington 01, which is not surprising because this is a delayed scenario, and Fleming-Harrington is overweighting late events. So that's why that's the best method amongst all the methods we considered. But if you look more closely, the second best is the combination test, which is what this working group is recommending at this point. And also, it's neck to neck maybe with the Fleming-Harrington 11 as the second best. And the max combo test is actually much better than the log rank, which is the middle of the pack, I believe. And then obviously the The other methods that are based on the Kaplan-Meier curves, the streaming survival time as well as the weighted Kaplan-Meier, they don't do so well in this delayed scenario. So at least the Max combo method looks very promising in the delayed scenario. The other thing to note is just to echo something that was mentioned in Renee's point that remember all the simulations we have fixed the number of events at 210, but as you can see, the power across all those cases is the highest when we have 300 patients. So 300 patients, you have the highest power. The next three, the sample size is 600, they have a little bit less power. And then when you get to 1,200, even less power. The reason for that is basically it's related to the maturity of the data. When you have only 300 patients and you want 210 events out of 300 patients, the data got to be very mature. It's 70% event rate. For a delayed scenario, you're basically capturing all the delayed separation. So that's why the smaller sample size actually gives you better power than a trial has a much bigger sample size. So that's a little different than our typical oncology trials because people usually think like as long as you have the same number of events, you get the same power. But obviously that's not the case for the MPH scenario. And also, if you look within the same number of the same sample size, the longer duration, 12, 18, and 24, also, the longer the duration, you get better power as well. And the explanation is exactly the same, because the longer duration, the more maturity of the data, and that's why you get bigger power in terms of capturing the treatment effect. So both of these 2 points, I think, is worth calling out because it's a little counterintuitive than our typical oncology trials. So the next scenario we'll look at is the diminishing effect. The diminishing effect is usually very challenging just because the Kaplan-Meier curve, they have to separate and then come back together. Usually the overall treatment effect is somewhat relatively smaller. And in this case, our best, the most competitive method, I believe, is the Fleming-Harrington 1-0 and also the weighted Kaplan-Meier method. But the Fleming-Harrington 1-0 being one of the most competitive methods is not surprising because it gives more weights to early events. But also, in fact, the log rank test is pretty competitive as well in this situation. So it's almost neck to neck with the other 2 methods. But if you look at our max combo test, this is basically the 4th method. One of the top 4 methods. So it's also pretty competitive. It's maybe about 3 to 4% less power than the log rank, which we think is a kind of a very small power loss. In this case, the other thing to note is obviously it's the opposite of what we said in the delayed treatment effect scenario. In this case, the bigger the sample size, you actually have better power because you have now the data is less mature, but because of the treatment is happening early on in the Kaplan-Meier curve as opposed to later separation, so it's not surprising that those early event immature data will give you better power for the same sample size, for the same number of events. Crossing hazards, according to our simulation, actually it's pretty similar. All the conclusions are pretty similar to the delayed effect scenario. The most competitive method is the Fleming-Harrington 0-1, which is not surprising, but the second best is the combination test, which is this dark brown line. And then the log rank could be severely underpowered. And then the Kaplan-Meier-based method doesn't perform as well. But the conclusion is pretty consistent with the delayed effect scenario. The PH scenario is a scenario where we know the log rank test performed the best because mathematically you can show that the log rank test is the most powerful, non-parametric most powerful test under the PH scenario. Nevertheless, it's important to evaluate the trade-off between the log rank and the alternative methods we're analyzing here. It's important to see that our simulation basically confirms the log rank is the most powerful test. But also, it's very encouraging to see that our combination test is very competitive. It's generally within kind of 4% in terms of power of that log rank test. The other thing to note is this orange line represent the Fleming-Harrington 01. And this is a test where you want to use it in the delayed treatment effect. But under the proportional hazard scenario, actually the power loss is pretty substantial. So it's more than 14 or 15% power loss on average for the Fleming-Harrington 01 test. It's important to know that because when you're designing a trial, you're concerned about the delayed treatment effect, but you're not exactly confident that it's going to happen in your trial or not. And then if you design using the Fleming-Harrington 01 as your primary test, it could be problematic because if by the end, if there is no delayed treatment effect and it turns out the trial is a proportional hazard trial, then using the Fleming-Harrington is going to basically give you a underpowered study. So I think I just mentioned that. So our max combo test, remember right now, has 4 member tests. We have the log rank, Fleming-Harrington 01, 10, and 11. So every time you invoke the max combo, it basically picks a member test that maximizes the test statistic. This chart just summarizes the selection probability of each test for every scenario. And then, so for instance, for the delayed scenario, the max combo test is selecting the Fleming-Harrington 01 majority of the time. So above 60% of the time, it's selecting the right member test from the max combo. And similarly, for the diminishing scenario, majority of the time, the simulation is picking the Fleming-Harrington 10 as the kind of the maximizing test. And for the crossing hazards, 80% of the time, it's selecting the Fleming-Harrington 01. And for the proportional hazard, the selection probability is pretty even across all the four tests that we consider in the max combo test. Okay, so that concludes the estimation, the testing part of the simulation work. Next, we're going to move on to the estimation part of the simulation. The plot you're looking at, this is probably, at least for me, when I first looked at it, this is my first time I saw those plots. It was first introduced to me by Kevin Anderson from Merck. The plot is known as the half-violin plot. It's a plot to visualize the basically distribution of 2 random quantities. So here you're looking at, for the left half-violin, that's the density of the Cox hazard ratio from those simulations. And the right half of the violin is the max combo estimate. And then we're looking at 2 scenarios, the null scenario and the alternative scenario of proportional hazard with a hazard ratio equal to 0.68. So as you compare the center of those distributions across all cases and between these 2 scenarios, the center of the distributions are almost identical between the Cox model and the max combo estimate. And the reason for that is the max combo, remember, has 4 member tests. Each test is the weighted average of the hazard ratio. But for both the null scenario and the proportional hazard scenario, the hazard ratio is a constant across the Kaplan-Meier curve. So if you do a weighted average of the hazard ratio, a weighted average of a constant is still a constant. So that's why each member test is still unbiased in terms of estimating the original hazard ratio. But there is a little bit selection process going on, right? Because you have 4 member tests. You're basically picking the one that maximizes the test statistic. So we're not saying, I mean, it's completely unbiased. There's a little bit bias due to the selection process, which you will see on the next slide. So for our estimation, under the null, the max combo hazard ratio estimate is 0.95 versus the truth is 0.1. And under the proportional hazard scenario, the hazard ratio estimate is 0.65 versus a truth of 0.68. In both cases, the point estimate bias is very negligible, and the bias is mostly due to the selection process of the max combo test. In other scenarios, the point estimate of the max combo is the weighted average hazard ratio. Obviously, now it's going to depend on the weighting function, and it doesn't make sense to compare that the average hazard ratio with the Cox hazard ratio now. So, so how do we report the treatment effect? I think this is, that's almost the same slide as what Satyajit has in his presentation. The working group right now recommending basically use the max combo as the primary test to reject the null hypotheses. Once the null hypothesis is rejected, then our second step is use some kind of diagnostic tool to look at the MPH, either through a GT test or a graphical tools to see if the proportional hazard is violated. If proportional hazard assumption holds, then our recommendation is to report the standard Cox proportional hazard ratio, as well as the average hazard ratio of the max combo test with its corresponding 95% confidence interval. If the proportional hazard assumption doesn't hold, then in addition to these treatment effect estimates, we also recommend reporting the difference in resuming survival time at a specified T-star, as well as the differences in milestone survival rate at pre-specified time points. And the secondary analysis could include the piecewise hazard ratio with their corresponding 95% confidence intervals as well. So we'll see more of these in action in session 3. So our conclusion at the recommendation at this point is we believe the max combo test is very robust and is agnostic to types of non-proportional hazard scenarios. It has a very strong upside under the delayed effect scenario as well as the crossing hazard scenario. Both are very common in immunotherapy studies. There is very acceptable loss in power under the proportional hazard and diminishing effect scenario, usually based on the simulation, somewhere between 3 and 4%. Such trade-off should motivate max combo being a competitive test in oncology clinical trials. For effect estimation, under MPH is somewhat more very complex. The max combo estimate, the bias, there is a little bit bias, but usually it's negligible under the null scenario and under the proportional hazard scenario. To report the treatment effect, we basically recommend take a stepwise approach and also more data-driven, and then depending on whether you see proportional hazard or not. So I think with that, that concludes my presentation of the simulation results. And this slide are the questions that we have for the panelists and the reactants. So I will just read through them. Do the panelists agree the max combo is a robust and appropriate test when the trialist is uncertain of MPH? Would it make sense from both a statistical and clinical perspective to use max combo test as an alternative test to log rank test as the primary test in certain clinical settings? Do the panelists agree the max combo estimate, together with the milestone estimate and resuming survival time, is a useful measure of treatment effect in the presence of MPH? Do the panelists agree that how to report clinical benefit should be dependent on the presence of MPH, i.e. tailored to a specific data set? So that's all. Thank you.
Chun-Li, thank you very much. Great questions for our panelists. Now you've had a chance to hear about the specific alternative methods that were recommended by the group and simulations that illustrate how they work, at least in these contexts and practice. Now to help answer the questions about their value and how to apply them, I'd like to introduce our panelists. And we'll do this the same way, with the panelists each making their opening comments, then everybody's going to come up on stage. First, David Harrington, research professor of biostatistics at the Dana-Farber Cancer Institute and the Harvard T.H. Chan School of Public Health at Harvard. Then Kunthel Bai, statistical researcher in the Office of Biostatistics at Cedar FDA, and Gideon Blumenthal will be back up again for comments here. So first up is David.
So thank you very much for the invitation to be here. It's always a bit forbidding for panelists to try to say something in 5 words after some very smart, very committed people have worked for more than 2 years on a problem. And to think that I could compress that into 30 seconds is either egomaniacal or delusional. So I'll do my best. The other thing I will say, just as a matter of a sort of personal thing that I was describing to Kevin at breakfast this morning, I put the weighted log rank tests in the category of work that I call my rusty staple work, which is to say we don't have paper copies of our papers anymore, but if we did, and I stapled it when we did it, and we put it in the drawer, the staples would be rusty. It would have been there so long. So it's interesting to see it reemerge. So if someone could call up the slide that has the questions, so I can see it here. That's great, thanks. I'm going to actually start with a few general remarks, and I will touch on some of those questions along the way, and if I miss something, I'll go back to it. So what I'd like to do is divide what I'm going to say into the usual components here that we think about in these trials, and that's design and testing and estimation, and how these things link. So I think that for me, and I've known this for a while, we all have known this, the key thing that's coming through about design, and it's been mentioned several times, is ignorance. It's basically, we don't know what we're going to see in these, especially in areas where things are very new and the mechanism of activity may be new, and the treatment patterns and the adherence within the trial, the possibility of placebo effects for agents in cancer that are no longer completely toxic. All those are going to affect the pattern. So what I'd like to see over time is more direct acknowledgement in the design of the ignorance, which is to say you can either characterize that by saying, well, there are sort of families of distributions here that we can parameterize through exponential models or time-varying hazard ratios, et cetera, or we can go back and we can lean on some of the very nice work that's been done here by people in this working group and the stuff that's on the in the GitHub repo and say, these are the patterns we've seen. And we really don't know which of those patterns might emerge, but we want to design this trial in a way that is somewhat robust to whichever of those might emerge. And I think the design, those of us who review clinical protocols and review papers, we're more than willing now to see extended discussions of design that don't say, we assumed that the baseline failure rate was X, we assumed that the hazard ratio was Y, and we assumed that the dropout rate was Z, and here's our sample size calculation. That's just no longer realistic in this setting. So design, I think, should acknowledge the ignorance here. And I'm sort of prefacing my remarks, and I'm assuming that we're staying within a frequentist domain. There's lots of ways to use that prior information in Bayesian analysis, but we're not going to touch that right now because then that becomes chaotic, right, from the agency's perspective and everybody else. Testing. So I'm going to be agnostic about a test in the sense that it's an experiment. You design your experiment. You specify in advance how you're going to analyze the data, how you're going to test your null hypothesis, and it doesn't really matter to me what you say your test is going to be as long as you use it. And as long as it's linked to your design, as long as it's linked to power calculations that you've done based on the various things that you can see in the design. So testing for me, there's no sort of tabernacle where the log ranks test sits. It's just a test. And in fact, when we're all being honest with ourselves, this whole idea that we are basing approval of an agent based on a single p-value from a single test is vastly oversimplified. So the role of the test needs to be put in context. So the test is just the test. Now, of course, what happens when you move away from simplifying assumptions like the proportional hazards and the log rank, is if you're going to use a complicated test, you lose the link, the direct link to a parameter estimate, or at least a single number summary. Although there's some work that's being done here which shows that there probably are some single number summaries which are lurking beneath some of these tests which might be useful. So for me, that's the key step here, is how you make the step from testing to estimation. And I think that I like to think about the estimation problem as in two domains. One is what has to be in this little teeny package label, right, which the FDA and the companies have to sort of shoehorn so it fits in a little box and it can be unfolded and people can read it, versus how do you convey the information of what's going on here. And there are, I'm going to sidestep the label. That's something that FDA has to work out with companies, and let's assume that it's always going to be a compression of information that will have some loss, some data loss. But let's work with this sort of estimation in a way that conveys what's going on. So I think that my own bias in listening to this is that these single number summaries, they were okay when they were linked to the log rank test. They're just not going to do it right now. And I think that this is more of an intuitive sense, a visceral sense, that watching people do these wonderful acrobatics to squeeze point estimates out of these combo tests is sort of sidestepping the main tool we have here, which are the pictures. And the pictures are really important. And I think that we undersell the ability of treating clinicians and patients to get information from those pictures. I'm old enough to remember the days when we firmly believed we would never be able to explain sequential designs to clinicians. Okay, so we can now, and they understand it. And they understand what the pattern in a survival curve means. And it's not really, as I was saying to someone earlier, it's not really the patient that deals with it. The patient's in shock, right? The patient's been diagnosed with cancer. It's a potentially fatal disease. They're in shock. They don't emerge from that shock until probably 6 months to a year after their initial treatment. It's the patient's caregiver. It's the adult children. It's the people working with them that are going to look at these and think about them. So I would like to see us move away from single number summaries and provide things that are both visual, the curves, and numerical, the quantiles. Basically, the curves are giving you both the percentiles and the quantiles, but the quantiles, we already have some history with the median, right? That's a quantile. Why not give the 10th, 25th, 50th, 75th, 90th quantiles of these distributions and, let people think about them? Now, I think that if I'm, really want to be a bit transgressive here, I will say that I'm one of your colleagues. I don't believe that we should be the ones to decide what the way is to summarize with a single number summary a treatment benefit, because it's not a statistical decision, it's a value decision. And it's really a value decision that has to be made in the trenches by the treating clinician. Now, I know in the background there's regulatory approval and there's payer things and all that sort of stuff. Unfortunately, as an academic, I don't have to deal with that. But I think that we just really need to sort of press on the point that people can absorb a lot more information than we think when they're faced with treatment for a life-threatening disease. Now, so what have I missed here? And let me do this quickly because I'm probably bleeding into other people's time. Limitations and advantages of each of the methods presented, easily understood. Well, easily understood for me, but I've had a lifetime of doing simulations to calculate power. So I think so. I think that there are ways to tune it. There are ways to explain it. Given the simulations, are the assumptions for the simulations appropriate? Probably. They seem to match pictures. Oh, I want to cycle back to something. I'll say it in a minute. They seem to match the pictures we've seen, et cetera. So don't let me forget a thought that's kicking around the back of my head. Should reporting of clinical dependent be on the presence of non-proportional hazards? Well, yeah. I mean, it should. The way we report the clinical benefit should be to display as much information as possible once the decision is made whether to move forward with the product. So I think that it shouldn't be linked to any particular modeling assumption. More than one summary measure, yes, the picture. Is the proposed combination test robust? It seems to be. I don't know, maybe there are simulations that people haven't thought of. It seems to be reasonably so robust. You know, in practice, designing trials in the federally funded area and otherwise, a 3 to 4% power loss is not that big a deal, really, especially since a lot of these simulations were showing 60 versus 65% power. What you really care about is up at the 90% power range. That's where you really need to know if you're missing something that's important. By design, the combination test will potentially downweight early events. Is this an acceptable approach? Maybe. It just depends what you're seeing. And other alternative methods. I'm not going to touch that because other people have thought about this a lot harder. Now, so the piece that I forgot. So the two things that I think we have to address with quote-unquote estimation, which is conveying the picture to people who are making the clinical decisions, are one, how do we help them understand what those patterns mean, and two, how precisely measured are those patterns? So, you know, we saw slides that said, here was a study at which the hazards appeared to be constant up to about 2 years, 18 months, or cross at some point. So how precise is that point being estimated? How much noise is in those pictures? That's really important because, and I don't know how to convey that necessarily, but it's very important because those survival curves are great at suppressing uncertainty. And that's what I think we need to figure out how to get across to people who are using these for decision making. What's the uncertainty there? So that's all for now. Thank you, Mark. Should I stay or sit down?
I think we're going to go ahead and stay up here. Next is Kunthal.
Hello. Thank you for inviting me. So regarding the presentation, I'd like to comment first about testing. There's a lot of discussion about weights, the use of weights in hypothesis testing. And at FDA, whenever the sponsor proposes weights, what usually comes to mind, well, the first reaction is we get spooked. The second reaction is we always try to think about testing and estimations as sort of flip sides of the same coin. So when we propose weighting survival curve, whether emphasizing early events, emphasizing later events, first of all, how do we defend emphasizing what events are important? And second, since testing and estimation is sort of, as I said earlier, flip sides of the same coin, how would you translate those weights into, let's say, the Cox regression setup? Do those weights have corresponding weights in the Cox regression? So I could imagine after I test and my test says, you know, emphasize early events, what would be the algorithm of taking those weights that emphasize early event and translating it into the data so that in the Cox regression setup, those early events are also emphasized. As Raji suggested earlier, I used to work in safety. Whenever we think about using weights, we always look towards the end result. So you use weights to do estimation, and you get a hazard ratio. How do you interpret that hazard ratio in the presence of that weights? In observational studies, we often use weights where, for example, to balance cohorts in the beginning or to balance cohorts throughout the follow-up time, that's, I think the corresponding concept is trying to make the cohorts look well balanced throughout time. So in the end, how would you interpret the results when you do estimation with these weights? And I'm not sure that the presenters have thought about this idea of thinking about the end result. Now, I have, the crossing hazards, I wonder whether the crossing hazard is, I know Renee says, don't think about the underlying process that creates the crossing hazard. But I'm wondering, when you look at the data and you see crossing hazards, is that truly crossing hazard, or is this the uncertainty of the Kaplan-Meier curve giving you the presence of crossing hazards? There are a lot of sub-process taking place, and I wonder, having any type of testing procedure that tests for a pattern without really trying to understand why the Kaplan-Meier curves behave in the way they do, I think that's a little bit too simplistic here. And so that's a good segue into estimation. So what would I do if I see non-proportional hazards, at least in the Kaplan-Meier curve, and to the extent I could trust the Kaplan-Meier curve? And what I'm about to say is probably statistical heresy, but I would, even though it's not pre-specified, it goes back to conveying the information to our clinicians, which is to consider stratification based on risk factors or prognostic factors, because those things, I think, could drive the shape of the Kaplan-Meier curve as well. And I know like at FDA, we always stress pre-specifying all these analyses, but it's really, you could only do so much in the pre-specification phase. In the end, in terms of conveying the information, we have to, I think, be a little bit more open to going beyond pre-specification and looking at some other analysis that give us a better picture of how the treatment effect is working. So I would do stratification, even possibly doing subgroup analysis, GASP, and I know that Raji's probably going to have my hide when I go back for saying subgroup analysis, but I would consider, at least in terms of characterizing the effect of the treatment, I would stratify and consider subgroups as well. Now, I have more experience with the delayed effect, as Raji alluded to earlier, at least on the safety side, because we look at cardiovascular outcomes and also prostate cancer outcomes or other cancer outcomes. And usually, in talking with clinicians, we really don't know when the separation takes place. So in terms of the design, it's really not clear to us how to specify that time point. But I think specifying some time point is better than not specifying any time point, at least in terms of delay effect. And the working group didn't really, to me, give us some sense of, if you misspecify that threshold for which the curve starts to separate, how bad does it get? And perhaps maybe the working group could provide that information later on. And I had a question about the simulation results regarding the max combo test. Satrajit mentioned earlier that I think you have 4 tests that you calculate, and you pick the one that gives you the biggest value, bang for your buck. And in the simulation results, it was suggested that the max combo test did really well, but there was also a suggestion of data-driven and multiplicity, was that taken into account in the calculation of the paragraph? Okay. That's all I have to say. Thank you.
And last of our panelists is Gideon Blumenthal.
Okay, I'm back. I guess because Mark, you owe me a beer later because you missed your train, or your train was delayed. But this, I promise, will be the last you hear from me today. So just a couple of thoughts. Just some themes we've heard today. From a design and analysis standpoint, I kind of like the idea of being agnostic to the test. Not being solely wedded to the log rank test. I could see from an agency perspective there might be some dyspepsia or discomfort. You look at log rank, or you see non-proportional hazard, and then if it's non-proportional, you start looking at other things like RMST or milestone, and then you sort of pick the winner, and whichever summary statistic looks best for the company or for the drug, that's the one that you bring forward. So I could see some problems with that that we'd have to work through. I also agree that the pictures, the Kaplan-Meier curves, they can say a lot in addition to the hazard ratio or the medians. I'm a little bit biased on this point. We've done some analysis on, for example, milestone analysis in some non-small cell lung cancer trials. We found a pretty actually good correlation between 12-month milestone survival rate and OS hazard ratio. But I think milestones are pretty intuitive for patients and for doctors. You see them now. In addition to the medians, you see them described in, for example, New England Journal papers. Well, they'll show, they'll have the dotted line to the 12-month OS or the 24-month OS. For a patient or a doctor, I think it's fairly intuitive to explain, in this trial, X amount of patients survived a year, survived 2 years, et cetera. I do also worry about once you start getting into the far tails and you have a lot of censoring, the stability of those analyses at the tails, and sometimes with irrational exuberance, investigators might overhype some of the results at the tails, where you might only have 1 or 2 patients going out that far. So some sort of baseline ground rules. Maybe it's you need to follow at least 10% of patients or 10% of the amount of expected events out that far, before describing the tail or something like that, from this working group to provide best practices, I think would be quite helpful. So that is pretty much all for now, and I could turn it back over to Mark.
I'll go to the middle over here. And if you could put back up the list of questions for the session. And I'd like to pose these to the rest of the audience. I know we have a lot of thoughtful people here, including many who have been working on these topics. While you're thinking about that for a moment, I also want to get our original speakers back up for this discussion as well. So come on up. In fact, I'd like to start with each of you, if there are any comments that you'd like to make in reaction to what you've heard from the panelists so far.
So, first of all, thank you for giving so thoughtful comments, and they're very useful. We'd just like to mention one thing, that, of course, when we looked into the methods, every method has their good and the bad side, but we're trying to look for a method which can take care of the uncertainty which Dr. Harrington was getting at. And that's what we came up. We thought our combination method gives at least that robustness over this, over this uncertainty. But definitely, the estimation part may need more thought of thinking, and I think you will know more about it in the next case study session when you will see 3 important case studies and how these estimation things work out. Maybe that will be helpful.
Tianli?
So I would just add that we really like the combination test, as you can see from simulation results. And then the combination test obviously has 4-member test. So people will naturally ask about whether there is a kind of a multiplicity issue. And that's actually where our contribution is. Our method for the max combo test right now basically addresses the multiplicity issue. As you can see from the simulation for the null scenario, because the type 1 error control is well controlled under 2.5%. So that's one comment I will quickly make. Maybe it doesn't come out very clearly in my presentation. And the other thing I want to mention is that when you have proportional hazard, the testing and the estimation, they're kind of married together, because asymptotically log rank test and the Cox pH model, they're asymptotically equivalent. But in the MPH scenario, I think what we're trying to say is that we should try to somehow decouple the two. The testing is trying to say if the drug is, if the experimental arm is doing something more than the control arm. And the estimation procedure, I think, is now completely separate issue. I think it's just how to communicate to the payer, the patient, the treating doctors what the treatment effect looks like. So it's not just one number, because if you think about MPH, one number is now not going to summarize everything. So you have to take a more kind of data-dependent approach.
One issue to follow up on is to David's point about how the pictures look really compelling and can be communicated to patients and physicians. And I know, David, you said that, well, looking at this from a statistical standpoint, you're not necessarily making decisions about regulatory approval or payment. And I do think the regulators and the payers are looking for some more objective standard that comes out of this qualitative, or this impressions from the picture. It's there's a lot of different art out there, and different people interpret it in different ways, and having some standards beyond that is really helpful. It does seem like the combination approach is intended to try to find some balance there, to use some key pre-specified statistics that would be adjusted based on some key features of the actual data that are observed in these summary survival plots. But also heard from Kunthal about maybe thinking about some ex post analysis on top of that. I wonder if there are any comments from the rest of you. I guess the concern gets to overfitting and the usual kinds of statistical errors that can arise with ex post analysis.
That comes with the territory.
Yeah.
So I don't have a good defense for that. Whenever you do ex post searching, you run the risk of being hunged, if you will. But I want to go back to Dan Liu's comment earlier about the tests being powerful. So my question is, you have Kaplan-Meier curves crossing, and your test says, okay, they're different. What do you say? Because you have to define the estimand. Somehow I think they're linked together. Your test says the curves are different, but without clearly defining what that estimand is, I'm trying to figure out how would I interpret that test result.
Because to some extent, I still think that testing and estimation somewhat should be decoupled here, because for testing, all you're trying to do is reject the null hypotheses. So that's what the testing is trying to do. The estimation, I think, coming second, and you're trying to describe what your experiences is based on the data. So I think that's just the nature of the MPH scenario. Like I said, in the proportional scenario, I think these are coupled, but in the MPH, I think you're not trying to estimate one thing. So it's out of our traditional kind of hypothesis testing realm now.
Yeah, so if I could just add a bit, I mean, I think the real problem there is that we're trying to find a relatively short sentence to describe a relatively complicated picture. And, you know, for a while, until we understand this better, we'll probably all fall short on that. That, of course, doesn't solve the problem that the agency has in approval of drugs and what the payers want to understand. And so I'm sidestepping that to the smarter people. I think in the National Press Club, I'm allowed not to answer the question that was asked, right? But any other question I think of.
So we try to do it differently in our sessions. So.
The other thing that strikes me that's important here is that Raji said something in her very, very nice presentation, but something which I don't agree with, which is that when we see proportional hazards, we know that everybody's benefiting the same. Well, not really, right? Because what we're getting is an overall population effect. And we know that really what's happening is that there are people who are failing early and people who are failing much later. And so there are real differential effects based on complicated underlying features of the disease or patterns of treatment, et cetera. And I think this is forcing us to deal with that. I think this is forcing us to say, all right, now we can't rely on that simple explanation, which was only partly true before, and we know can't be true here. And that's what's so hard. That's what makes it a difficult problem.
Yeah. So, Judith, you want to?
Yeah, I'd just like to add one thing to Tianli's comment and your question. We did decouple it, but we didn't completely decouple it. We're saying average hazard ratio, which is used with a weight, it's still part of the estimation. But the question is that solely not interpretable in this scenario, so you may need more. That's what the proposal. The dual estimates is still present.
And it does sound like there's some agreement in the group with about the need for pre-specified testing that can lead to significant results based on what are some very heterogeneous and hard to anticipate complex pictures of survival patterns. I'm not really hearing any fundamental disagreement with this approach of trying to put some effort into specifying those kinds of models up front, or pre-specifying a way of determining to a first order what kind of pattern is going on, and then also pre-specifying what kind of analysis will be done for that broad pattern. I'm not really hearing any disagreement with that kind of fundamental approach up here. It's obviously hard to figure out what the exact test combination would be, and hopefully that will get better over time as prior knowledge on different kinds of cancers or treatment responses accumulates. But I'm not really hearing any disagreement about the fundamental approach here. So people can maybe, if there are, please let us know, and if not, please keep kicking the tires. So over here.
Hi, this is Xiaoling Wang from Acerta Pharma, a member of the AstraZeneca family. My question is, I mean, when I see the very beautiful work from the working group with all the simulation stuff, but what I can see, there's so many chickens and eggs. You can see the sample size, the number of events, the hazard ratio, the duration of follow-up. They're all power. Everything is correlated. They're all. It's very hard to really. I think one of the challenges is when you design a study, especially when you're first in class, it's very hard to know where you will land. And the decisions are binary. You invest this many patients, follow this long, or not. So I think one question we'd like to know, hopefully from your working group, is and sort of more like a risk analysis that if you are wrong, how much can you afford to be wrong? I think that will be very helpful to hear. The other thing I think is important, I think it would be very helpful to hear more from the panel and also from the working group, is that if, because another day, it comes down to your treatment objectives and the measure of clinical benefit in that context. And that may be very different, for example, one of the things we work on is chronic lymphocytic leukemia, very, very long survival. And for the naive patients, the frontline patients, people trying to get to transplant, therefore, depth of response is very important. And that may be very different from a non-small cell lung cancer. Your objective is for survival. So I think if with FDA and the working group can comment on, in this complex situation, I think we're all recognizing that the field is evolving. We're willing to go beyond a one parameter situation. But how do we frame it? Maybe the answer is very different depending on your disease setting. But is there a framework that you can define, things to consider that coming out of this exercise, so that when we design studies, we can have a more a productive conversation with the FDA? Because at the end of the day, we may come across like trying to game the system. We don't want to game the system because unless you're better, you're not going to win. And we want to be better and win. We want to get the right drug approved. So any kind of guidance that you can give us, maybe come out of this workshop so that we can have the right framework to
yeah, I think it's more as a framework rather than a specific approach that could be adjusted based on some key considerations in a particular clinical context. A lot of head nodding, yes.
I think
is that where the recommendations are now, or is that going to take a little bit of extra work?
Absolutely. I think in this session we mostly looked into the analysis method, but maybe in the session 3 or 4, session 4, where we more come into this planning stage, and I guess it will answer some of the question that you posed on. But these are really very important points we acknowledge.
Yeah, so that's why our work stream, we have the analysis work stream, we have design work stream, and then the next session we're going to talk about the estimation. So I think a lot of things is going to be covered during this workshop. So it's not just about, a test to reject the null hypothesis. Hopefully we're not oversimplifying the problem. And then another comment I want to make very quickly is that the max combo test we're proposing here is not panacea. It's not just say it's one size fits all kind of approach. We're going to use that. If we can leverage some kind of prior knowledge to kind of reduce that risk, to mitigate that uncertainty. So like if you know the delayed effect is the only thing that you worry about, you don't worry about the other scenarios, then you can reduce the dimension of that max combo test, which will bring further efficiency and also flexibility of the test. So I think, so it's not saying that always you're going to use this. I think you still need to leverage some kind of prior knowledge.
Good, thank you. Thanks.
Yeah, again. So I have two questions. One is, maybe you have done other simulations, but it seems like the effect that you have chosen here are very huge effects, whether it is delayed effect or, et cetera. So have you looked at where, the effect is not that large? Yes, in the initial portion, there is no difference, but then you have difference, but not that great of a difference.
Yeah,
when you say effect, you mean effect size overall, or
yes,
effect in terms of difference from a proportional hazard assumption?
It is the relative effect that I'm talking about. Okay. So, you had a hazard ratio of 1, and then you had a hazard ratio of 0.7 instead of 0.34, something like that you have considered.
Yeah, so like I said in the simulation slide, all these simulations are informed by real trials. So we know a priori these are in the alternative space. We know the drug work. So I think for the two scenarios there, for the delayed scenario and the proportional hazard, we know, as long as it's not, the curves are separating, they are alternative. I think where it becomes a little ambiguous is when you have the crossing hazards, right? Because the crossing hazard, for some patients, they are actually getting harm, and then later on, the other patient are deriving benefits. Is that really alternative, or are the drug is really providing benefit? So I think that to your question, Raji, I think I'm not too concerned about the effect size being should be a little smaller for sensitivity for the delayed and the proportional hazard scenario, because there you just can increase the number of events to kind of compensate for that, and all the simulation conclusion, I think it will still go through. I think it's it's only the The crossing hazard scenario maybe need a little bit more thought, but there I think, again, there's ambiguity, and we don't want to just make up hypothetical numbers, say this is a scenario with crossing hazard and the drug works, because it's very hard to defend. But using the real trials, and then we have to stick with those numbers, and then we know we're in the alternative space. So that's the motivation for those numbers.
And also, the previous question was also similar to that, in the sense if it's a long term, then these events will occur much later, and how do you account for that? But other than that, you did consider, supposing for a moment we think that if there is a crossing of hazards, it's a problem with interpretation and it's difficult, there must be a subgroup or something else is going on, and those we can say maybe are uninterpretable to a certain extent, and just let's say we are not looking into them at all. You did try the other combination as well. How did that perform?
So the combination that we like the Max combo because, you can pre-specify the tests very clearly and very cleanly. So the Fleming-Harrington. Obviously, we have this, we look at the Breslow combination. We also like it because you can just specify the test of acceleration, the log rank. We also look at other combinations, say, combining the log rank with a milestone survival rate. There, I think there's a limitation, is like the other method, you have to pre-specify a time point. And that time point is something that we don't like because, it's pretty judgmental, and sometimes it's somewhat data dependent, and it's hard to kind of specify a priori. So there's this Logan 2008 paper that talks about different combination of other methods, but usually it requires some kind of specification of a pre-specified time point.
I was actually asking, you said you looked at combo 1, combo 2.
Oh, sorry.
And what's the combo 1? You presented the combo 2.
Yeah. So, sorry, I misunderstood the question. We look at only combining the log rank and the delayed treatment, the Fleming-Harrington 01 in the case studies. We didn't look at them in the simulation because we know a priori if we only combine the two, then this method is going to be better for the scenario that works. For instance, you combine with Fleming-Harrington 01, for the delay scenario, it's going to be even much better than the current combination method. But obviously, it's going to be very bad if you have a diminishing treatment effect. We didn't look at them explicitly, but I think we can easily extrapolate the conclusion from the simulation that we have right now.
So maybe I add, Raji. So if you look into individual performance, that somehow give you, if you only combine these two, in some cases, it start to work much worse.
Okay. We have about a little bit less than 10 minutes left, and I would like to try to get in. This is a great discussion with meaty questions. So let me try to get in a few more before we finish. I'll go over here first, and then there.
Yep. This is Ji Lin from Eli Lilly. So I think just a couple thoughts to add to this discussion. So I think in addition to the estimate and the testing, have you considered to come up with some measurement in terms of the magnitude of the deviation from the proportional hazard assumption? So if you look at the two cases where we have a constant hazard ratio but with moderate effect size versus where we have a non-proportional hazard model data there, but with a subgroup receiving super good efficacy, with the other receiving none or even negative efficacy. Even though the p-value come out from either the traditional test or the novel test that we discussed today, with the same p-value, they really mean very different scenarios. And I would assume that a physician will have their treatment decision quite different when they are facing patient. So I think in addition to what we have discussed today, I think another angle to kind of touch this question is that you will think from the physician perspective, there's a need, I feel, that to distinguish these two cases. And then on top of that, I think there could be a certain type of penalty put on this. So if in a subgroup of population where the hazard ratio is even greater than 1, then even the overall adjusted p-value kind of comes out as a significant. Does it really justify to treat all the patients with the knowledge that only a subgroup will benefit, and the subgroup may even kind of get a negative benefit from the treatment. So just I think to the panelists and the speaker, is there any kind of literatures or methods that you have considered to address this?
So maybe I can try to address that. So the methods that we try to present here, so just to give you context, it's still in a most confirmatory trial. We talk about those subgroups are there, but the primary analysis is still on ITT. So what we're trying to propose that having a combination test at least gives you to a position where you start to talk about other things like subgroups and others, but you still need the first regulatory gateway to pass. But of course, that's a more complex and more dimensional problem to look into when you have a subgroup differential effects, and that may complicates your final labeling or final effect size that you report. But what the combo test we're proposing on, that's basically give you the first stage to come into the picture.
Next.
Hi, this is, I don't know if this is on. Okay. Hi, this is Ray Lin from Genentech Roche. So I'm trying to address some of the questions because I'm also a part of the working group. So one of the points that was brought up is about how to report the treatment estimate effect. In fact, Larry Liang and I proposed a method, this weighted log-rank, weighted Cox model. That's actually mathematically equivalent to the weighted log-rank test. And in this model, we report the hazard ratio as a time-varying effect. So rather than reporting a constant hazard ratio as one single number over the whole time course, we can propose estimate kind of like a time profile of the hazard ratio. So just like the plot that Raji showed very initially, that the hazard ratio could be a function of time, so it could be changed over time. So that could be one way to try to address this question. And also we published a paper late last year. In that paper, we also looked at the misspecification. So what is the consequence of if we misspecify the weight function? If it's different from the truth, then what is the bias and what is the power loss in that case? And the third one is related to Raji's question regarding kind of the median treatment size. And so in our working group, we use some of the case studies. So the hazard ratio we observed are pretty good, so very substantial treatment effect. But in our paper, we also look at more moderate treatment effect, like hazard ratio around 0.68 or 0.72, that kind of effect. And you will really see there could be a pretty substantial advantage of using the weighted log-rank test. So this is just want to share with the team, and then we will also, this has been proposed inside the working group, it's under discussion, so we will also love to hear feedback from other people as well. Thank you.
Great. Thank you. I appreciate the working group members adding to this discussion too, and again, what sounds like we'll come back to some of these issues with the case studies after lunch. Next.
Thank you. Luis Rojas, again from Acuivia. Thank you so much for the person who just spoke about the elephant in the room, to consider the variability in the hazard ratio. I think that is the most appropriate approach, and that answers somehow question 2 about the assumption for the simulation. I think that those simulations should consider that variability, and as well the effort to do subgroup analysis. I think that is obvious. So my question was already answered, and I thank you so much for your clarification. I think that thank you for putting the light in the elephant. Thank you.
Yeah. I don't know if either of you want to comment further on this elephant, but that is the part of the big challenge here is just not knowing what the true hazard patterns are. Yeah.
But that's true for, I mean, when you're in a design stage, right? I mean, that's a challenge for any trial. When you never know the truth, and the whole idea was to mitigate the risk in proper way. And that's what we are trying to have. And that's what we're coming in, that a hazard ratio is not an adequate measure, and you need something more to express it.
Yeah, and I think while this group certainly has its hands full with just describing what to do at this stage in the investigation and development process, putting this in the context of how the conclusions from this study can then inform future work, maybe what would be highest priority for post-market studies or for further studies will be helpful as well.
Because I think so far what we look at is this method looks very promising and robust and agnostic to the different scenarios of MPH, but we haven't told you how to use it to design a trial just yet. So just as a plug, I mean, you will see
And
more
how it
in
fits into
Kevin's
the rest of the
session
process.
this afternoon. So exactly incorporating what we learned from the simulation work and how to incorporate that into the design stage up front.
Okay, we've got 2 more people with comments. I'd like to get to both of those. So if you all don't mind, we maybe do both of your comments and then try to answer after.
Boris Friedland, NCI. Quick question on simulation. So from what I remember, the pictures, you simulated situations from relatively advanced disease, so curves went down pretty steeply, pretty fast. So I just wanted to basically ask for comments on this situation that basically the statistics you discussed are the weighted log rank tests. So the weights and statistics are defined by pool survival function. So as long as the survival function goes down deep, you have the 4 tests are really different from each other. But if you look at the, say, adjuvant setting, then actually will be relative, where pool survival function will be pretty flat. Then there will be almost no difference between the tests, right? Between the 4 weighted tests. So the performance of you demonstrated might not apply in this setting.
Okay. Very quick.
So to some extent, our large sample size, I mean, scenario, so we have this 1,200 event, and then we only required 210 events out of those 1,200 sample size. To some extent, it's trying to address the scenario for the adjuvant setting. So I think the general conclusion still hold there.
Thank you, Cyrus Mehta from Cytel Corporation. I think it's really important to separate out the question of hypothesis testing from estimation. In very simple cases, you can have an estimate that is absolutely consistent with the hypothesis test in the sense that the confidence interval will exceed the hazard ratio of 1, and if you have a statistically significant level. But usually, but in these more complex situations, that's not the case. So make sure that the testing method is rigorous, because that's what the regulatory authorities need for their job. That has to be absolutely a deal breaker if you can't have a strong control of type 1 error. In terms of estimation, I go with what David Harrington said. You should really look at all different ways, try to make it clinically meaningful, and try to explain to physicians what it means, and not boil it down to one number. It's just a comment.
Thank you, Cyrus, I think.
Okay, good comments to end on. We are going to take a break for lunch for an hour now. When we come back at 1, we're going to focus on applying these methods to a range of completed trials. Now, that is retrospective, so you have the advantage of seeing all the data and seeing what the plots look like and then imposing some methods after the fact. But I do want to recognize this tension between wanting to respond to the fact that proportional hazards is likely to be incorrect in many of these models, but what exactly the alternative hypothesis should be is also not likely to be clear at the time that you're doing the trial, and that does create, from a regulatory standpoint, this risk if you don't specify in advance of coming back and fitting some data rather than coming back, but then really having a clear understanding of the mechanism, the truth that's out there. So let's come back to some of these very important issues after lunch. Thank you all for an excellent discussion. Thanks to the panel. So lunch is on your own. If you need information.
Okay, we'll go ahead and get started. I'm Greg Daniel. I'm Deputy Director in the Duke Margolis Center for Health Policy, and I'm going to give Mark McClellan a bit of a break for this next session. It's kind of a challenge to moderate every session throughout the day, so we're going to give Mark a little bit of a break. So for this next session, presenters will use case studies to demonstrate how the proposed approaches that we discussed this morning can be applied in real-world trials and how they compare to the current statistical tools already in use. We'll begin by a series of brief case study presentations, followed by a discussion. In total, we have like 9 people coming up here to talk in a short amount of time. So I'm going to make sure that we stick to the timing. So if you're presenting or you're a reactor, try to stay on time, and I'll help you out with that. So we'll go ahead and get kicked off with Bo Huang, Director of Biostatistics at Pfizer. Bo, come on up. Thank you.
Hi. So in session 2 this morning, Satyajit and Tian-Le gave very nice presentations on the different methods in the presence of non-proportional hazard, and also the summary of simulation results, comparing different methods in various NPH scenarios. In this session, we'll present some case studies applying these methods to completed oncology studies. Next slide. Okay, so there will be 4 case studies to be presented, covering different non-proportional hazard scenarios. I'll be the first speaker presenting the INNOVATE trial, which is a delayed effect and long-term remission case study. Kay from BMS will present another different delayed effect and long-term remission case study. After that, Larry from Roche will give a presentation on diminishing treatment difference case study, and this is a study actually sponsored by Eli Lilly. And lastly, Praloy from AstraZeneca will present a crossing survival curves case study. So first, I'd like to acknowledge the entire NPH team. The team has done tremendous work over the past year and a half. I would also like to thank a few colleagues from Pfizer for providing the INNOVATE trial data and also getting internal clearance to use the data. So the INNOVATE trial data, the trial is an open-label, 2-arm randomized phase 3 trial in patients with relapsed or refractory acute lymphoblastic leukemia. There are 2 primary endpoints, complete remission and overall survival, with alpha equally split between the 2 endpoints. The sample size is 326, with 1-to-1 randomization to either inotuzumab, the experimental arm, or standard chemotherapy, the control arm. The power for overall survival is 80% to detect a hazard ratio of 0.67 at a 2-sided alpha level of 0.025. So here are the trial results of the primary endpoint. For complete remission, the p-value was highly significant with a couple of zeros after the decimal point. For overall survival, the results are quite interesting. So here is the Kaplan-Meier plot of the study for OS. The blue curve is for the treatment arm. The brown curve is for the control arm. As you can see, the treatment curve is always above the control curve, but the separation is pretty small in the first 15 months. And if you take a closer look, there's actually pretty good separation in the first 5 to 6 months, but the separation diminishes afterwards until month 15, so indicating a potential crossing hazard pattern. And after 15 months, there's pretty wide separation, and the separation is maintained until the end of the trial. And also the curves for survival are pretty flat at the tails, indicating there are very few events. So this is a long-term remission pattern. In terms of hazard ratio and p-value, we have both the stratified and unstratified results. And for unstratified, the hazard ratio is 0.748 with a one-sided p-value of 0.01. This is the Schoenfeld residual plot. As we know, the Schoenfeld residual plot is a diagnostic plot for testing a proportional hazard assumption. Since the Schoenfeld residual test, or the GT test, is sensitive only if there is a linear trend. As a result, the p-value cannot be used as the sole basis for detecting non-proportional hazard. We should also look at the graphical presentations. In principle, the Schoenfeld residuals are independent of time, and the plot that shows a non-random pattern against time is evidence of a violation of the proportional hazard assumption. If you look at the plot on this slide, it seems to be there is a quadratic fit of the scaled residuals over time, so indicating a potential deviation from the proportional hazard assumption. So here are the results of the proposed test and estimation. We look at the log-rank test, the Fleming-Harrington class of the weighted log-rank test, the two combo tests, A and B, and also the RMSD test for the RMSD difference. So these are described in details in the morning session by Satyajit and Tianle. And for FH01 put more weight in the late events, FH11 put more weight in the middle, and FH10 put more weight in the early events. The p-values are one-sided. The treatment estimate are hazard ratio or weighted hazard ratio for the weighted log-rank test or the max combo test, and RMSD difference for the RMSD test. So in this trial, we at one-sided alpha level of 0.025, the log-rank test, FH01 test, the two combo test, and the RMSD test are significant, with the RMSD test having the smallest p-value. What comes as a surprise is that if you look at the combo test, the p-values of the combo test have higher p-values, are higher than the p-values of the log-rank test. The main reason for this is because although there is a late separation, but the number of events after the late separation is pretty small. So putting more weight on the late events, that doesn't really move the needle. And also, as I said, there's a potential crossing hazard pattern in the first 12 months. And of course, the multiplicity adjustment also increases the p-value. If we divide the follow-up time into 3 intervals, 0 to 6 months, 6 to 12 months, and after 12 months, we can see that the piecewise hazard ratios are quite different within each time interval. We have a piecewise hazard ratio of 0.76, 1.2, 0.3 in each time interval, respectively. So this also indicates a potential deviation from the proportional hazard. And this provides useful information on top of the overall hazard ratio of 0.748, which, by the way, could be misleading and difficult to interpret because we know the treatment effect varies over time. The RMSD difference is also a very useful summary because it is both statistically meaningful and clinically meaningful, and it doesn't depend on any model assumptions. And in this trial, RMSD difference of almost 4 months is the expected gain in life expectancy between the treatment arm over the control arm within the window of follow-up. Since we have a long-term remission pattern after 15 months, the milestone survival or the survival rate at a specific time point could also be very useful to capture the long-term effect. The 2-year survival rate is 22.6% for experimental, 9.6% for control. The 3-year survival rate is 22.6% for experimental and 5.7% for control, with non-overlapping 95% confidence intervals. So here are the key takeaways from the first case study, the Innovate trial. We have observed a late separation of Kaplan-Meier curves after around 13 to 15 months. There's heavy censoring. There's also a small number of events, and mostly in the control arm, after a late separation of around 15 months. Despite that, we follow patients up to 40 months. We have, at one-sided alpha level of 0.025, we have observed statistically significant difference in unstratified log-rank test, the FH01 test, the 2 max combo test, and the RMSD test, with the RMSD test for the difference having the smallest p-value. The max combo tests have higher p-values than the standard log-rank test potentially because of a small number of events after 15 months, possible crossing hazard within the first 12 months, and also the multiplicity adjustment. If we look at the estimation and interpretation part, which is equally important as hypothesis testing, as we heard from the panel discussion this morning, the RMST test, the RMST difference, the RMST is which can be summarized by the area under the Kaplan-Meier curve, is easy to interpret, and it doesn't depend on any model assumptions. And in general, the mean is more informative. Is a more informative global summary than the median, considering that the sample mean is the sufficient statistic for a number of common distributions within the exponential family, and the median is just an arbitrary percentile, which may be subject to a lot of variability. And for this trial, we have a mean difference of 4 months versus a median difference of 1 month. The second useful measure is the piecewise hazard ratio, which can be quite informative for understanding the changes in treatment effect over time. So offering a temporal profile on top of the overall hazard ratio. And lastly, the milestone survival, or the survival rate at a specific time point, is also useful to capture the long-term benefit at clinically meaningful time points, such as 2 years, 5 years. It is particularly useful for late separation and long-term remission cases, such as the INNOVATE trial in this case study. So with that, that concludes my presentation. I'll pass the ball to the next speaker.
Okay, sure. Great. So our next presenter is Kei Tatsuoka, full development statistical lead at in immuno-oncology at BMS. Thank you.
Okay, likewise, thanks to the organizing team for inviting me to give this case study. Next slide, please. So we actually looked at 3 ipilimumab studies. The one I'm going to focus on is a phase 3 study comparing 2 doses of ipi for melanoma, 10 mg per kg and 3 mg per kg. The patient population was patients with untreated or previously treated unresectable stage 3 or stage 4 melanoma. OS is the primary endpoint. The sample size was 727 with 1-to-1 randomization, and it was powered at 90% to detect a relatively modest treatment effect, hazard ratio of 0.744. There was an interim analysis that was planned in the protocol and discussed in the publication. At 67% of the planned events, it was canceled because the accrual was very quick in this study, and it was felt that there would not have enough events, enough follow-up for this number of events. However, you'll see, I'm mentioning this interim analysis because we're going to conduct one post hoc anyway, just to see some of the effect on the statistics. Next slide, please. This is the curve, the survival curve. A couple of things of note in this. There was a long delay of 9 months comparing 2 doses of ipi. Perhaps this may be more representative of the type of long delay we're going to see as we look at more IO plus IO combinations compared to IO. The second point of fact is there is a long patient, I wouldn't call it a plateau at this point, it's at 4-year follow-up, but still there are a large number of patients at risk very late in the study. Next slide. These are the 2 other studies we looked at, and I won't be presenting the results, all the results for these 2 studies, but we'll try to summarize them at the summary. The first is an adjuvant melanoma study. Again, there's 3 endpoints: OS, recurrence-free survival, and distant-free metastatic survival. Again, all 3 endpoints feature A little bit of a delay, not as much as the one that we're presenting, the 169 study, but some delay and long survival plateau. The other study is our Checkmate 024, our first-line metastatic melanoma study for ipilimumab, which formed the basis of the approval. Again, some delay, large number of patients at risk, going up to 5-year follow-up. Next slide, please. The Schoenfeld residual plots, well, for 169, the dose-ranging study on the left, you do see evidence of non-proportional hazards. The p-value notice, and also on the right is the Schoenfeld residual plot for the first-line metastatic study. The p-value should be interpreted with caution. It's not representative of any non-proportional hazards. Next slide. Okay, so now let's go through some of the results. I won't go through every line, but keep in mind that this particular study had a delay and had a large number of people at risk with very long follow-up. So you can see from here that the weighted log rank test, which emphasized the late events, that's 0, 1, 1, 1, they do quite well in this study. The log rank test does not, it's significant, but if you look at the stratified estimate on the right, the upper 95% CI is 0.99, and unstratified, it doesn't cover 1, has a ratio of 1. So from here, the RMST also does well, so you can conclude that the weighted log rank test, which emphasize the late events, and RMST do well in this particular study. The piecewise hazard ratio, we divided into 2 intervals, less than 9 months. Remember, there was a 9-month delay and greater than 9 months. And you can see, again, no effect in the first 9 months, hazard ratio of 0.957, a modest effect, 0.737 in the second portion. Next slide, please. So we conducted a post hoc interim analysis with 20-month follow-up. And if you go back to if you remember, the delay was 9 months, so that's a large proportion of the follow-up of 20 months that we're assuming here. So I think it backs up or supports some of the simulation results that we saw presented earlier this morning, namely the log rank test at these nominal p-values it was not significant, 0.051. The weighted log rank test, which emphasized the late events, they do well. The combo test does well, p-value 0.0142. The estimates of the confidence intervals do not cover 1, of the hazard ratio does not cover 1. RMST does not do as well. I think that is consistent with the simulations, the p-value of 0.1111. Next slide, please. So let me sort of summarize both in words. You can read what was significant and what was not significant in the slides here. But we looked at 3 studies with melanoma, ipilimumab in melanoma with long-term follow-up, some delay. In the first-line metastatic study, basically all the weighted log ranks tested well, including log rank test, RMST. There wasn't much to differentiate these tests that emphasized the log rank test or the ones that emphasized the late events. And RMST did quite well in the first-line metastatic melanoma study. Also, similarly for the adjuvant melanoma study, all the tests did pretty well. It was only when we came to the study that had the very long follow-up, 169, the dose-ranging IPI study, that we began to see the differences. The log rank did not do as well. The weighted log rank test, which emphasized the late events, did well, including the combo test. RMST did well as well. But when in the context of planning an interim analysis for this 169 study, the dose ranging study, RMST did not do as well in the interim analysis, whereas the log rank test, including the combo test, which emphasized the late events, did quite well. So that's what I have. Thank you.
Great, thanks, Kay. Our next speaker is Larry Leon, Principal Statistical Scientist at Genentech.
So I move the slides. Okay. So first, I'd like to thank the Lilly team for the opportunity to present their data. And of course, any errors would be due to Lilly. So this is an exploratory phase 2 trial in the metastatic prostate cancer setting. So their primary endpoint was composite progression-free survival with key secondary of overall survival. As I mentioned, it's an exploratory phase 2, randomized, approximately 130 patients randomized equally to both treatment arms. And the Kaplan-Meier curves suggest pretty consistent benefit early on with somewhat of a merging curves. It seems like they start to merge around the 13-month time point. Definitely by the 15-month time point, they seem to have merged. And then you see the cumulative hazard plot showing a similar pattern. A formal test of the proportional hazards assumption, the Gram-Thurmond test, as others have nicely explained. Here in this study, it's actually significant, around 0.025 is the p-value for evidence of non-proportionality here. The test statistics applying so the Cox model, the one-sided p-value is around 0.028. So not formally meeting significance in this exploratory phase 2 trial. The Cox model hazard ratio is around 0.7. Looking at across the different weighting estimation approaches, the 1-0 is the maximizer of the combination test with the maximum combination test around p-value of less than 0.01. That corresponding weighted Cox model estimate using the 1-0 Fleming-Harrington weight, which again emphasizes, well, in this trial, it emphasizes later differences, earlier differences rather than later differences. So with the focus on early differences over later differences, that corresponding Cox hazard ratio estimate is 0.55. And then you see the confidence interval of 0.34 to 0.9. The piecewise hazard with a cut point of the 0 to 3 months, that's estimate is around 0.55, and then from 3 to 9 months, 0.59, and then after 9 months, 1.4. So I think the time periods between the first 3 and 9 months are fairly consistent. And the RMST, in addition, suggests benefit, on average around a 2-month improvement in average PFS. And I think you see similar patterns with looking at the survival Kaplan-Meier curves at specific time points, 3, 6, and 9 months. So for the OS analysis, which is again a secondary endpoint, pretty much exhibits similar pattern with somewhat a consistent benefit early on, and then definitely seems to be merging around the 15-month time point. Here, though, there doesn't seem to be too much of evidence of non-proportionality, but keep in mind, though, this Gram-Thurmond test, at least in some of the simulations that I've seen and that my colleague Ray has seen, the GT test is not really that powerful. So for example, in some of the simulation settings with the 3-month separation, even though it was a pretty strong separation, I think that power is less than 50% to formally reject using the GT test. So the GT test, so there could indeed still be underlying non-proportionality here, but just a power issue, especially with only 130 patients. I just want to comment on that. And so here, no matter what, across all the analysis though, in this OS analysis, there doesn't seem to be any evidence of any differences across the different analysis methods. And then and the RMST also suggests, doesn't seem to be indicating any difference. So in summary, the max combo test and the different weighting approaches seem to have some promise and potential in identifying non-proportional hazards effects. That is, effects that potentially vary over time. And as you saw in the application of the log rank and Cox, that didn't seem to kind of pick up any differences here, whereas the weighting approaches seem to indicate some differences in the PFS endpoint. Thank you.
Okay, great. Thanks, Larry. Our next speaker is Pralay Mukhopadhyay, Senior Director and Biometrics Team Leader, Immuno-Oncology Group at AstraZeneca.
Thank you. I'm very happy to be here presenting the AstraZeneca data on IPASS study on behalf of the Non-Proportional Hazards Working Group. So just a brief description of the study. So this was the study of Iressa versus paclitaxel carboplatin in first-line non-small cell lung cancer. The patient population was Asian patients with first-line lung who are either never or former smokers. The primary endpoint was PFS in all comers, and overall survival in all comers was a key secondary endpoint. The trial was powered. The trial randomized approximately 1,200 patients equally in both arms and was powered to first demonstrate a non-inferiority with a non-inferiority margin of 1.2. And then if NI was demonstrated, then the goal was to continue testing for superiority. So these are the results that have been presented in the New England Journal of Medicine. So if we focus first on the curve on the left-hand side, that represents the PFS curve. And you'll notice that at about approximately 6 months, there's a crossing of the PFS curves. So for the initial 6 months, the treatment effect was favoring the chemotherapy arm. And then beyond that, it started favoring the experimental arm. The overall hazard ratio that was reported was 0.74, which was still highly statistically significant. And then if we look at the curve on the right-hand side, that's the overall survival curve. I think it's showing a similar feature, at least visually, with a crossing at approximately 8 months. The overall hazard ratio that was reported was 0.91, which did not reach statistical significance. So for the case study, we reanalyzed the data set, and when we did so, we had to exclude around 67 patients, primarily because of withdrawal of consent since the publication of the New England Journal of Medicine. However, this did not impact on the interpretability, because if you see the plot on the left-hand side, the PFS curves looks almost identical to what was presented in NEJM, as well as the hazard ratio from the unstratified Cox model was 0.73, very much consistent with the previous reporting, with still a significant result. If you look at the plot on the right-hand side, so you see that the Schoenfeld residuals, as described in the previous case studies, they show a non-random pattern over time, which would suggest violation of the proportional hazards assumption. So these are now the test results for the PFS analysis, and I'm not going to go through all the numbers individually, but perhaps a couple of points to make. So all the tests that we looked at, which would be the log rank, the Fleming-Harrington 01, 1011, the combination, and the RMST. So all except for the Fleming-Harrington 1-0 was statistically significant, and I think that would make intuitive sense because the Fleming-Harrington 1-0 is putting more weights in the earlier part of the curve where there wasn't a benefit for the ERESA arm. The second point is that in this example, the combination test picks up a weight of 0.1, and I'll come back to that during the end of my discussion. And just through visual inspection, we figured that perhaps presenting a piecewise hazard ratio, choosing the first interval up to 6 months, and then the following interval beyond 6 months would make intuitive sense. And so if we look at those estimates in the first 6 months, it appears from the point estimates that the treatment estimate, the hazard ratio effect is a little bit over 1, but beyond 6 months, there seems to be a clinically meaningful effect in favor of the ERESA arm. So now if we look at the overall survival results, visually, I think it shows a similar characteristics as the PFS curve in terms of the crossing survival hazards. Again, when we ran our analysis, the unstratified hazard ratio is 0.9. The p-value is slightly over 0.05 in this case, so still not statistically significant. And the plot on the right-hand side shows a similar picture in terms of the non-random pattern of the Schoenfeld residuals over time. So I think, despite the p-value from the GT test that Larry was mentioning, which I agree with him, I think it also shows that the proportional hazards assumption was likely violated over here. So let's take a closer look at the survival results now. So if you look at the log rank test, it misses the statistical significance just by a notch. And if we think in terms of statistical significance, the only two tests that reaches statistical significance is the Fleming-Harrington with weights 1-1 and the combination test with 1-1. And now, the other thing to notice over here is that if you look at the p-value from the Fleming-Harrington 1-1, it's 0.009, and the combination is a little bit larger with 0.17. And I think that point has been well described in Satyajit and some of the previous talks because of the adjustment for the multiplicity. So, perhaps, two points to note over here in this example. When we looked at between the PFS and the overall survival results, the combination test was able to adapt itself a little bit in terms of selecting the weight. So in the previous example, it was selecting 0.1, whereas on this one, it is selecting a 1.1 as the weight. Now, if we think of the Fleming-Harrington 1.1, visually, and I think Satyajit had a nice slide in the earlier part of the description, visually it puts more weights in the middle part of the curve. So we figured that perhaps it would be informative to present the hazard ratios in 3 intervals, and we divided it into less than 8, 8 to 35, and greater than 35 months. So if you look at these point estimates now, the hazard ratio in the first 8 months seems to be about 1 with no treatment effect. Between 8 to 35 months, it's about 0.85, so a modest treatment effect. And then beyond 35 months, it's greater than 1. However, from the survival curve, there was a lot of censoring at the tail of the curve, so the point estimate towards the tail, may be difficult to interpret. Okay. So, key takeaways from this case study: in terms of PFS, other than the Fleming-Harrington 1.0, all the tests were significant. However, the real question beyond that point is, once that significance is observed, how do you quantify that benefit? Because neither the hazard ratio as a single estimate, or even I would say the difference in restricted mean survival is not necessarily a good summary. The piecewise hazard ratio, obviously, we computed it based on visual inspection, and there was no way for the study planners to know a priori to set up a piecewise hazard ratio within 6 months or greater than 6 months. But as far as describing treatment benefit, it can be informative. And a very similar situation for overall survival, although from a statistical significance perspective, only the weighted log rank and the combination test was significant, but I think the points that were mentioned before, when you are a priori planning the study, there's no way to select a weighted log rank of 1:1. So perhaps thinking of a combination test that is selecting a couple of weights based on the plausible alternatives could be a good practical way to address some of these problems. Okay? So perhaps a couple of closing thoughts, at least from those of us who were working on this working group from an industry perspective, and the motivation of the combination test. So I don't think there is any debate in this room that there will be many common clinical settings where we will observe non-proportional hazards. Okay, and I don't think from the perspective that we were coming from, the idea of the combination test was to find a statistical way of coming with the smallest p-value or designing trials with smallest sample size, but it's mostly to take into account the clinical context and sometimes the MOA that researchers wouldn't have any knowledge of prior to designing the trials, and which is also illustrated in some of these case studies. So our goal was, the first step is to see if there is a statistical evidence of difference between treatment arms, i.e., is there a treatment effect? Now, once the treatment effect is determined, and that needs to be, the test needs to be a priori set up with the right alpha control and so on. But once that is determined, I think the question of how to define that clinical benefit and how to communicate that clinical benefit may be a slightly separate one that sometimes needs to be decoupled. And I think the IPASS case study is a nice example of showing why a single measure often wouldn't make sense. So let's look at the PFS results over here. So a PFS has a ratio of 0.73, and it'll often be reported as a 27% reduction in the risk of PD. But is it still meaningful for patients who are progressing in the first 6 months, where clearly the risk is in favor? The risk was greater in the experimental arm. The piecewise hazard ratio. So if one were to communicate based on that, you could say, well, if you in the first 6 months of treatment, you could potentially be in a slightly increased risk of PD. However, if for those that can remain progression-free beyond 6 months, there's a significant advantage of treatment at that period. So that that may be difficult to define a priori, but sometimes it's a simple message to communicate to a broader audience. We looked at the restricted mean survival, and so how would you communicate that? So you can say on a 21-month follow-up period on this trial, on average, you'd expect patients in the ERASA arm to be 1.4 months longer progression-free than in the control arm. I think it's less susceptible to NPH assumptions, but from an overall interpretability given the shape of the curve, that also does not make sense, that does not make intuitive sense, and perhaps considering multiple truncation points would be helpful. And lastly, the milestone PFS has been discussed quite a bit. I think it is informative, but I think it's also we need to keep in mind that it's a single point in the curve and more susceptible to variability. So perhaps presenting multiple the PFS rates at multiple time points would make more sense, and I think we had received similar input in earlier part of the presentation. So that concludes my discussion. Thank you very much.
Thanks, Pralay. Our last presenter is Cindy Gao, mathematical statistician at CDER and FDA.
Excuse me. So good afternoon. I'm also going to present some case studies for analyzing the time to event endpoint when there is a non-proportional hazard. Okay, great. This presentation reflects a view of the author and should not be construed to represent FDA's views and policies. So the first case I'm going to discuss is a randomized open-label phase 3 study in patients with previously untreated multiple myeloma who are either older than 65 years, either 65 years of age or older, were not a candidate of the stem cell transplantation. In this study, about 1,623 patients were randomized at 1:1 randomization rate to either of the two treatment arms, treatment arm 1, treatment arm 2, or the control arm, arm 3. In this study, for illustration purpose, in this presentation, we're going to only include the data from the arm 1 and arm 3. In arm 1, patient only received the treatment. In arm 1, patient received the treatment until disease progression, and in arm 3, the control arm, patient received the control therapy only up to 1.5 years. For this study, it met the primary objective. The PFS, the median PFS time, is 25.5 months versus 21.2 months control versus treatment versus control. Using the Cox proportional hazard model, the hazard ratio is 0.72 and gives a log-rank test gave the statistically significant test result. However, looking at the Kaplan-Meier curves, we can see the Kaplan-Meier curves overlaps from the beginning until about 1.5 years, which suggests some degree of the non-proportionality, and indicate the hazard ratio from the Cox regression model and the convention median may not capture the treatment effect. We also examined the non-proportionality in the hazard rate using the Schofield residuals, which showed a deviation from 0 in the slope, in the pattern in the slope of the residuals. So due to the driven by this non-proportionality, we performed some post hoc analysis using alternative measures. The max combo test shows a statistical test result consistent with the log-rank test. And the restricted mean survival time analysis showed that with this follow-up time duration, a patient in the treatment arm is expected to have a progression-free survival time about 4 more months than a patient in the control arm. With the heavier weights with heavier weights on the later events, the weighted hazard ratio is 0.56. And using the piecewise Cox proportional hazard model, the hazard ratio is about 1 before the 18 months and 0.63 after 18 months. The second example we're going to discuss is also a randomized phase 3 study. It's an open label open label phase 3 study in patients with metastatic or recurrent non-squamous non-small cell lung cancer. In this study, 582 patients were randomized with 1:1 ratio into the treatment arm or the control arm. As one of the secondary endpoint, PFS had the median about 2.3 months in treatment arm versus 4.2 months in the control arm. However, the hazard ratio from the Cox model regression gives a 0.92 and favoring the treatment. The Kaplan-Meier curves of the progression-free survival were shown on the left side of this figure. At the beginning of the trial, a higher proportion of the subjects with a PFS event were observed in this trial in the treatment arm versus the control arm. The Kaplan-Meier curves appeared to cross approximately at 7 months, with the separation favoring the treatment beyond this time point. The right figure shows the hazard ratio over time, and we can see estimated hazard ratio is above 1 at the beginning of the trial, and is close to 1 at the beginning of the trial, and reduced to below 1 over time. Here in this plot, in the plot of the hazard ratio over time, we added the red lines representing the hazard ratio from Cox regression models. The hazard ratio is closer to 1 with the upper bound about 1. For comparison, in this figure, we added the weighted hazard ratio from the max combo test in the confidence intervals. heavier weights were given to the later events, so the weighted hazard ratio estimate becomes smaller compared to the hazard ratio from the Cox regression model. And in the end, here we also add blue lines representing the hazard ratio estimate from the piecewise regression. The hazard ratio is above 1 before 6 months and smaller than 1 after 6 months. So similarly driven by the non-proportionality, we performed some potential informative alternative analysis methods on the data. Compared with the p-value of 0.31 from the log-rank test, the max combo test gave a smaller p-value of 0.001, less than 0.001. For treatment effect, restricted mean survival time shows that with this follow-up time, a patient in the treatment arm is expected to live 0.4 months longer than the patient in the control arm. With a heavier weights given to the later event, hazard ratio is equal to 0.64. And using the piecewise Cox regression model, the hazard ratio is 1.25 before the 6 months, decreased to 0.34 between 6 and 12 months, and further reduced to 0.23 after 12 months. So in these examples, we perform the analysis retrospectively on existing studies. Ideally, the statistical analysis would be pre-specified in the statistical analysis plan, at least to some degree. This would also impact the study design and also including the power calculation and how interim analysis should be performed, including the futility analysis and also the efficacy analysis. And the difficulty exists in the alternative measures, such as how to choose the weight, how to interpret the weight for the weighted hazard ratio, cutoff time point for the RMST, and also the cutoff point for the piecewise exponential. And yeah, that's it.
Great, thanks, Cindy. I'm going to go ahead and invite our panelists up. So we're going to try to fit 9 people up here. So just to give you some orientation, I'll ask the reactors, the 4 reactors, to sit in the first 4 chairs, and then our presenters to come on back up in the last 5 chairs, at least to help me orient yourselves. So for our reacting panelist, Lijun Zhang, Senior Statistical Reviewer in the Division of Biometrics 5, Office of Biostatistics at CDER, FDA. Eric Rubin, Senior Vice President and Therapeutic Area Head, Oncology Early Development at Merck Research Laboratories. Susan Halabi, Professor of Biostatistics and Bioinformatics at Duke University. Robert Brown, Principal Biostatistician at Syneos Health. And then we'll also have our presenters back up here. So if you presented, please come on back up. Thanks. Okay, so a lot of results were presented, and I think we've learned a lot from at least the real-world case examples that were used to provide more a better way to test the combination test from a hypothesis testing perspective, but also to look at ways for describing the treatment effect in terms of a clinically meaningful way to describe the results. So I'd like to just ask our initial panelists here, your reactions, your conclusions to these tests, both from what do you think about this combo test, but also as we're developing, and I think earlier this morning, folks have mentioned the need for more of a general framework to guide considerations in the context of non-proportional hazards. How do you think about approaching hypothesis testing as well as describing the treatment effect? So let me start off with Lijun for your thoughts. I have some slides, so maybe I can... Oh, sure, yeah.
So I also prepared some slides so that I can show you the results of applying those proposed methods on an FDA database. That database has 25 randomized clinical studies. So basically in session 2 and session 3, we are discussing two major questions. The first is how to test the treatment is statistically better than the control. The max combo test has been proposed versus the traditional log-rank test. The second question is how to summarize the benefit magnitude. Several metrics have been discussed, including the traditional hazard ratio, weighted hazard ratio, piecewise, RMST, and the milestone rate, etc. So in this session, several cases with different non-proportional hazard patterns have been discussed. In most of the cases, the combo test had a smaller p-value compared to the log-rank test. The multiple methods of effect size estimation have been used, but interpretation of each is different. There is no consensus on the optimal method yet. So here, I would like to show you some results of applying those methods to a FDA database. The database has 25 randomized clinical studies submitted to the FDA from 2003 to 2016 for the treatment of metastatic non-small cell lung cancer. There are a total of 27 randomized comparisons and about 20,000 patients. The database includes both positive and negative studies. Based on the residual test and the plot, we identified 13 studies with PFS violating the proportional hazards assumption. So first, we test each comparison using both the combo test and the log-rank test. The x-axis here represents p-value from log-rank test, and the y-axis is p-value from the combo test. Green dots are studies with the p-value assumption holds, and the red dots are studies with non-proportional hazards. So you can see from this plot, for studies under proportional hazards, p-values from both tests are similar, and the dots are around the diagonal line. While for studies under non-proportional hazards, the p-value from the combo test tends to be smaller compared to the log-rank test p-value. But the question here is that the combo test uses different weights, and the weights used suggest that some events are more important than others. It's crucial to have adequate clinical justification for the weight, rather than just based on the data. So we also tried different methods to summarize the treatment effect for the lung cancer studies. Here we used the 4 studies as examples. The left 2 are studies with delayed treatment effect, but the first one could also be considered as a study with crossing hazards. The right 2 are the studies with a diminishing effect over time. The red line is regular hazard ratio estimated from Cox model. The second line, the yellow line, is from the weighted hazard ratio using the weight chosen in the combo test. We can see here the weighted hazard ratio point estimate is smaller than the regular hazard ratio across all the 4 studies. But considering the change of treatment effect over time, piecewise hazard ratio could be more informative to understand the changes. So the next 3 lines represent the piecewise hazard ratio estimates in 3 intervals, 0 to 6 months, 6 to 12, and 12 beyond. For the left 2 studies, there was a negative or a small treatment effect in the first 6 months, and the treatment became more effective after 6 months. For studies 3 and 4, the benefit of treatment is larger at an earlier time, but decreases over time. The last one, the purple line, is RMST ratio. So we know that each summary metric has its own advantages and limitations. The regular hazard ratio has been commonly used, but under non-proportional hazards, it's difficult to interpret the results. The weighted hazard ratio uses the weight chosen in the combo test, so justification of weighting is needed. It's an average hazard ratio, also the interpretation could be a problem. When treatment effect size changes over time, piecewise hazard ratio could be more informative. But for that, we need to identify and pre-specify the intervals. The RMST estimate depends on the truncation cutoff time point, and the time point should be pre-specified, clinically meaningful, and cover majority of the endpoint outcomes. As a single value of cutoff time point is too fragile, we should evaluate the robustness of RMST estimate in a suitable range around the cutoff time point chosen. In addition, RMST estimate is affected by censoring, so studies should have adequate follow-up. With that, an interim analysis using RMST could be questionable. Milestone rate also depends on the time point selected, and the heavy censoring before the time point could affect the reliability of the estimate. In addition, we are concerned that estimate at one time point may not reflect the overall curve. So if we use the milestone rate, estimates from multiple time points would be needed to describe the curve. So in summary, the combo test is a powerful test, particularly for studies with non-proportional hazards. However, adequate clinical justification is needed for the weight. A weight only based on data is questionable. Although there are multiple choices for effect size estimation, each has its own advantages and limitations. So in the cases with the pH assumption holds, we should still use a hazard ratio from Cox model. However, for studies with non-proportional hazards, one summary statistic may not be adequate. We may consider using multiple methods to describe a treatment effect. If multiple methods are to be used, the study should consider that at the design stage in order to accommodate the requirements of each summary method. That concludes my discussion. Thank you.
Okay, great, thanks. I'm also told that Susan has slides as well. If you want to present those now, then we can get on with the panel. Okay.
Although they do overlap with the previous speakers, so I may skip some of them. I think, thank you for staying through the session. I know it's not easy after lunch, but hopefully this is going to be rewarding for all of us. So this audience knows about immunotherapy, and I can skip that. I think the four speakers in this session have done a marvelous job in going back and doing retrospective analysis based on the data. And we all know the different scenarios of non-proportional hazard in terms of either crossing survival curves or a delayed treatment effect. And we all know the challenges in terms of the analysis that log rank test is most powerful when the proportional hazard assumption is met. And as a fact, we know that immunotherapy is most often proportional hazard assumption is not satisfied. So I'm going to first focus on the usual measure in terms of what is the best, if we can say the best measures that will quantify the clinical benefit or the treatment effects, and talk a little bit about the test to use. I think based on what the four speakers have presented, I don't think there is a one-size-fits-all, but what is the biggest challenge for the statistician is how at the design stage can you bring in or make those decisions. So if you know upfront that the clinical trial you're designing, that there are differences, perhaps one approach is to use a weighted log rank test, specifically gamma family based on the Fleming-Harrington test. And again, you know, the key question is what type of gamma to use, because you want a larger gamma to basically indicate that you have a late effect. Luckily for us, there are not too many tests to use. I think the MAX combo definitely did demonstrate, based on some of our speakers, an advantage. Now I'm going to switch a little bit to estimation, and I think we've seen restricted mean survival times, and there were some consistencies in some of the examples. I think the biggest problem is how you interpret that, how you choose the truncation time. I think often the most challenging thing is not statistical, but clinical judgment, and this is where the truncation time needs to be clinically decided upon. We know that it is going to be affected by follow-up, so this definitely will have a huge impact in terms of follow-up time. And there are different approaches that I'm not going to spend a lot of time on how you estimate the RMST. I think, again, it's reasonable a priori at the design stage to say what you're going to report besides our usual hazard ratio. And I think I do like what Dr. Harrington mentioned earlier today. You know, we're all familiar with quantiles, so why not report quantiles at different time points? And then either the weighted hazard ratio or piecewise could be like a set of measures that are reported, and this is decided upon early on in the trial at the design stage. And the whole reason why I think it's good to report all these is just to show the robustness of the data, and also because you don't, I don't think one measure can do the trick. It's going to be sort of data dependent. So we all know analysis is driven by the scientific question, potential challenges and solutions. We know you are all have heard that, and the working group has come up with really some nice questions and potential solutions. So these are my initial thoughts. Thank you. I'm trying to stick to 5 minutes.
Great, thanks, Susan. So I'll turn to Eric and get some of your reactions to these test cases.
Yeah, thanks. So thank you to the organizers for inviting me to give a clinical, a medical oncologist perspective as well as a clinical trialist perspective. I think that both the discussion earlier this morning as well as the case reports, to me, make a pretty convincing argument that max combo seems an attractive way to approach this issue of non-proportional hazards. And I like the way that, in fact, doesn't look like you have to pre-specify the weights. It will do that for you automatically and, surprisingly to me, without much of a multiplicity penalty. So I guess to me it seems like why not employ it as the general test. I think for communication, again, there's been a good discussion. I do think that while it adds some complexity over a typical discussion of Kaplan-Meier and hazard ratios, that's never been an easy discussion to have with a patient. I think, again, a lot of clinicians probably struggle over what a hazard ratio means. So this has a little bit of complexity, but I don't think it's, it should be preventative in any sense. I do think that the question will come up, though, from clinicians and patients as to what's driving this. And I guess for me, I could step back and say, well, why would we expect there to be proportional hazards? Maybe they should all be non-proportional. So maybe folks my statistical colleagues can help answer that question. But I think that's going to be the question. And one of my perspectives, it seems when I've seen this, a lot of times it is driven by a subgroup. So what's going on is that it's a heterogeneous population, and you're getting non-proportional hazards because some people are doing really well on the drug and some people are not doing very well. And one of the comments I have just around the use of the term delayed treatment effect, to me, can be a little confusing because I think there's two different scenarios there. One can imagine a completely homogeneous patient population where the effect of the drug is the same on everybody, but for some reason it takes a long time for the drug to actually start working. I'm a little skeptical that could actually be true, but maybe, again, people can give me counterexamples. I think more likely is that delayed treatment effect means, again, that there's patients who maybe are sick or they have the wrong biomarker. They're not going to do very well on the experimental arm, and they'll die relatively early in the study. But there's another population that will do very well, and over time, they're the ones that drive the overall effect. And if they're in majority, then you have a positive study. So those were, I think those were my comments. And maybe the last one I have is just, I think that I know with my colleagues, Dr. Anderson among others, we get to some pretty sophisticated alpha allocation around some subgroup analyses. And it looks to me like this would not impact that, but I guess I just have a question. I assume this would be layered sort of over that, and this would not impact the way we've, again, allocated alpha for specific subgroups that we're interested in.
Thanks, Eric. You bring up a good question about whether or not the going-in assumption about having proportional hazards from the beginning should be that you don't. So that's interesting. Bob, thoughts from you?
Well, I certainly would like to thank you for the opportunity to comment on the results that were presented here today. I also wanted to commend all of the speakers, all the presenters on all of the work that's been done. It's obviously been a tremendous effort put into putting all these things together, but some very thought-provoking results presented. I think it was a lot of very good work. Do I think the proposed method is optimal? No, obviously it's not. It can't be optimal in any particular situation. There was a fairly extensive simulation study done a couple of years ago comparing the performance of 18 or 20 different non-proportional hazards methods, specifically in the context of crossing hazards. And the Fleming-Harrington 011011 methods were included, and they came out pretty firmly in the middle. And the procedures that performed best were procedures that were designed specifically for crossing hazards. So using the max test, you're not going to get optimality in any specific situation, but that's okay. You weren't looking for optimality. You were looking for robustness, but you have to keep in the back of your mind that in order to get robustness, you're sacrificing something in any particular situation. Do I think the proposed methodology is useful? I think the simulation results and the case study results all are very promising. I think that it would be very useful, especially in situations where the non-proportionality is due to some characteristic of the treatment itself, delayed onset, diminishing results, particularly when the non-proportionality is caused by some characteristic of the treatment effect. But I think also that, as other commenters have said, if the non-proportionality is more due to population heterogeneity, then maybe you should be looking at other things, some stratified analysis, stratified on prognostic or risk, benefits, or perhaps some more formal latent group or finite mixture analysis that's trying to look for subsets of patients with more amenable survival curves. There are certainly ways to do this. McLaughlin's book on finite mixture analysis has a whole chapter on survival distributions. They're mostly, if not all, they're based on fully specified parametric survival distributions, but in the context of an exploratory analysis to try to look for possible reasons why you're getting non-proportional hazards, I think that may be an acceptable trade-off. And this is it's not something that's unique to survival analysis. In any longitudinal study of efficacy of any kind, you're going to have responders and non-responders. And most overall analyses ignore that. And maybe it's good, maybe it's not, I don't know. When I think of the impact of trying to implement these procedures in the real world, for the real person trying to write up a protocol, the thing that strikes me the most is trying to write the statistical methods section of a protocol or the analysis plan itself. For the testing phase, things are pretty straightforward. I think you can pretty much see how to write up the details of how that's going to proceed. When you get to the estimation phase, things get a lot more hazy. If you're going to allow the estimation phase to be at least partly adaptive and data dependent, then what do you say in the method section? Is it going to be acceptable to just say estimation methods may include RMST at various data-dependent time points, piecewise hazards at various data-dependent time points, et cetera, or are you going to have to specify the actual characteristics of the results that would lead you to use piecewise hazards or RMST? And that would get to be very problematic, how to specify that in detail in advance. So it becomes, at least I don't see how to write up a detailed statistical methods section that provides those kinds of details on the estimation phase if it's going to be data dependent.
Great, thanks, thanks, Bob. So getting back to just the, in terms of the hypothesis test itself, it sounds like, and at least I heard from the majority of folks that, the MAX combo test seems to be a pretty good choice to use when you don't have the proportional hazards assumption met, but that it might be optimal when the nature of the non-proportionality, as Bob just mentioned, is due to the drug effect over time itself versus the population characteristics, although that might be hard to tease out in some cases. So thoughts from the rest of the panel on that and approaches from there. Do you agree with the sort of the overall conclusion that this combo test might be the right way to go? Even from the earlier presenters, the ones that did the actual test case examples? Any thoughts?
So maybe I could give a little bit of comments on my feeling on the MAX combo test. So I think I hear repeatedly people say this is maybe a good test. I think it depends how we define good or how we define optimal. I think if we define optimal, if we define reducing the p-value as the only criteria for optimal test, then obviously, as shown in these examples, MAX combo test sometimes, a lot of time that can be cut at the, not all of the time because we do have example, but sometimes we have the smaller p-value. However, whether this is only goal or how this would be weighted when we choose an optimal test, I think this is needs to be worth to be addressed and I think of because we wanted to test and measure to capture treatment effect, a good summary measure, rather than maybe only winner pick to get a test that give it the smaller p-value. I think this is also an important question. I think we need to think of when we design or design and write the statistical analysis plan.
Great, thanks, Eric. Yeah, so along those lines, at least to me, and again, this is my medical oncology clinician view. It seems to me the usefulness here would be if we use log rank on a treatment that's going to have non-proportional hazards. We don't know ahead of time, right? That's the problem, is that we can't know ahead of time, right? So, but if that turns out to be the case, then one could have, quote, a negative study, at least to me, with a drug that might actually be effective for a large part of the population. So it's a way to allow the study to be positive, to do further studies, maybe around what's driving the non-proportional hazards. Maybe there'll be some subgroup work that's pre-specified in there that can help with that. But that, to me, seems the attractive part to it, that you can avoid having a negative study in a drug that actually could be pretty effective.
Great. So we do have some time for additional questions or comments from folks in the audience, so go ahead and use the mics in the room, and for those online, you can go ahead and submit questions via the email that was provided earlier. Okay, go ahead.
Hi, Mary Redmond from Fred Hutch in Seattle. So I wanted to keep going on that topic of subgroups and along the lines of what Bob and Eric were just saying. So if you have, I guess my question is, do we want to be robust to crossing hazards when you don't know if there's a subgroup? Because at the end of the day, you're trying to make treatment decisions about how to treat a patient. So if you all you can say is that there is a difference, there isn't a difference, but the EGFR example is good in that you have an oncogene and a drug that targets that oncogene. And so once we found the subgroup, that was great. But in immunotherapy, it seems more complex. And so I'm just wondering, could we think a little bit more about, do we want to be robust to crossing hazards, or maybe we should be thinking about things slightly different in that setting?
Any thoughts from the group?
Yes,
Bob?
I think you want to be robust against crossing hazards, especially in a case where you don't know why they cross. So something like the max combo is a good thing. I just would like not to rule out any exploratory analysis on trying to find out why the hazards cross. I just think that those kinds of exploratory analyses could be very helpful down the road. But in any given case where you don't know what's going on, yes, you certainly want to be robust against crossing hazards.
Larry, did you have a
comment? No, I would just, yes, I agree. You need to be robust against, I think, all settings, which is the motivation for the weights, the family of weights we use, the 4 different weights for, those kind of 4 main type of scenarios. And then, I think the reason why it's the combination test did so well in the simulation studies because we pick those families so that they're fairly flexible, even though they're not going to be optimal in any specific setting. Each one of the tests themselves are optimal for what they're designed to detect. But when you have the maximum combination, you're trying to be robust. And as the simulation showed, the loss of power, even against the log rank and proportional hazards was pretty small. So I thought it was I think there's a lot of potential for it, and the robustness, I think, is really attractive.
Yeah, and maybe one other point to add, and I think Eric, you had brought up a really good point about whether the default should be NPH or not. And I'm not sure that's necessarily the case because we can find as many examples of proportional hazards just as examples of NPH. I think. And I don't think we're suggesting that this test be used as a default. So there needs to be careful planning at the time of the design stage in terms of making sure whether it makes sense or not. But that's why the choice of weights that were proposed were not like 10 different weights, but it was restricted to situations that are plausible, and especially in the early stages of development, you don't know whether you'll have crossing hazard. If we knew we would have crossing hazards, we would never, essentially do the study. We'll try to figure out which patients are benefiting versus not, right? So I think it provides hopefully a practical benefit by restricting the number of weights and looking at this test.
Any other comments from the?
Yeah.
Okay.
One criticism we've heard about about weighted log rank tests that emphasize the late events are, in the case of crossing hazards, you're discounting the early events. Why is that a reasonable thing to do from a clinical point of view? I will say that there are going to be many situations, the example that we showed, IO plus IO versus IO, where IO is now the standard of care, where you don't expect crossing hazards, where you expect a delay and possibly long-term survival. And then in that case, the combo test would be a very good choice, I think, in that sort of situation can be justified.
Go ahead, Larry.
I just want to comment again on the estimation part. I very much like the discussion about the heterogeneity part, and when you say you have a late separation, these Kaplan-Meier curves, that's just describing on the population level. So saying that it's a delayed treatment effect, like you mentioned earlier, it could be some patients just don't benefit, and some others have a very strong constant benefit. It's not necessarily that benefit only kicks in at a particular time point. It could be from the beginning of treatment. So I think, and when you have the weighted log rank Cox estimates that we described, those were the analogs, the corresponding Cox estimates corresponding to the weighted log rank test. So the weighting Cox estimates kind of just, in my mind, describe what the benefit is corresponding to the weighting. So if you have a weighting scheme that identifies, gives heavier weights later on, then that's identifying patients that potentially have, stronger later benefit. Then to me, when you look at the standard Cox model, that might be an overall treatment benefit. So maybe overall there's not a strong benefit, but maybe for a particular population that are deriving benefit, it's a much stronger benefit.
Great. So Raji, question from the audience?
So Raji Sridhara here. I have several questions. One is, in one of them, you presented the interim analyses. I didn't quite get when you said interim analysis with 20-month follow-up. Weren't you doing the interim analysis based on first X number of events where the interim analysis would have occurred, or am I understanding that incorrectly?
Yeah, so usually, obviously, as in the protocol, as in the publication states, they specify the interim analysis based on number of events. In the case of that particular study, it was 67% of the events. Part of that calculation involved 18 months follow-up. They've done the accrual assumptions, and that number of events would correspond to 18 months follow-up. So, sorry, thanks. That number of events would correspond to 18 months follow-up. So in this particular case, we set it at 20 months follow-up, more as an illustration of, what happens when you have less follow-up for some of these statistical methods, rather than setting this up as a formal event-driven interim analysis, more for the statistical purpose of illustrating the effect of less follow-up on some of these methods.
Okay. The other question, although we are sort of circling around this p-value, and I get what Eric is saying, that, if p-value is less than 0.05, then you won, and then you can argue about what subgroup, et cetera, et cetera. But here we saw some of the examples where in the log rank test or the Cox proportional hazards, the hazard ratio is 0.7, and with weighted one, you get a 0.5 or something like that. So, however, when we are approving, we are saying it is approved for everybody. It's not like it is this particular subgroup or what have you. Are we right then in saying that the overall, on an average, effect is 0.55 or 0.73 or somewhere in between? And so, although we want to kind of put the testing separate from estimation, it comes down to how do you then say what is the treatment effect? Yes, the test is saying there is an effect, but what is that effect? I think the decision of approval is based more on the latter. Yes, there has to be an effect, but what is that effect and is that meaningful? So that piece is equally important.
Yeah, great question. Eric?
Yeah, I can just maybe add some comments to that. So I think you're right, Roger. I don't think it'd be right to say that for everybody the hazard ratio is 0.5 in that case. I think, and this came up a little bit earlier, one of the other commenters spoke about it. I think, you know, you're going to have to explain it to the patient, maybe look at the picture to point out that there is, you know, for again, people who live long enough to live past a certain time period, they're going to do really well, but we can't tell you whether you're going to be one of those people or not, right? And this goes back to the, you know, the patients can ask, well, why can't you tell me that? But I think, you know, we don't know, right? And so all we can tell them is that, you know, there's a chance you might be on the wrong end of the curve if you're one of the people that dies early, but there's also, you know, a chance that you're going to be on the latter part. And I think, you know, typically, I think the way these things work, right, is that for a lot of patients, they are going to be on that latter part, right? So, you know, there's at least some hope that it's not you know, that there's little chance of benefit, for example, something like that.
So the other thing that's important kind of thing is if the treatment, you know, we are talking about the curves. Let's see just the visualization aspect of it. The treatment is always above or at the standard of care that you're comparing to. I think there is less to argue about. Okay, there is some treatment effect, but when certainly it is crossing. And when you know the treatment may be doing actually harm to some patients, how can a weighted hazard ratio kind of mitigate that process and still be able to use that?
Well, I think, and, you know, it reminds me, when I was thinking about this, is some of the discussions we've had with you on eligibility criteria, because, you know, to me, it's, again, you know, we tend to try to select patients early on that we think will most likely benefit. So we look for high performance status patients, good organ function. But it doesn't mean that patients that don't meet that, you know, they could still benefit, right? And if you put them all together, you could get, I think, something that could look like cross curves or NPH, but it could be coming, again, just from those sick patients that you allowed to get onto the study. So it's back to where, you know, it'd be great if we could identify the patients ahead of time, because we can tell them, you know, there's not, you know, you're going to do well, there's no concern, but we just, I think in some cases we just don't know, and so you're back to it's, to me at least, I'm not sure you'd want to not develop the drug because there's some patients in there who we could harm, right? Because the majority probably we're not going to harm. We just don't have a good way to identify those folks sometimes at the beginning of the trial. And I think some of this goes back to the discussions we've also had about retrospective subgroup analyses, you know, having a role, but it can get complicated because you're then into non-pre-specified subgroups with the multiplicity concerns, and then you're also sometimes in cases where it might be a you know, a test that's not you know, hasn't had a lot of analytical validity and it's not really ready to be deployed as a, you know, as an FDA-approved diagnostic test. So we can get into that complexity as well.
Okay, we're a bit out of time for this panel, but any quick thought from the rest of the panelists on these points?
I mean, if I can just make maybe one quick point on Raji's question in terms of if the survivals are crossing, you know, what does the weighted estimate mean in that time, right? I don't think it is also that helpful in terms of the weighted estimate. I think if there's a clear crossing of the curves, we, you know, relying on any single number to explain the treatment effect can get challenging. And I think at that time it may be good to look at the visualization of the curve and try to understand how better to explain it. I'm not sure the weighted estimate will be you know, any more helpful than the single estimate, unweighted one.
Yeah, go ahead.
And I also wanted to, I know we haven't talked much about interim, but also there's no reason why during monitoring of the trial that people can look at different quantiles in addition to weighted hazard and piecewise hazard ratio.
Okay, great. I'd like to thank all of our panelists for all of your thoughtful comments. We're going to go ahead and take about a 10-minute break, so back here for the next session at about 2:43.
Get started with our final sessions in just a minute, so I'd like to ask all of you to head back to your seats. It's been quite a discussion so far on a range of topics related to dealing with potential for significant effects of non-proportional hazards in these models for estimating the effects of treatments. So we've covered a wide range of issues across this set of topics, and now we're really honing in on kind of the final issues for the future. Where do we go from here? And this session, session 4, is on considerations for improving future trial designs. This is going to focus on how to design future clinical trials when, like we've talked about today, there is uncertainty if the trial will encounter non-proportional hazard issues and how to use the proposed alternative statistical approaches in this context. We have one presenter for this session, Kevin Anderson, who's the executive director at Merck. And then we'll have 4 panelists presenting or discussing this topic after Kevin's presentation. So first we'll hear from Kevin, then I'll introduce our panelists. Please come on up.
Okay, I'll blame any potential incoherence on this strange Philadelphia football game last night. The more relevant sports analogy is perhaps baseball, though I think I feel like you've been told I'm batting cleanup here. And in cleanup, you can strike out and everybody goes home disappointed, and what's the point? You can hit a single and kind of keep the game going, which I would be perfectly happy with, or you can hit a home run and clear the park and everybody goes home cheering, like the Eagles fans last night, which was awesome. So we'll talk a little bit about some pragmatic considerations for sample size calculations. And even though I don't particularly talk about, real specifics of timing of analysis, I hope I can give you some pragmatic feel for when you should look and when you shouldn't look, and what are going to be the problems if you look too early. Satyajit has been a wonderful collaborator, kind of throughout this, and did a lot of work. Larry Leon, who you saw in the last session, has done a tremendous amount of really nice work along with Ray Lin. Chris Gauss is somebody that is just, I count on heavily for statistical coherence at Merck. So she is another co-author. Oh, and I did want to acknowledge we had some contact with Takahiro Hasegawa, who's done some methods for sample size that we applied here, and a couple of my Merck colleagues who helped me with sample size calculations for the Lakatos method, for those who are familiar with that. Now, okay, let's see. So don't blame any of this on Merck, Pfizer, or Roche, Genentech. Anyhow, so I'll talk a little bit about, give some introduction and background, talk about power, sample size, and time. Time is a key thing, and I like to say some things that are a little bit different than what was brought up about event-based analysis timing. A little bit of a how-to guidance, and what I'm trying to do is to work with a single challenging scenario. So I'm not trying to solve every problem for you. I'm trying to say if this is what I'm concerned about, how can I deal with it? Then I'll talk some about interim analysis and expectations for the protocol and analysis plan. And so I'm going to start out by considering minimum follow-up time after enrollment complete when setting analysis time. So it's a variation on a theme of what you've seen earlier. So I'm going to assume for this design my control treatment has an exponential distribution with 7-month median. That may or may not be realistic, but hopefully you have good data to have some basis for some assumption like that. And like some of these cases, I'm going to assume there's actually a crossing hazard, that over the first 3 months the hazard ratio is 1.2. It's higher in your active group than it is in control. And then afterwards it's 0.5. And this is a pattern we've seen from time to time. You may not want to power for 0.5 afterwards. But 1.2 is not an outrageous early effect size for a crossing hazard scenario where you have one therapy you're trying to replace with another, particularly. If you're adding on to something, I think you're in a different scenario than I'm talking about here. But for this scenario where you might be replacing one thing with another, I think you can expect the mixture people have talked about, but we don't know in advance, for the reasons Eric said, what the diagnostic is, what the subgroup is. And so it is a common pattern to immuno-oncology. So here's what it is. So for 3 months, the red line is the exponential distribution with the median of 7 months. And then the blue line is my assumption for experimental therapy. It looks worse for 3 months. Now what's key is not that it's 3 months, but it's going 30% into the distribution. So through 30% of the distribution, you're doing 20% worse consistently. Now that could be over 3 months, that could be over 6 months, whatever. When you're doing rank-based tests, time really doesn't matter so much. It's kind of this percentage of the distribution you're hitting. So hopefully that theoretical alternative hypothesis curve is one where you would say that's a potential clinical benefit in the situation where we don't know what the subgroup is up front. And we would like to be able to design a trial where we can detect this difference. That's our objective. So I started out by doing the sample size incorrectly, but it ended up being an instructive sample size exercise. So I say, I'm going to enroll for 12 months in all these designs. And then I started out by saying, okay, I'll just follow for an additional 6 months, for 18 months total. That's the study duration. Or I'll follow for a total of 24, or 30, or 36 to get different minimum follow-up times of 6, 12, 18, and 24 months. And so here, with my incorrect sample sizes and stuff, you can see power calculations for the log rank in power.lr and for the weighted log rank combination or max combo test over in the right-hand column. I probably should have put power for max combo there. So you can see, first of all, that early on, for that 12 plus 6, your weighted log rank does substantially better. So that's not so much different what you saw with Kay's example when he did an interim analysis. And for all these examples, you're in that 80 to 85% range. So maybe my power calculation wasn't too terrible, except it was supposed to be for a log rank test statistic. So you can see that with increasing follow-up, the log rank will do better. So when you get additional follow-up, you're going to help the log rank by getting more observations out in that tail. But you're still pretty far away from the power of the weighted log rank, right? So do you want to go into this trial with a power of 85% in this scenario or a power of 60%, say with that last line, or 83% versus 54%? So that's your motivation for using the weighted log rank. You can get an answer sooner. You still don't want to look too early. Okay, so guidance for sample size. We kind of want to select the worst and best case scenarios. So I think the worst case in this case is what I just presented. You pick out something, you go talk to Eric Rubin or his corresponding person in late development, and you say, well, what bet do you want to place here? Do you want 0.5 after that break, or do you want 0.6? What do you want to protect against? Do you want to protect against equal hazard initially? Do you want to protect against 1.2? Do you want to protect against 1.4? And for how long? And so you agree with your clinical colleagues as to what's meaningful, and hopefully that picture suggests to you that it was a meaningful, positive difference between those populations to offer that new therapy as an option. And So I'd start with a log rank test just to select time frames for enrollment and follow-up, and try to get some window on that. You compute an adjusted significance level required for the combination test. This is a work in progress. Satyajit has done a lot, Larry has done a lot, and based on stuff in the literature, but for the design purposes, some of the stuff we did for the, with the correlation should extend here to be able to get that adjusted significance level for the design. So instead of, in the example I do, I'll design it p equal 0.02 for an alpha of 0.025, assuming that's the penalty I have to make. So that was just an approximation in this case, and we're working on getting the answer right. Then we'll compute sample size for each Fleming-Harrington test that's in the combination with that adjusted significance level. Okay? Now in this case, the 0, 1 uniformly is the most powerful. So if we take that sample size, we know we'll be well powered for 0, 1. Okay? But that also means we'll be well powered for the max combo because the 0, 1 is in the max combo. Okay? So the effort is to be a little bit conservative on sample size. And so once a strategy is selected, you confirm the design properties with simulation. The nice thing about having the asymptotic formulas is you can look at a lot of scenarios very quickly. Whereas if I'm running simulations for these, I was very aggravated when I was running the simulations because they took much longer than I liked to get answers. Okay. So, and nicely, Satyajit came across this Karrison article in the Stata Journal that builds on some earlier work, but very specifically described the correlation structure for the Fleming-Harrington test. And Dave may be able to tell us that that problem was published sooner and we should have used an earlier citation, but that's what we had. And it's very nice. So we can directly compute our adjusted significance levels. And I used asymptotic methods here for the sample size. You could, Lakatos method for log rank is what I used. We had that available, and it's in the package as well. Ed Lakatos did actually publish on weighted log rank, but we didn't have software for that. But Hasegawa did have that software, and so we modified his software a little bit because he was working with vaccine studies where you would just expect a delayed effect. In this case, I'm trying to protect for this crossing hazard scenario. But that was quite nice software and worked reasonably well. So here we said, well, if we do that 18-month study with the weighted log rank combination test, the max combo, you know, if it's an 18-month study, you've got 858 for log rank versus 384 for weighted log rank with the max combo. Okay? So obviously 384 is much more attractive in terms of, you know, getting the most you can perhaps out of your investment, at least in terms of power. And then you increase, and you notice that the number of events over on the right for the weighted log rank goes down as you get increasing follow-up. Okay, you don't need as many events if you're getting more of your events out in the tail. So now I can tell, you know, one of my Merck clinical colleagues who used to like to over-enroll, well, you were actually lowering the power when you were over-enrolling your trial because his analyses were based on event-based timing. So here maybe you decide 30 months is the optimal trade-off between time and sample size and events, you know, that it's something you can do in a reasonable amount of time and have reasonable power. And so you can see, you know, sample size reductions can be, you know, 38 to 55% with the max combo test. Nonetheless, you know, we still don't want to make this trial too short because regardless, you need to get data in the tail. So, so you do an asymptotic calculation. You follow it up with simulation to make sure that you believe your results at this point. And as we gain more trust in the asymptotics, I think you'll need less simulation. Now, one particular case I wanted to draw your attention to, and we've had to deal with this. This is not completely fetched out of thin air. There are trials where events come in faster than expected. Okay? And that means we're not going to get as much information in the tail as we had originally planned for. Okay? And that's going to substantially impact our power if we make our analysis completely event-based. We're shooting ourselves in the foot. So we spent time amending trials. I was going to stay here an extra couple of nights, but thankfully the FDA, at least the first time we've submitted it, allowed us to submit a design where we don't do analysis until we get both the required number of events and the required number of follow-up. And I won't go into exactly how we do the group sequential piece of that, but I can get that information to people who are interested. Oh, yeah. Yes. Yeah. So it's kind of a combination of a calendar-based and event-based. So you take the minimum of the two. It's actually kind of a design-predicted information versus the information fraction. And so not something that there's time to go into in this 15-minute talk. But let's look at what a trial looks like. These are the same 1,000 simulations. And I'm just cutting data at different points in time. So remember, I enrolled over 1 year, and then I follow patients for a minimum of 18 months. It's 30. So if I look 4 months into the trial, almost all my data is in that initial 3-month period where the hazard ratio is 1.2, right? So my one-sided p-values are all, looks like there's nothing going on. Well, they've got news for you. There is nothing going on. In fact, if anything, it's going on in the wrong direction. So if I'm looking that early, I'm likely to detect things that aren't there. And then as you go through this, you see the right-hand curve, the density in blue, kind of starts to squeeze down because it's weighting more in the tail, right, than in the early. So there's more and more data beyond that 3-month period, and it's weighting that more than the initial 3-month period. Whereas the coral or pink on the left has equal weighting throughout, and so it doesn't gain some of the benefit that the combination does. So, and I would encourage the FDA and my colleagues from other companies to take a trial you've done and do this exercise. And you may find a trial that, if you had looked really early, you would have been really uncomfortable about what to do with that trial. And it raises the question of, how patient can you be? And I think that's a great discussion item. This is just hazard ratios for that same set of simulations. So you can see at the beginning they're spread out all over the place. There's a lot of them between 3 and 0.33. There's a lot of random variation if you're looking early. And then you can see that on the left, the green, you kind of go, if you had done this trial as proportional hazards, it's powered for about a 0.72 hazard ratio. And you can see that green is kind of settling around that center. So you could say, well, if it were proportional hazards, it's somewhat representative. I think the average hazard ratio does mean something. Now, in this case, the blue, on the other hand, is lower, and that's emphasizing more the late. So these are kind of subtle things. I think the other descriptive measures we've talked about are nice, but in the context of this design, having these two, always having the Cox is useful, I think, as a contrast to the weighted Cox. And they describe two different things. Larry has, I think, summarized it better than I can. You know, it's kind of like, well, looking at that later, longer-term benefit, in this case, is more what the blue curve is, whereas the green is averaging overall. Okay, so those figures kind of drive how I think about early interim analysis, and I tell you, I don't have the answer. But you know, you would like to ensure some follow-up in a presumed period of benefit. So enrolling a lot of patients and exposing a lot of patients and very quickly getting early follow-up where you might have a lot of things going in the wrong direction may be a disservice to you, your company, and to the patients. Whereas if you collect them a little bit more slowly and follow them a little bit longer, you're putting fewer patients at risk, and you're getting a better idea if some patients may be benefiting later on. So I tend to design these things for, rather than looking for futility in the typical sense, if the true hazard ratio were 0.7, I say, well, I don't want to stop early unless there's a sign there's safety. So I'm putting my futility bound early is based on no difference, right? So I don't want to cause harm particularly. So that's an approach that I take. So further interims may be useful for possibility of early and late benefit, and I think Kay's example is great for that. Although ultimately, you know, the more follow-up you get, the better. And it raises another issue, you know, a bone that I would have to pick with some regulators is that, you know, we do a primary analysis, we establish statistical significance, but, you know, it may be that additional follow-up is actually useful information to get into the prescriber labeling to describe the benefit for patients. And there tends to be resistance to getting that in. So, but that's a point about, you know, somewhat post hoc decisions on how you describe benefit. That's just one more aspect to that. Okay, so the analysis plan in the protocol, the idea is that you're presenting a robust design, as I think several people have said. This is not optimal. In fact, I do want to make one point. An optimal design for crossing hazards would give negative weights to early events, right? Well, we're not doing that. And we would never propose that. And also one common suggestion is zero weights earlier. We're also not doing that. And I think one of the reasons I like some of the tests we have, but they're worth further evaluation, is if you use a larger gamma, which means you downweight early observations more, you may be going further into that curve. You know, I said you were 30% into the curve. Well, maybe with a gamma of 3, you're allowing, well, like 60% into the curve and still sensitive to something out further. And so that's the kind of consideration I would have for what test statistics we have, and is it still clinically meaningful, even doing some downweighting. So, okay. So, but final analysis timing may require both follow-up duration and event count, and I think I have an example of that. Estimation methods, so always present Kaplan-Meier curves and the Cox model hazard ratio estimates. I would also do a Graf-Thurneau test, but as noted earlier, it's not going to be particularly sensitive for a lot of cases. The max combo test is maximum that's not log rank, may suggest when it's important deviations from proportional hazards should be considered. And that capital MAY means I don't have a definitive answer here. I think the piecewise Cox estimates, and I think actually piecewise exponential, are quite useful for describing these distributions, but piecewise Cox is fine. I think having them either quantile or time-based is of interest. We've often specified in protocols that we're going to look at 6 months or 12 months for PFS, and we're going to look at 12 months and 24 months for overall survival. And I think those are meaningful for people, and I think they've often gotten into the label. So each estimation procedure has its own limitation. The most descriptive ones I think we feel wouldn't work well for testing just because going off a single time point doesn't work well. So under delayed treatment effect, designing with the proposed max combo test substantially increases power. I think we have some nice suggestions for how you can pragmatically approach this. Early analysis is problematic. You really need to walk through it carefully with all involved. And, you know, I've said to a couple people today, you know, you really have to appreciate people volunteering for these trials because, as Eric says, you don't know who's going to benefit and who's not going to benefit. And these people are risking their lives for these things. But it is ultimately providing meaningful data to make decisions for people in the future. And hopefully some people within the trial will receive substantial benefit. So asymptotic methods and simulation are both currently required, and analysis plans should present at least 2 relevant scenarios for which a trial is well powered. And while Cox estimates of hazard ratio may always have relevance, additional summaries are needed. Oh, and I did want to go back. I think I had something in one slide that I did not get to. Yeah, so if I do that 5-month median, if my control group is 5 months, so the events come in faster than expected, okay? Now, I lose power if I just do an event-based analysis. But if I wait both for the planned events and the time, the max combo in that case for that 5 months instead of 7 months still stays at about 87% power for the log rank. So I would urge the FDA and others to consider that there's more than just events that you need to get to where your analysis is done. And I guess the 5 versus 7 was 84% versus 74%, 75% basically. If 84% if you wait for events and time, 75% if you wait for the events only, and the average final analysis would be at 23 months instead of 30 months. So you didn't get out to that tail behavior adequately if you only look at events. And this is really driven by amendments we've had to write in recent months where we felt, you know, events came in too fast to really have a good understanding of what's going on with the distribution. So sorry I missed that when I was walking through earlier. But I think that's where I wanted to stop, and I have questions, but I think there may be other slides that people want to go through first.
Thank you. All right, thanks, Kevin. It's a great overview of these key questions about how we've had all the discussions today. How do we actually apply these ideas in critical issues of trial design? We've gone through sample size issues, follow-up issues, timing for looks. And now I'd like to get some other perspectives on these design topics and just ask our panelists to come up and join us. Kun He, Associate Division Director and Division of Biometrics 5 in the Office of Biostatistics. Mary Redmond, Associate Member in Clinical Biostatistics at the Fred Hutchinson Cancer Center. Elad Sharon, Senior Investigator for Cancer Therapy Evaluation Program at NCI. And Cyrus Mehta, the co-founder and president of Cytel. So, Kun, I'll start with you. Do any of you have slides that you're going to present? Okay. So maybe if we'll start with you in that case. If that's,
Well, thank you. I thank the organizers for inviting me. And I think this was an admirable work, a real team effort by industry statisticians in collaboration with FDA. It's just been really impressive. And I congratulate you on this excellent teamwork. I wanted to talk a little bit about interim analysis, since that was not part of the not really spoken about that much. How do I, yeah, okay. So the opportunities are like this, that interim analysis is routinely performed for safety, so I'm not talking about that type of interim analysis. But the thing is, what about interim analysis for futility or for efficacy? Is it even appropriate in a situation like this where the hazards are diverging over time? So it does depend on whether the survival curves separate out early or separate out late or don't separate out at all. So, for example, futility stopping, it would be definitely risky if the survival curves are separating out late. And Kevin has mentioned that he wouldn't perform at least for the first 6 months. On the other hand, efficacy stopping may be more beneficial because if you know from biological considerations that these survival curves are going to separate out later, and if you already see a strong signal at an interim analysis, why wait? You can get this drug to market earlier. You can benefit more patients. So a group sequential design with early stopping for efficacy. If a strong signal emerges early and you believe that this type of separation will persist, could be beneficial. On the other hand, you may not get as much scientific evidence as you would like from not being able to follow the patients for the full 30 months or 35 months or whatever. Now, the MAX combo does provide a robust final analysis regardless of survival curves. I like the MAX combo a lot. I do have a question for this team. That you did mention another method which is also robust, evidently, but was not used in this exercise. This method of Yang and Prentice is mentioned as a method that assigns adaptive weights to the survival curves, to the log rank statistic, actually. So whereas here you assign a weight based on the pooled Kaplan-Meier only, so it's a fixed weight. This Yang and Prentice method assigns weights based on the actual estimated ratio of the hazard ratios over time, and it specializes to exactly the log rank if there is no if the hazard ratio is constant, but it gives you benefit when it doesn't. So it should be considered, I think, a competitor to this MAX combo, and I just wonder why it was not considered. Now, and my last point is that either of these methods, certainly MAX combo, I thought it was a good method, but it could be refined, I think, by adaptive methodology. And I'll tell you what I mean by that. My next slide. What I'm saying is that if you look at, you can look at the data, and the whole essence of an adaptive test is that you can look at the data and react to it. Now, in what's been presented so far, you're not allowed to do that. You have to kind of right from the beginning provide as robust a test as you can without any opportunity to refine it. On the other hand, an adaptive test would allow you to react, to look at the data, and if you see that it's separating very early, you may not need that much follow-up. If it's separating late, you might need more follow-up. An adaptive test will allow that. There are some technical issues with it, and quickly, I don't want to take up other people's time. So I'm saying suppose you have up to capital J looks. Let's say typically J might be 2 or 3, and then you have these 4 Harrington-Fleming statistics, Z1, Z2, Z3, and Z4. Then what you need is the distribution of this multivariate statistic at different time points. And although it sounds like a complicated problem, it can be solved because What's the properties of that Z statistic? The correlation at any given look is known. That was what was presented by Satrajit. It is this Garrison method gives you the correlation at a given time. What about across time? Across time, it has independent increments. That was proven by Ciatis in a JASA paper in 1984. So you have something that's multivariate, normal. You know the correlation at a given time. You know the independent increment structure across time. You could exactly find the max of that statistic, which we've done in my group at Cytel. That has recently been done by Pranab Ghosh and us, how to do that computation. So we do have a way of doing adaptive group sequential with this max combo test, and I think that would be something to look at because so that gives you the distribution theory. Then there's also the issue if you do an adaptive test, how do you control the type 1 error? And there also there are methods available. These very flexible methods of Muller and Schaefer can be used for mid-course corrections by controlling the conditional error rate. And the advantage is that you can look at the data, make data-dependent changes depending on the nature of the survival curve that you're currently seeing. And I think these methods would complement the robust statistics that have already been proposed for non-proportional hazard settings, and they're statistically valid. We need more work and maybe extensive discussion with regulatory agencies. I won't talk about the estimation problem. A lot of people have talked about it. It's a really complicated, difficult problem, and I don't think one number is sufficient. I think many different options are available. Thank you very much.
Thank you.
Do you want to take just a second for a discussion of the Young and Prentice? Larry, I know you looked at that quite a bit. Do you want to comment on the Young and Prentice at all?
Yeah, so yeah, we may need you next to a microphone for this to work well. Thank you.
Yeah, so we looked at the Yang and Prentice adaptive weighted log rank test. It did generally quite well. The point estimates, though, the short-term and long-term effects were really quite biased. They did really quite poorly. But there was somewhat of an elevated type 1 error issue with the Yang and Prentice. And I had some correspondence with John O'Quigley. I forget what, he's in France, so some institution over there. He mentioned that he also is finding type 1 error issues with that test. And he's saying that their coding is actually invariant. Whether you code treatment as 0, 1 versus you reverse the coding, you get completely different answers. So it's a little concerning. Yeah.
You may be right. It was a difficult paper to read. I certainly haven't programmed it.
Well, the point about building in an adaptive component to these trials would seem to have some merit if you have so little understanding going in of the hazard patterns.
Yeah, I've tended to. Well, Mary, we were talking about. Do you want to comment on that? Or
well, Mary's going to get some other comments to make too.
Yeah.
Go ahead.
Well, Cyrus and I and others have this debate continuously. And the question is, how useful are the interim estimates of anything? And, you're designing a trial that's big because you need a big trial to figure out what's going on. So making guesstimates based on small amounts of data at the interims is not risk-free, whereas with the general group sequential approach, you do have the possibility to adapt small. If it's proportional hazards, you will finish early. If your hazard ratio is 0.6 from the start, you'll be able to stop easily in an interim, I think, as Cyrus excellently pointed out. But to me, the adaptation piece is something I've often struggled with.
Well, I'm glad we've had some good discussion already. I do want to bring in our other panelists. So, Kun, I think you're next.
Okay. Now we see the combo test and the regular log rank test. So one question is whether FDA should accept this combo test. So we use the regular log-rank test for a long time. You see all our approved are based on this log-rank test. So you know the FDA preference is we usually use a non-parametric test. So let's say even in other disease area, whether we do ANOVA, ANCOVA, or CMH, it's based on the central limit theorem. So theoretically, we know it's a correct test. So statistically, it's sort of model fitting, right? The bad part is for same data, different people can come up with different conclusion or different number. So to avoid that argument, you see, we ask people to pre-specify in order we have argument later. Pre-specify method is correct? We don't know, but at least we try to avoid argument. So during all those years, for our most approval, I would say the model is indeed in the pH, I said as we have the proportional hazard. We can see occasionally the model is not proportional hazard. So only recently we have these IO products. It's sort of systematically we see this phenomena. So now question is, do the regular log-rank test not valid anymore? Well, it's not optimal, but it still do it work, right? Think about how many trials failed if you use a regular log-rank test, right? Except we probably have a larger sample size. Then it come out question, what's the real gain we use the combo test? I think twofold. One is we save the sample size. So look, if it's not proportional, we set an NPH. So probably according to the simulation to the other work, the sample size may be reduced. Is it significantly less? I don't know. But definitely in certain situations, the regular log-rank test has a much smaller power, I agree. But you look at what Li Jun presented at 25 IO case, in that slide, is all for the IO products, but only few are non-proportional hazard, 7 or 8, right? For the other part, even it's from IO, the proportional hazard are still okay. Also, if we think of the sample size reduction, because for those trials, we don't have the first-hand calculation. Let's say we do the back-hand calculation. If we originally designed this trial use a max combo test, what is the right sample size for that trial? Then we use that trial data at that time. So do we have the same p-value? Let's say we didn't lose anything. So if we know that, that's really the one gain we have if you use max combo. But of course, if we compare to other disease areas like a cardiorenal trial, it's a 1,000, 10,000 trials. Very often, if you save a lot of sample size, it's really, really significant. Now, if we think our oncology trial, if we have two arms randomized trial, 400 is very common, even 300. I don't know, save 50 patients is a big deal? I don't know. So the other argument I think people say about this max combo is, I have no clue. Okay, I don't know whether it's proportional or it's not proportional. We have early events, we have late events, or even we say we have the middle events. So to avoid that, I just do this max combo to take care of the case. That's really, really nice, right? We have one thing to solve all the problem. I wish I have that, yeah. But of course, you have lose, right? According to that slide, among the 25 IO products, only 13 is like non-proportional hazards. So in that sense, for most part, regular log-rank tests are still quite good. Well, of course, you waste the sample size, I would say that way. So in that sense, I will ask how many trials, because we use the regular log-rank test, we do the proportional hazards, we failed from my experience, from all those submissions, probably all the trial, if drug is work, if you use regular log-rank test, it still get approved. You pass the trial. Of course, again, you waste sample size. Definitely, if we can reduce the trial size, it's really nice. But I don't know how long, as Kevin presented, you have the longer duration. I feel that from sponsors' side, has a longer follow-up probably is not in their favor, right? They wanted to put the drug in the market faster. Of course, in FDA side, we wanted to put a working drug in the market as fast as possible. That's why we do so many expedited approval. We have beat all the PDUFA deadline. You can see that. So, but the other things is, as I presented for the interim, we have the adaptive design. So you see FDA is now sort of very, very flexible now, right? I will not mention the company name, but lots of company, okay, they send lots of amendments. So from the beginning of a trial, so later on, they change the primary analysis population from RTT to a subpopulation. Later on, they argue, oh, it's not working. I switch it back. Then they switch primary and secondary endpoints from OS to PFS. And so, because we have another trial, PFS not great, I will switch it to OS. Okay, then they switch it off. Okay, then they have said, oh, because it's not good, I change it from 1% cutoff point to 25, then cut it to 50. I think, in a sense, I feel the trial, at the end of the trial, is really completely different than what they originally submitted. I would say that way. Do we always say no? People have experience, right? I think we are very, I would say very flexible. I think as long as you have the valid rationale to support your evidence, okay, because we are all human beings. At the beginning, nobody really knows what's going on for that drug. Let's say if later on you have another phase 2 trial finished, or even from other published trial, if you make the amendment, we think it's reasonable, right? We never say no. So in that sense, I will say, Eric has a consideration, said, oh, if I don't do this combo, I maybe fail a trial. So far, I didn't see this, okay? I feel people are very alert, okay, to what's going on in the field. They make the valid, or I not said valid, they make what in their favor to modify the trial. So the other thing is, you develop a drug, it's not a drug itself alone. You see PD-1, PD-L1, many of them are very similar, right? You borrow information from, let's say, Pembo, Nivo, it's all similar. So one trial is published, is out, so you immediately make certain guess, you make the right modification. So in that sense, I feel that not because you do the regular log-rank test is ruined your trial. I don't see it right now from my experience. Of course, I completely agree. If we have something can cover all the issues, it's great. Okay, but you have the trade-off. As I said, according to Li Jun presented that the 25 trials, majority of are still proportional hazards. So do we accept the combo test? I said, probably not right now, not immediately. So you see the regular log-rank test used for so long time. So all our approvals are based on that. So where the combo test really better than our regular log-rank test, yes, I agree, it's better. But we need a fairly comparison to all the approvals. So probably in the future trials, you can make we call the supportive analysis. In present the regular one, you can make additional paragraphs. So if you use this, we will do this event or at the time. At the trial end, you also present that result. You have the fair comparison with the regular log-rank test. Then you can argue how much sample size you saved. So what's the time you saved? What the p-value are different. If we have quickly get that experience, of course we can back calculate all the finished trial. Then we can ask the advisory committee to have the discuss, asking the pharma group to present. So probably you can shift that. But even if we shift that, the question is, for many other drugs, we probably still a proportion hazard. Again, even for the IO products, it's still, some of them still are proportional hazard. So it's really pro and con. So I'm sorry I don't have the firm answer to you. Yeah.
That's okay. Thank you for discussing those pros and cons in design. Appreciate that. Mary, next.
Yeah, thank you. So I wanted to provide some context as to what I'm doing here. I am at Fred Hutchinson in Seattle, but I'm also the lead statistician for the lung committee within SWOG, and I lead the LungMap trial, which is a platform or master protocol evaluating multiple biomarker-driven therapies and immunotherapies in currently squamous non-small cell lung cancer for patients who have previously received platinum-based chemotherapy. And what we've seen, we've been evaluating immunotherapies, but what we've seen of late is that the major unmet need is evaluating therapies for patients who have received both platinum and immunotherapy. And so what's the next line of therapy to happen next? And so our current endeavor is to expand the trial into evaluating immunotherapy combinations for patients who have received both immunotherapy and platinum. And so as part of that, we started launching this idea, I can't even remember, a year plus ago. And the first, so when we were trying to design the trial, we didn't necessarily even have drugs or combinations in mind, and we just started thinking about the best design in this setting. And as I started thinking about it, so well, what's the best endpoint to use even in an earlier phase trial? We know that response and progression-free survival do not really work very well as surrogate endpoints in this setting because you're talking about immunotherapy working on the immune system versus directly on the cancer within a patient. And so even in a phase 2, we thought about survival as an endpoint. But then we were also concerned about the potential for delayed treatment effects. And really, especially if you just want to see if there's evidence of a drug that's working, you don't want to miss those effects. And so I'll take a step sideways to mention that the first 2 companies that have come forward to work with us on the immunotherapy combination platform are Merck and Lilly. So I've had great opportunity to talk with Kevin, and Andreas Segeji at Lilly to evaluate pembrolizumab and ramucirumab in combination, randomization against standard of care chemotherapy. So our goal is to see, is there a benefit? And the question is, how do we look for if there's benefit? And when we started thinking about having to deal with that wanting to find signal, I thought about looking at landmark time points and other weighted time test statistics as well. And so independently of this lovely pharma group that came forward, I also evaluated and thought about looking at a combination test statistic. And so, so I'll talk a little bit about that in the moment, but I think we also want to think about and take a step back as we're looking at all of these things is what is the science, what is the goal? We're trying to find effective regimens to further patients' lives. And so we've been seeing non-proportional hazards, and there's a reason behind it. And we don't completely understand the reason. The reason is most likely that some patients are benefiting and some aren't, and some patients are benefiting to a different degree. And so it would be helpful if we could know more about that when we're designing our trials, but we need sufficient evidence or data to be able to evaluate that. So we're kind of in this little bit of a loop, and I think that's why we're having this problem here. Because in order to have sufficient power to look for subgroups and to look for the activity within subgroups, we need larger randomized phase 3 trials. The phase 2 trials with smaller sample sizes, definitely single-arm phase 2 trials, are really not going to provide sufficient evidence to be able to evaluate these things. And so how do we move forward, in particular when we don't have good surrogate endpoints? So I guess I plead to the scientists if we could do more to evaluate surrogate endpoints to measure biologic activity of immunotherapies, if we could pool data across different settings, maybe we would have more information that would go into the design of our phase 3 trials, and maybe this would no longer be a need because maybe we wouldn't need to do the weighting because we would in fact have clear separation of curves to begin with. So all of that is a background to just a few comments that I want to make on what's been presented here so far. In terms of study design, I completely agree with what has been stated earlier, where I think we need to get more data from less patients so we have a more complete picture, and relying upon just these huge studies with a fewer number of events and less follow-up, I think can be misleading. In terms of which test to use, so the test that I evaluated was just using a standard log rank, and then the G-row gamma with row of 0 and gamma of 1. So, and I like that test. I do have some questions about the combination test, and I would like us to do a little bit more to evaluate when the test breaks down. So what happens when we have less data out at the tails, and if there's noisier information? Are there scenarios when the combination test breaks down? And so I like the approach. I just think we could do a little bit more to evaluate the properties. And I think what would be really helpful is to have a simple calculator or something, thank you, that says what scenarios the test that you're using protects against and where you might be in trouble. In terms of effect measure, I think throughout the day what we've heard so far is that there's a difference between an effect measure that's being evaluated for scientific advancement of a study or a regimen versus a patient-physician discussion. That looking at time-varying hazard ratios can be beneficial in terms of understanding the biology behind what's happening, but when it comes to talking with a patient about the effect of a treatment, telling them that if they lift long enough that their hazard ratio is going to be decreased doesn't really make any sense. And so we maybe thinking a little bit more along those lines would be helpful. I do have to mention that I like so Mark Boyes and colleagues have come up with a measure that's called the net chance of longer survival, so the probability the random patient will do better with one treatment or the other. I think it'd be great if we could do a little bit more to evaluate that. In terms of interim analyses, I think yes, we should do them, but I think that the number should be limited. And I'm not clear how, I think we need to think more about the properties of those analyses, how we weight information. I think that we have to view that interim analyses with caution. And in particular, futility analyses, I think that looking for harm is important, but just null effects is not going to be very beneficial. It'd be great if we could come up with surrogates so that we could do interim analyses a little bit earlier to be able to assess harm in that setting. And at the end of the day, when we present the results, we need to be able to present a full story that looks at the experience of the treatments over time and potentially different subgroups. And as mentioned earlier by David Harrington, all of our approaches need to acknowledge the variation and the value of information that we have from our studies and the contribution to the bigger picture. And so at the end of the day, we need calculators. We need to be able to present information clearly, and I really appreciate all the work that's going on into this research question. Thank you.
Thank you very much, Mary. Next, Alad.
Yeah, thank you. I think that given the fact that I have probably less statistics background than almost anyone else on the stage, I'll probably be a little bit briefer. And I appreciate being allowed on such an these esteemed colleagues. I work at the Cancer Therapy Evaluation Program where I do immunotherapy drug development for the most part with colleagues from our biostatistics research program, which we feel provides us with excellent both advice and suggestions as to move forward. So we have about 900 clinical trials that we run across the U.S., and an increasing proportion of those are immunotherapy clinical trials as time goes on. And we work with Merck, we work with AZ, BMS, Roche, and all the major players in the field, big and small biotech. What I think strikes me, and I'm a medical oncologist, and I think what strikes me when we're going through all these discussions throughout the day is obviously mostly about how we translate these models and ideas to the patient. And I was talking earlier to some colleagues from the FDA. In fact, this is not the first time that patients have had to decide between therapies that potentially have higher rates of mortality or morbidity that occur early in therapy versus what may happen down the line. And I think particularly before we had some TKI therapies like imatinib, this was a common situation that we had to deal with in the CML population, where patients had a risk of transplant. Transplants probably had bone marrow transplants had a worse prognosis at the time as compared to today even. And patients potentially, especially in the early days, probably had to think about a 30% chance of mortality in the case of getting that bone marrow transplant versus what they might have, which is a cure down the line. So, that essentially really was the definition almost of a long tail. And we still have situations like that, even if we don't have that with CML. We do have that essentially today with AML, which is also a scourge, acute myelogenous leukemia, where you have patients that have to decide between transplants that may or may not be available. There are some places that will do things like haploidentical transplants, and they have generally okay results, I guess, or whether or not they want to continue receiving more and more therapies that may prolong life, oftentimes don't, and that's why we have such dismal response rates and 5-year survival rates for those patients. So when we're going through this, I think that the truth is patients do have, I think, an understanding, if we're able to provide them with the data from earlier phase trials, as to what those risks might be, not just on the trial, but also at the point of approval or bringing these therapies out into the clinic. We do have situations where we can say, okay, when we're offering this to you, we're aware that the early part of treatment might actually lead to this rate of mortality. We'll try to help you, or morbidity, we'll try to help you go through this, but at the late stage, we'll be a little bit further along, and these are the results. So I think that patients are probably sophisticated enough to understand that if we're able to glean enough information from those early phase trials and pay very close attention to what those results might be. The other thing, and I guess just to also expand on what Kun had said, is that no model is going to be perfect. I think Mary also mentioned this. And so it would be useful to focus and look at how one might actually certainly protect from the problems that can arise from a given statistical model that we're using to establish benefit. Without that, I think that a certain amount of skepticism should be used with anyone who's coming along and saying, I've got the best model that's able to provide us with all scenarios and all benefits and no real harm. And I guess the last thing, because I guess I don't have that much time, in fact, is really that that we have this scenario that we're dealing with essentially because immunotherapy is relatively new in certain fields. I think that when we get to the point where, at least within specific cancers, we'll have established therapies and we'll understand, and I think this is not that far along, that combination therapies with immunotherapy agents are essentially the norm. We won't have the problem of non-proportional hazards where one arm is actually getting immunotherapy and the other arm is not getting immunotherapy, because both arms will probably be getting some form of immunotherapy, and we might be able to actually revert back to something more similar to what we've been doing in the past. So I bring those up more just as food for thought, that we're almost in a little bit of a birthing process of this new era, as opposed to necessarily always facing this dilemma going forward.
Great. Elad, thank you very much for the comments. I thank all of you for bringing some very valuable perspectives on this important topic of design. We are quite pressed for time. I do want to get to the next panel. And just looking over the questions that were put up for the panel, I want to thank you all for addressing most of these pretty well, including interim analyses, early unfavorable trends, and pre-specification issues, adaptive issues, and so on. I would like to give you a chance for just quick final comments. And one that I don't think we really got to was a question of if there is more FDA guidelines or guidance in this area, any particular advice on what that might focus on based on what you've heard and thought about. And
Yeah,
Kevin, do you want to start?
I'm happy to jump in based on some of the questions that have come up. There's been some talk about, robust design in general. And usually what we've been focusing on is, do we know a possible targeted population? And if we do, we'll place a bet on that by splitting up alpha. But we've often seen we don't know what the target is. And so we'd like to place a bet on this general population. And in that general population, we probably guess there may be a mixture. And so maybe an ideal design for right now would be, okay, we'll place some bets on targeted therapy and one bet on the overall population. And for that overall population, we'd like to use this max combo test for the opportunity to potentially get something to patients more quickly that it looks pretty clear there's benefit, but we haven't figured everything out, I think, as Eric expressed pretty well this morning. And it would give us the opportunity to make those early advances and then encourage further follow-up to figure out how to optimize patients for our therapy for all patients. But in any case, when our clinical head for late development said, well, if the FDA liked this, would we want to do it for every trial? And I said no. And Chris said the same thing. And I think it is where you really don't know the answer, we might like to try this as an additional insurance policy.
Great. Thank you. Any other final comments, particularly about clear guidance on study design using this combo approach from here?
No guidance, but many companies you can see, in a sense, we accept your delay effect model because usually we ask the median difference, like for example in PFS, like 4 months or more. But in many trials, you said because delay effect, our median PFS like 2.5 months. We said it's okay because we acknowledge this kind of model. We will not look at the median difference. Rather, we will look at the end whether there is indeed a treatment effect. So the other things, use max combo is not means penalty free. I think one panel said because it's sort of all the statistics of the multivariate normal. But the only question is, this is really the non-parametric part. I don't know whether convergence is very fast. I think convergence rate must be very slow. So for the oncology trial size, at the end, whether those rejection values are very well defined.
Well, I think the simulations, at least for the cases we had, suggested it's really well controlled.
Okay.
And simulations also seem to verify the power calculations pretty well.
All right.
Yeah, okay. I would prefer, in sample size calculation, do simulation is fine, but at the final alpha, I would rather prefer not to have simulation because...
No, but we have both.
Yeah,
I
both.
think is the point.
Okay. Otherwise, it's very difficult to control. Yeah, I thought it can be theoretically adjusted.
Right. Yeah. And I guess the other practical point that you made is that the FDA is always willing to listen and to be flexible about issues like this.
We need to accumulate experience.
That's right.
So we cannot blindly say yes, right?
That's right. All right. I want to thank you all very much for the discussion this afternoon. Thank you. All right, and we are now moving on to our final panel for the afternoon, which is going to be co-moderated by Greg Daniel from our group and Rajesh again. So while they're coming up along with the panelists, and we don't have any opening comments here beyond those introductions, let me say just a couple of things about what I've heard so far. First, in terms of shifting the focus now to a broader range of stakeholders, away from just the design issues per se, but to considerations from the issues that we've talked about today in terms of barriers to implementation, how well these new approaches would be taken up, what unresolved issues or concerns they might have. We've got some very good perspectives for that discussion coming up now. So with help again from Greg and Rajesh, I'd like to introduce our panelists and then turn it over to them. So we have Samuel Wagner, Executive Director of Worldwide Health Economics and Outcomes Research at Bristol Myers Squibb, Saishang Chen, Head of Biostatistics for Clinical Pharmacology and Pharmacometrics at Bristol Myers, Mark Thierat, already introduced, Acting Associate Director of Immuno-Oncology Therapeutics in the Oncology Center for Excellence, Francesco Pignati, the Head of Oncology, Hematology, and Diagnostics for the EMA, who I think is joining us on the line. Hopefully we've got the technical issues worked out there, and Andrea Ferris, the CEO at Longevity. And just for one more word of introduction before turning it over to Greg and Rajesh, I think we heard pretty clearly today that people do want to continue to see those non-parametric Kaplan-Meier hazard graphs for studies like this, and they do want to see a Cox proportional hazards model and how well that fits the data. But you heard a lot of good discussion today about why there could be real benefits to this two-step process, including the max combo test if there is some suspicion of heterogeneity in the hazard models, but not a very clear understanding of which exact patients or what exact nature of the hazard could be addressed. In that case, I think we saw some good examples from both simulations and retrospective case studies suggesting that this model could be helpful. You also heard about a range of factors that might influence whether and how exactly to impose a test like this, what kind of impacts for sample size and time for study, and when interim looks might take place, and so forth. It would depend a lot on things like maybe some suppositions about differences in effects across patients, or about the clinical context in which the particular trial was being performed. For example, if making it to transplant was an important consideration, or if there's an expectation that there might be some particular long-term benefit. So all of those are factors in applying this, which would suggest that more guidance and more work, building on what this group has done so far, could be very valuable. But I do want to emphasize, as the last panel I think brought up very nicely, is that this discussion is highlighting that we are at a point where kind of crossing over between maybe what was adequate for many therapies around looking at a proportional hazard and a fairly straightforward model for a fairly broad population, to an era that's marked by a much better understanding of mechanisms. We talked a lot about IO therapies that don't always, but maybe increasingly may have a different pattern, not only of hazards over time, but also across different types of patients. And since we don't have a payer perspective represented explicitly on the panel, I can say that what a study like this would do maybe be enough evidence for good basis for approval of a drug, but does leave a lot of questions not fully answered about whether there are subgroups of patients for whom that higher short-term risk could be avoided in a model that shows some crossover, or whether the long-term benefits accrued through to some particular subsection of the population that could be better targeted for use of the therapy, or whether the therapy itself for these complex immunotherapies and others could be modified in some way to have more benefits for a broader range of patients. That all suggests that the kinds of evidence that can be developed in any single trial is not going to be enough as we keep improving our understanding of the science and the capacities of bringing together a lot more data to answer questions like this. As we've talked about in other meetings that we've done at Duke-Margolis, big data is not the same thing as big evidence, though. So I think it's very important for the careful work that's being done in this kind of analysis for potentially an early or even a pivotal trial to fit into other evidence that could be developed subsequently and that could help address the not fully answered questions that are going to come out of applying a method like this to what hopefully will be increasingly personalized and effective medicine. So with that as a way of a few opening comments, I'd like to turn this over to Greg to kick off the panel.
Okay, great. Thanks, Mark. Thanks for those comments. So I'm going to turn directly over to Sam. I think you might have some. Do you have some slides, just a couple, to get your thoughts? Thanks.
And as Mark is leaving, I just want to say thank you for being here right from morning and sitting through all of the heavy stat. Yeah, thank you.
All right. So. Obviously, I'm big danger here. I'm one of the people who stands between you and dinner and possibly an interesting beverage. But let me make a couple of disclaimers first. Obviously, I'm an employee of Bristol Myers Squibb, and statements I make here does not necessarily reflect what I think about the same issue. So I do have a clicker here. I just want to make sure I know how it works. The other disclaimer I want to make is that I'm not a statistician, I'm a health economist, and in some cases, I feel sometimes like one, but I'm not one. And other times, my colleagues, like Ty, who works with me at BMS, I think he gets into our area, and he may have a few comments about that too. And there's sort of this gray area, and I want to touch on a few points because the bane of my existence is to convince payers across the world why they should pay for our drugs. And therefore I have a team that are very competent, much more competent than I am, and has been very instrumental in getting access to patients across the world. So let me just give you a few things or a few ideas. It's kind of random. I want to give you a primer in just a few minutes on how we approach and how the HTAs think these health technology assessment countries. If you don't have experience of that, a 5-minute primer is going to make you an expert. That's actually a joke. I can talk for days about some of these issues. It's become very complex and it's become very complicated, especially in the field of immuno-oncologics. You've heard a few people mention that we're moving into an era where there's, in the future, near future, already happening, more and more trials will be IO against IO, IO combinations against other IO combinations, some new modes of action that's rapidly becoming kind of known since we've finished with some of the earlier trials, not only BMS, of course, a lot of companies are in that same mode. And we're looking for new signals, we're looking for new targets, and as these become available, from my perspective, I have to earlier and earlier understand what the trial design is going to look like, what the statistics in the trial is going to look like, because this does translate into pretty significant challenges at the time that we submit for reimbursement in many places in the world. So, without further ado, just a few points here. Cost effectiveness models are the key drivers, as you see there, of HTA and subsequent reimbursement decisions. When these treatment impact survival, as the case is always in cancer, cost-effective analysis typically involves estimating the costs and the benefits. Emphasis here would be over a lifetime period, and I'll get back to a little bit about that. Overall survival estimates are often the most influential parameters in economic models, and most HTA authorities that use these economic models have used mean OS in the form of the area under the curve. It's very similar to some of the previous approaches you've seen in the trial, on the trial basis. This does take the durable survival into account. However, to do that, we have to agree with the authorities how to do the long-term extrapolations of the trial data. We often have only maybe 18 months of data, and we have to my earlier point, we have to extrapolate this to either lifetime models. In some cases, it's a 5-year model, 10-year model, 15-year model. It's all over the place. There's no consistency really in it, and it can differ from, in the same HTA, from trial to trial. So the other few pieces that you need to just keep in the back of your mind: response to IO therapy may lead to dichotomous population, as you've heard also earlier, and understanding these long-term OS benefits separately in a responder and a non-responder population can be informative from both a clinical and reimbursement perspective. And then there was mention earlier also of subgroups, and we have the same issue. We have to deal pretty seriously with subgroups analysis, often in times, again, of incomplete or immature data with health authorities. I'll talk a little bit in more detail about the cure models in a moment, but as you can see there, and this is very, very standard, and this we do pretty much all the time. We decide very quickly or early on whether a standard 3-state health model will work for a specific submission, but more and more we're looking at responder-based models. The partition survival model and Markov model are two examples of the standard 3-state health model. Both are very commonly used in oncology in these submissions, although the Markov model does allow for easier modeling of subsequent treatments. Now, on the right-hand side, you'll see that the responder-based model, which is something that's been around but not widely accepted, we're trying to, with that, to attempt to reflect the disease mechanism, the differences in survival between responders and non-responders, and it does aim to capture the plateau effect by disaggregating these responders and non-responders. Related, but somewhat dissimilar, is the mixture cure models. Now, Megan Othis and the other scientists at Fred Hutch and University of Washington worked on this analysis, and I'll get into slightly more detail. This is the mixture cure model that we try to now get to the attention of many of the HTA authorities, and you can see that the probability of cure, background mortality not related to disease, and excess mortality is really the basis of this model. In our case study, in this initial publication or initial presentations, we used the standard Weibull model and the cure model, and you can see that the cure model fits the data there very well. It shows a slow decline in expected mortality. And actually, we did this with about 4 years of data. Some new data has come up to 10 years, and this model is pretty close to what we predicted if we consider the real world or the real follow-up analysis in the real data. This led to this paper, and this paper, the reason I bring it up here, it is very widely cited by now. It must have triggered some thinking, and several presentations at congresses has since been sort of the basis of, that's why I say it's a basis of many more presentations. It must have triggered something, as I said. Basically what this does, and this is to my last few points I also want to make, is that this showed that if you use this approach, the cured proportion or the cured fraction has a cost-effectiveness ratio that's very, very much acceptable to payers, versus if you look at both populations, the total population. About 347,000 versus 113,000 per quality-adjusted life year. As a result of some of these follow-up presentations, we've seen that in some of these, and this was the latest one at ISPOR meeting in Glasgow 2017. Pralay, who presented earlier, was also there. Very, very quickly, the two basic conclusions there was that the cure model and the responder models offer insight into the biomechanical mechanisms of action, but more data is required before we have any confidence in there. And there's a few questions, that came from the different fora, amongst others, the last one, consider KIR models in the protocol of your trial. So those of you that are interested and have the time and effort to do something really exciting is to go and look at some of these submissions, and you'll see how the whole problem of non-proportional hazards has been addressed, and these are just a few examples. Bottom line of this is that if you allow the non-proportional hazard, or if the proportional hazard assumptions are violated and the authority agrees with you and you don't do anything about it, they will model it out. They have their own modelers as well. And the cost effectiveness of your product will not be reflected in the correct way. Basically, the ICERs go up incrementally, and in some cases, like the NICE submissions, of course, NICE has thresholds. If you're over 30,000 or over 50,000 pounds per quality adjusted life year, they simply won't pay for the drug. Patients don't have access, and that's not a good thing. So just in the interest of time, I just refer you to these. It's just a Google, a few terms that you type in, and you'll get there very quickly. It's very easy websites to find. The NICE, I just put France in there because that is just to show that it's kind of universal, these discussions. And these were all related to non-proportional hazards, and how we addressed it in these submissions to get our drugs paid for. I think that's it.
Okay, great. Thanks, Sam. I'm going to maybe turn things over to Francesco from EMA. Francesco, if you're on the line, can you hear us?
Yes, I can hear you well. Can you hear me?
Yes, that's great. Go ahead.
Okay, thank you. Well, I'm very grateful for the opportunity to be part of this panel, because obviously we are dealing with similar issues. And generally, we're open to explore different approaches and to discuss pros and cons of different methods. And I would be very interested to see how our statisticians at EMA would look at some of the approaches that have been discussed here today. Now, we haven't done this formal consultation, so we might have to get back to that. But just personally, I remember when joining EMA, I was surprised by the fact that EMA was only using very, very, how shall I say, traditional survival analysis techniques like log-rank hazard ratio, and nothing about, I don't know, the multiple parametric analysis that could be done and so on. So although over the years I kind of rarely found reasons to deviate from the more traditional log-rank and hazard ratio approach, I think we should always be open to see any whatever techniques that can complement what we do. But even after listening carefully today, I must say I have not heard good reasons to deviate from our, let's say, more traditional approach, at least in general. Now, from my experience, I must say that whenever some type of weighting approaches have been proposed by companies, either in the design stage or in the evaluation stage, well, they have often met with quite significant skepticism for reasons that have been already mentioned. How can it be justified from a patient perspective that certain events are more or less important than others? And perhaps some of these approaches have their merit in secondary analysis, exploratory analysis, but definitely when it comes to primary analysis, this is the critical stumbling block. I also don't understand sometimes, let's say, testing and estimation, testing of hypotheses and estimation of effect today has been referred as almost two things which are totally independent. I think the appropriateness of the test and of the estimation should start from something different, which is really what are the objectives of the trials, what is the choice of the estimate, and then everything should follow coherently. And perhaps one of the beauty, again, of log rank and hazard ratio is precisely how complementary they are with this respect. Now, under non-proportional hazards, of course, things are much more difficult in the sense that a simple question like how does drug A work compared to drug B has no simple answer. And this is something we have to live with, and we have to perhaps learn how to deal with this heterogeneity. And perhaps some of these techniques are helpful to explore this, but it's a heterogeneity which not only has to do with how different people perhaps react to the drug, but also one needs to take into account that there are multiple effects that the drugs have, and that people value these effects differently. And so I'm very much for all the many speakers who have advocated that there is no single metric that is often sufficient to describe all the data, and that the starting point should be the Kaplan-Meier curves and something which is often forgotten, the uncertainties which are related to the survival curves, and then a number of other metrics, but over-reliance on a single metric can be quite too simplistic. That's one of the reasons why also some of the more sophisticated metrics like QALYs or perhaps some of the clinical benefit scales that have been recently proposed for me suffer of an oversimplification, which might work for some stakeholders, but I think when we are to communicate to patients what are the benefits and risks, of course, of drugs, we need to always look at the bigger, broader picture. So a lot has been said about heterogeneity. How to deal with this is a good question. I think we don't have all the answers, but I think drug developers should be prepared, aside from exploring clever statistical techniques that may increase our power, also need to think that we should be prepared for much longer follow-ups. Too often we see that the follow-up is too short to answer the many questions which come after approval, and also subsequent biomarker studies which are often only starting at the time the drugs are evaluated for approval. So we need to a little bit think harder about, let's say, a longer and more comprehensive follow-up and development overall. So now these are just my personal view. I cannot comment on behalf of the EMA on the validity of some of these approaches, but I can definitely say that we welcome the discussion very much, and I thank you again very much for inviting me to this panel.
Great, Francesco. Thanks for those comments. I'm going to turn it over to Mark to get some of your thoughts and considerations from what you've heard so far today.
So I think this, the test. So with FDA, you're very interested in getting, drugs out to patients. As soon as it can be concluded that the drugs are safe and effective under prescribed conditions of use. If there's a technique, to make that conclusion earlier, that's obviously a, good thing. Some of the implications I think may not have been, discussed from a regulatory perspective, and really thinking about both evaluation of treatment effects, but also the, the understanding of those treatment effects, kind of relate to oncology being a different therapeutic modality. So often in oncology, we'll only have one, trial to evaluate a potential treatment effect, such that, within that one trial, we're often looking not at just one endpoint, but multiple endpoints in order to support that there is a true treatment effect with that product compared to, standard of care, for example. So it's interesting to see how these different analysis methods may impact on that assessment of a treatment effect, such as from a single trial, oftentimes, other than standard clinical trial techniques that increase the robustness of the evidence, but the statistical persuasiveness of a treatment effect on a single endpoint, for example, such that, how does these different analysis methods impact the p-value to make that, determination. Also, thinking about regulatory pathways, we have different pathways for approval. So we have an expedited pathway for approval, being accelerated approval. And various endpoints in oncology may serve as both endpoints for regular approval or an accelerated approval. And oftentimes, such as for PFS, the magnitude, of the effect may impact that clinical determination of whether that is sufficient for regular approval versus an accelerated approval. So, think about those types of impacts on the different analysis methods, would be something for considerations in the discussion. And I guess just lastly, the curves are extremely important, and in labeling discussions, we're always very attuned to putting the most important information, trying to summarize the most important information. And oftentimes, especially with immuno-oncology products, there's a lot of consideration to putting that Kaplan-Meier curve in, not just for the primary endpoint, for example, in time to event, but there's situations where a summary of PFS, for example, may not tell the story. So there are situations where we don't follow the standard, which is usually putting one Kaplan-Meier plot, for example, for the, for an overall survival curve, for example, but we'll also put in several Kaplan-Meier plots for the other endpoints because that tells, much more of the story often than the summary, of the estimation of the effect.
Great, thanks. Ty, I'm going to turn to you.
Absolutely. As a co-lead of this working group, I just want to take this opportunity to thank the entire team, the great work in collaboration with the FDA. So, first of all, just a little bit of personal reflection. I remember when I joined the industry many, many years ago, the type 1 error rate was introduced to me as the risk for the regulators. And the type 2 error is the risk for the industry, the clinical trial researchers. So obviously, from a regulator's perspective, they don't want to miss. They don't want to introduce a drug that is not working. But from a company's perspective, we don't want to miss a drug that actually works. It's gratifying to see that we're actually getting together. This is no longer an issue. This is a risk for the regulators, for the payers, for the companies, the clinical trial researchers, and most importantly, this is a risk for the patients. So this is just a little bit of reflection that I have. So there are two common threads that I want to make sure that we don't get lost here. Well, so before we get there, I'm just thinking about this morning. Professor Harrington mentioned about sharing the pictures of survival plots with patients' loved ones. I think to me that's really important. And you may not have the plots with you all the time, but to me it is really important that if you don't have the plot, and you have the summary statistics should allow you to recreate the plots, right? If you are not able to do that, that means that you don't have sufficient information. If you think about this in the scenarios that we talk about today, you have hazard ratio, you have median survivals. You think about this, and you think, okay, well, I only have these two numbers, then I can just draw the lines, assuming that it's a proportional hazards, then you're not going to see the plot that actually, that you actually have. So that's extremely important. Using this guideline, I think you should be able to, to help you understand what information you actually need at the very minimum. Okay, so coming back to the commonalities between the health authorities and also the HTA, the Health Technology Assessment, which is the focus of this session. I just want to make sure that these two common threads are not lost in the presentations. First of all, is the importance of identifying the right populations who benefit from the treatment. I think Eric mentioned about this, and also Mary as well. I think more research needs to be done in order for us to identify the right populations who actually benefit from the treatment. And Sam talked about this in responders versus no responders. And that was just an example, right? I think the purpose of that exercise is trying to identify who actually benefits from the drug. The resource is finite, right? But the need is, it's infinite. So we need to figure out something, in order to make the system work. The second point I want to mention is that one of the most important metrics that's being used in health technology assessment is ICER, right? It's incremental, cost-effective ratio. It is a function of quality, right? The quality adjusted life years. and also the life expectancy. And life expectancy essentially is the restricted mean survivals, or the mean survivals essentially is the area under the curve. Now, when you have a curve that goes down to 0, you can get that number. Now we are facing an issue that the curve is open-ended and it goes forever. If you do not have sufficient follow-up, that's a problem. And this is one of the most important common threads that we're talking about here, is that thinking from an R&D perspective, from a regulatory perspective, we kept talking about needing to have long enough follow-up. It is also the same for HTA as well. If you have a long follow-up, what happens is that any extrapolation is going to be more accurate. And I think this is one of the most important that I just want to make sure that we don't get lost. Thank you.
Great, thanks. I'll turn to Andrea.
Hi, I'm Andrea Ferris from Longevity. I feel like I'm on the Sesame Street segment of one of these things. It's not like the others. So I'm from Longevity. I'm a patient advocate. I've been at this for 10 years. By way of background, I actually have a finance and decision sciences background, and I only tell you that to say that I'm comfortable with numbers. So when Raji asked me to speak on this panel and to be part of this conversation today, of course I said yes, because you don't say no to Raji, but then I immediately went to Google to figure out what the hell a non-proportional hazard was. So when that failed me, I went to our world-renowned and esteemed scientific advisory board and clinicians who run clinical trials and asked them what a non-proportional hazard was. They too did not know what a non-proportional hazard was. So I no longer felt quite so stupid and out of my element. But I bring that up only in to say that a lot of the dialogue today, very interesting, and I'll come back to that, but we're kind of talking inside baseball here. The average bear in our scientific advisory board are from academic cancer centers. Where our patients are treated, 80% are in the community. So if our academics don't know what it is, forget about the person in the community. So where this will come back is when we get into the communication element. The dialogue today to me was very enlightening and very interesting, but also hugely gratified. I'm very grateful that you all are tackling this, because I think from a patient perspective, the most important thing is that the way that we look at data, gather data, analyze data, I mean, these people are giving you their time, their energy, when they don't have anything. I mean, that's the only thing that they can't buy and they don't have much of, is to make sure that we get it right. So to say that a treatment failed because we didn't design the trial correctly or we didn't analyze it correctly, that is the biggest criminal thing from a patient perspective. So I applaud you in the efforts of trying to now match the analysis to the new treatments that are available. In terms of communicating, I don't know that we need to show anybody a graph. I don't think they'd have any clue. They just want to know if they're going to live or die, quite honestly. I think to be able to say, as Eric so eloquently said earlier, is to explain to a patient of, it might take a little while before we know whether it's going to work or not, but we think that there's a high probability that it may work for you. I think that's a great thing. I don't know that throwing in terminology like hazard ratio or the plots or anything like that, unless it's going to help a doctor understand what that means and be able to communicate that, I think that we need to create sort of a middle level. There's the analysis piece, there's the what goes in the label piece, and then the here's what you should say to your patients piece. And I think that it's very important that we not overlook that.
And just saying, in terms of the discussion about the Kaplan-Meier plots and label, that's really for the physicians, to really understand, because it's very important to understand the, especially with the crossing over issue.
You're assuming that they're going to know. So the people in the community may or may not understand what that means or the implications of that.
Right. Not regarding non-proportional hazards, but just, the plot to see, like you had mentioned, it may take a while, for that treatment work. Simple concepts that you can actually gather from the curve is, very important to have
or
the curve.
spoon feed the talking points to them to interpret the curve on their behalf.
Absolutely. And in terms of the risk-benefit assessment for individual patients and for populations is very important in terms of how those curves look like as well. I think Elad had mentioned the issue of transplant. And there's certainly other clinical situations where there are therapies that have very long tails that were approved, but had very high toxicities. If you can explain that potential toxicities, but what are the potential benefits? It's a very important piece of information to have, when making a risk-benefit decision for that patient, make that individual risk-benefit decision with their physician.
Okay, great. So I do want to save some time for the audience. If there's anybody that has a burning question, to go ahead and go to the microphone. But before then, Raji, do you have any questions for the panelists?
So I think, I thank everybody on the panel to be here, and particularly Andrea, standing up and saying, yes, I will be there. Thank you so much. And of course, Francisco, even though it's odd hours in EU, he's still calling in. He tried his best to come, but he couldn't be here personally. So I had just a couple of things. Basically, Sam, I want to ask if this combination test is used, how would the HTA take this? And would this be something that they would consider at all, or would you see some problems with it?
So, the short answer would be is obviously hope so, right? Because it helps to standardize some of these analyses, and if I had more time, I could spend 2 days on showing where all the different directions that they can go in trying to. They really want to pay for the drugs, but there's only so much resources that they have, and there's often these trade-offs that they have to do, and that's a reality. So we try to help them and give them the answers that they need as much as we can. But to your question specifically, I think this is Sam's opinion, that the FDA is a respected body worldwide, right? And the EMA will look at what you're doing here, and if we do it right and we do it successful and you justify it and all this discussions, and I really listen to the, like I said, not a statistician, but I know that the implications of this for my work is going to be pretty, potentially, pretty significant. So, bit longer answer that you maybe wanted, but yeah, I think we'll get, once we know that it's going to happen, and once we get more clarity about the questions that was raised about limitations and so forth, we will go and sit down with NICE and with Haas and with GBA and all these authorities and talk to them about this and do what we typically call early consultation and say, this is going to be how this trial is going to be evaluated. Can you help us to get the right dossier to you so that you can pay for this?
Francisco, are you still online?
Yes, I'm still here.
So, we had a lot of discussions and the delayed treatment effect, but also the crossover and the sort of thing. What do you think when you see a crossover type of study and you see this? How would the EMA react to that? And in that case, if a combination test is used and they do show that there is a significance there, what would be the reaction?
Well, I can't say that we have a one-size-fits-all type of solution to this type of problems. We are, again, open to look at different approaches, explore what makes sense, and try to make the best out of it. If you think of it, this is a lot of what drug evaluation is all about. Yes, you have your pre-specified plans and statistical analysis and so on that we might have agreed or not and discussed in advance. But then the reality is sometimes very different, and we kind of have to deal with that. Now, on the issue of crossover, on the issue of non-proportional hazards and so on, we have no, how should I say it, single approach that we would today recommend that is different from, let's say, the more traditional way of doing things.
Thank you. And Andrea, to me, that's the toughest thing to explain if I were to explain what is this curve doing. So let's say we show the Kaplan-Meier curve or the survival curves to patients where it is crossing over, meaning that at the beginning the control does better and the treatment may be doing harm, whereas in the longer run the treatment may be doing better. How would the patients kind of take that and absorb that, and do they really understand the significance of that crossing?
I think a lot of it is predicated on the patient population. In the lung cancer space, it's a very vulnerable and it's a very acute disease. So my guess is most patients, they don't even want to see a curve. They just want their doctor to fix it, right? And so you're probably not going to be pulling out curves. It'll be more of a conversation of, look, there, again, as to Eric's point, that it's going to be something that we think may have a long-term effect, even though in the immediate term, you might not see or feel differently right away. But I would be highly surprised, with the exception of a very small % of the patients that would ever want to see a curve, to be honest with you. So I think it's more the dialogue that we need to help facilitate how the clinician can have that conversation with the patient.
So here, today we discussed a few of the ways of reporting, even in our product label or what have you. Did any of them sort of stand out for you? We had this area under the curve, the life expectancy kind of thing, or you had the milestone, like, okay, we present a 12-month, a 24-month, etc., the survival rates. Would they make better sense than putting a median survival, for example?
Yeah, that actually resonated quite a bit, Raju, when you said that at the beginning. It's not something that I had really considered, which is using the methodologies that we're doing now, we might be misrepresenting the information, which is a big problem. Mm-hmm. And so I'm not a statistician, so I don't know which is the right curve or which are the right, what's the right information, but to present the information that is reflective of the underlying treatment, I think is the best that we can do in the label. I don't have the answer of which is the right way of doing that, but when you this morning said that we were currently misrepresenting it by using older analytic methodology on newer treatments, that was horrifying.
Any questions from the audience?
I just have a comment in terms of these different measures of efficacy. The one that's probably the easiest to explain and as a clinical benefit is the area under the curve, the restricted means survival time, because people can. There's a clinical explanation. It says that in this population that is being followed for 30 months, if you're on the treatment arm, your average, on average, that population will live 6 months longer than the population on the control arm. So and that doesn't matter whether there's crossing hazards or whether there's non-proportional hazards. The hardest thing to explain to any physician, to any non-mathematician, is what is a hazard. It's first of all not a probability. It's just it is a mathematical construct, and a hazard ratio that changes is really very difficult to explain. And an average hazard ratio is an impossible thing to explain.
Great, thanks. So that takes us to the end of this panel. I'd like to thank our panelists for all of your contributions and perspectives on this. I'm going to ask you to go ahead and stay seated as we wrap up the day. So what came up on this panel, and I think consistently throughout the day, is that, and we heard this many times, there's not a one size that fits all, and that context does matter. We heard clearly that when you have proportional hazards, the traditional log rank is tried and true. When you don't have potentially this max combination statistic is a good thing to continue to look at. But then the question of treatment effect and how to interpret the treatment effect came up with what we really need is maybe a framework for thinking about the different statistical approaches or interpretations of the treatment effect, and a combination of things and a framework that might guide that would be really helpful, as well as considering how other stakeholders across the system, from payers to patients to providers, might understand these approaches and understand their impact in terms of the decisions that they're trying to make. And I think Ty and Andrea really brought up the questions that really matter most, which are which patients should be targeted for these types of treatments, which patients are likely going to see a treatment benefit, and also what does this mean in terms of survival and longevity are really important questions that shouldn't be overlooked. So thank you to all of you for helping us with these tough questions and providing some input. I'd like to thank all of you for sticking with us throughout the day in person and online, because we did manage to cover a lot of ground on some really tough issues, and a lot of work has gone into understanding the current statistical approaches, developing new ones, and then testing them with some real examples. I'd like to thank our colleagues at FDA who made today possible and helped guide the event. Rick Pazder, Raji, of course, Robin Bent, who did a tremendous amount of work to make this meeting a success. I'd also like to thank Renee Iacona and Tai for chairing the Cross-Pharma Working Group and for all of their contributions for planning this workshop, not to mention the members of the working group themselves who have put a lot of energy and time into developing promising solutions for this tough challenge. Lastly, I'd like to acknowledge individuals at the Duke Margolis Center who helped make today possible: Morgan Romine, Meredith Freed, Elizabeth Murphy, and Sarah Saprisi. And thanks to all of you again. Enjoy the rest of your evening. Thank you.